If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly.
Okay. I'm just gonna go. All right. I think we might be ready. Take a moment. Okay. In just a moment, we'll go ahead and get started. Okay, if we're good, then in that case, all right, welcome back everyone to day 2 of our Generic Drugs Forum, GDF 2026 here on the White Oak campus. Good morning, good afternoon, or good night, depending on wherever you are joining us in the world. We do appreciate your commitment to attending both days of this event and also overcoming, I'm sure, busy schedules to be here with us. For those of us that are with us here today in person, please note the Wi-Fi that is on the screens around you. There's no password. You'll just click an acknowledgement button to join. Now, yes? Okay, can you hear now? Okay, thank you. So my name is Corey Adair. I'm with the Small Business and Industry Assistance Program, which operates within the Division of Drug Information here in FDA's Center for Drug Evaluation and Research. SBIA is also joined today with our colleagues from the Office of Generic Drugs, as well as the Office of Pharmaceutical Quality. Please make the most of today's experience as always. Interact with the subject matter experts, attend sessions that interest you, learn as much as you can, and also submit your questions to us online so that we can have a very robust Q&A session as well. Please note this session does also offer a YouTube live stream, so feel free to share that link and invite your colleagues to join in as well. Now, this slide provides us QR codes for different ways to follow SBIA on social media. We will also put up this slide during the breaks, and you can use your camera on your cell phone to scan the QR code. Following SBIA is a great way to keep in contact with us, be aware of our upcoming events, and see our most recent posts as well. Okay, now let's turn our attention to some logistics for today. These apply to all attendees, both online and in person. First, this will be your link to the one-stop shop for today. This will have all the answers to your technical questions, including downloading files, accessing the survey link to obtain the certificate attendance, and more. For our online attendees, this is also the same page you used this morning and yesterday to access the event. So please bookmark that page. You may need it today. And again, if you have technical questions, please check here first. You'll likely find your answer here. Now next, there are going to be session evaluation links on this page. Those links were activated yesterday to complete the session evaluations. We do want to bring awareness to you that FDA is currently experiencing an email server issue that sends out our thank you emails post-event. And as such, some of you may not have received your thank you email yesterday evening. We are going to send a cumulative thank you email tomorrow. Once this email is this issue is resolved, it's expected to be resolved as early as tomorrow. We will also extend that usual 2-week deadline as needed if there are additional delays. But again, you can also just go straight to this link and that session, and that link is also live for you there. Now, upon completion of the survey, you can download your certificate of attendance, which is used in support of RAPS, SOCRA, and SQA credits, and ACRP as well. The trick is that you have to download the certificate immediately after completing the survey. Otherwise, that opportunity is lost. Once the server issue is resolved again, that survey and evaluation link are only available for 2 weeks. And so that will be May 7th, typically from the end of today. Next, recordings of the individual presentations will also be uploaded to the event webpage once available. But in the meantime, once those recordings are available, we will post the YouTube live stream link as well by the end of today. We also have the speaker bios available on the One Stop Shop webpage. And then for our online attendees, please submit your questions with the on-screen Q&A pod. We'll answer as many as time allows. For our in-person attendees, again, there's the Wi-Fi information for you. As you saw yesterday and for today, when the Q&A begins, you will line up at the 2 microphones that you can see here in the middle of the room. We only answer questions that pertain to the subject matter of today's presentations. And again, we are unable to answer any application-specific questions. A quick reminder for everybody, check your cell phones, make sure that they're silenced for today's presentations. We also do recommend that you use the internet sparingly today. It can impact our ability to provide that online delivery if everyone's using really high-functioning internet at the same time. And lastly, just as a small personal note there, for lunch, if you pre-ordered, please pay at the kiosk during the morning break. It helps facilitate the pickup time faster. and bathrooms can be found if you go out this main corridor over here to my left. You're going to go past the registration desk where you checked in, and then they're just around the corner there in that main hallway. Now here, we do also have a quick note about some of our upcoming events. SBIA invites you to join us first on May 5th for our OTC Monograph Drug User Fee Announcements, or AMUFA, Understanding Fiscal Year 2026 User Fee and Registration webinar, as well as on May 19th and 20th for the Regulatory Education for Industry, Ready Annual Conference 2026, which will focus on the innovative regulatory strategies to advance medical products. There are 3 tracks for drugs, devices, and biologics, and our plenary features all 3 center deputy directors. My co-host for today is Captain Brenda Stoddart, SBI Program Director, who also moderated yesterday's event. So thank you, Brenda. And with that, we can go ahead and start with our first session. First today, we're going to hear from Dr. Aaron Skoda, Supervisory Pharmaceutical Scientist and the Office of Product Quality Assessment 3, OPQA 3, and the Office of Pharmaceutical Quality. Then we will hear from Dr. Deborah Johnson, Supervisory Pharmaceutical Scientist and OPQA 3 within OPQ and CEDAR. And lastly, we will then hear from Dr. Madhasudan Gavravaram from OPQA 3. And after these presentations, our speakers will then convene on stage for our Q&A session for the day. Now, please join me in welcoming Dr. Aaron Skoda.
Good morning, everyone. Thank you, Corey. When we were tasked with coming up with topics for today's Drug Master File session, we were inspired by our Drug Master File workshop that we gave with SBIA in 2021 and the feedback that we've received from all of you since then. Excuse me. And with that in mind, we have some straightforward and actionable but impactful items for you to consider as you submit Drug Master Files to facilitate assessment. So we'll start off with discussing factors that can facilitate assessment of Type 2 Drug Master Files. In OPQ, pharmaceutical quality and our mission are at the forefront of our minds each day. And even though I won't be discussing pharmaceutical quality on each slide specifically, items in these slides, if you consider them, will help us to assess the pharmaceutical quality of your Type 2 Drug Master File. So with that, the objectives for this talk are to help facilitate assessment of your Type 2 Drug Master File by learning to identify items that DMF holders can include in their submissions. Likewise, identifying items that ANDA applicants can include to help assessment of Drug Master Files. And recognizing missing information in Drug Master Files that, if provided, can help with the assessment of your DMF. A couple of housekeeping items before we get into the rest of the talk. When I say Drug Master File, I'm referring to Type 2 Drug Master Files for drug substance. And I'm not discussing the completeness assessment guidance, but that is a helpful place to start. And so this is kind of after you've addressed everything in the completeness assessment guidance, these items that we'll discuss today kind of take the submission one step further. So if you've never submitted a Drug Master File, or if you're new to Type 2 Drug Master Files, the completeness assessment guidance is a good place to start. So first we'll start with identifying the items that Drug Master File holders can include. Specifically, my first item is the workhorse form of the DMF world, FDA Form 3938. This is a fillable form that is used for all Drug Master File submissions. It includes all administrative aspects of the Drug Master File, including contact information, facilities, referenced DMFs, etc. It expedites information processing because we know where to find all of the information that is related to your Drug Master File submission. And this file can be found on the DMF submission resources page that I've linked here and looks like what I've shown on the slide. And even though I just finished saying that I'll only be talking about type 2 drug master files, I want to point out that this Form 3938 can be submitted with any type of drug master file, types 2, 3, 4, or 5, and is very helpful and valuable in these submissions. With structured information in mind, we also have been seeing an increase in the structured data file submissions to type 2 drug master files over the last several years. These are chemical structure files that are in a mole connection table format, and they provide unambiguous chemical structure information to expedite assessment. And they look like what's shown on the right side of the slide. This is helpful to us since when you're preparing your drug master file, you are creating chemical structures. And so instead of duplicating work upon assessment, we can use these structured data files in our own assessment. It ensures that the structure that you intend for us to assess is the one that we're assessing. If you've never submitted an SD file, I've linked to a quick guide that we created several years ago that can be used to help you in these submissions. When submitting amendments to your drug master files, it's very helpful to include a summary of changes. These summary of changes are particularly useful when they're in a tabular format because it clearly helps us to focus on the updates that are included in a given amendment, so the changes since the last time you submitted. Some section headers, some column headers that I've shown here that would be very helpful to include in such a table are section, the current parameters, the change that you've made or proposed, and a justification. And obviously, a table can't really allow you to include a lot of data and a lot of justification, so more detail can be included outside this summary table. But the summary table helps us to focus on the changes that you intend for us to assess related to your drug master file. And this justification is particularly useful to us. It allows, if you explain and justify the change, including providing data as part of the justification and discussing risk to the process or the quality, it helps us with our assessment. Some things that are helpful to submit when making a change can be found in this draft guidance for industry on post-approval changes to drug substance. So this guidance suggests some data for the given type of change and ways to discuss risk. So those were items that DMF holders can submit to help us with assessment. And ANDA applicants likewise can submit certain things to help us in our assessment of the drug master file and to help expedite review. And in the realm of structured data, Form 356H, which I'm sure you're all aware of, is very helpful to us in that it lists the drug master files that should be considered for each application. So this form that I've linked here is submitted with an ANDA, and it includes all of the referencing information. So things like drug master file number or drug substance facilities are included on there. And this helps us to identify related information for us to assess in order to approve an application. Please assure that all of the DMF numbers are accurate and in this form, and please include all of the drug substance facilities so that we can assess the facilities and other information in a timely manner. Along with this, when a supplement is submitted to an ANDA, changes to a drug master file that if changes to a drug master file cause the changes to the ANDA supplement, it's very helpful to include that information in the cover letter. So oftentimes the applicant will summarize the information on the changes. It's very helpful if you make it clear that the changes are related to a specific drug master file. Also, including the drug master file number is particularly helpful to us. So oftentimes in the cover letter, it may say what the holder's name is or the facility information, but please do also include the drug master file number, as that helps clearly indicate your intentions for these supplemental changes. Likewise, so sometimes the DMF number is missing from a cover letter, whereas also occasionally missing information from drug master files, where if it is provided, can help facilitate the DMF assessment. One of these items is a letter of authorization. So these letters give another party the right to reference your drug master file. They're submitted to a referenced DMF. They're submitted in a referenced DMF with a copy in the referencing ANDA or DMF. These files are necessary for us to review the drug master file. They're so important that we have a separate talk coming up next from Deborah Johnson to give you more information on using these letters of authorization. And just a quick note about these, we often note that letters of authorization are included in the drug master file at the time of completeness assessment. But often when we do discover an LOA that is missing, it happens later, so after the completeness assessment. So please do continue to update your letters of authorization over time. Impurities are often very well discussed in drug master files, but some items for you to consider are, when considering organic impurities, a fate and purge discussion with data is very helpful so that we understand your rationale for the way that you're controlling the impurities in the drug master file. That really helps us to assess the control strategy and to assure that we are on the same page. When providing justifications or data, we recommend that you consider the documents, the ICH Q3A, ICH M7, the USP monograph, and nitrosamines. I've linked to the nitrosamines website here. This includes links to the guidances for small molecules and NDSRIs, as well as gives updated information on the nitrosamine control strategies and information from the agency. Related to impurities, it's helpful if you include and consider the maximum daily dose when submitting your type 2 drug master file. This can be obtained from the drug product label for your referencing applications, and it's helpful to consider all of the referencing applications that you intend to use. It's important because this affects the impurity limits per ICH Q3A and ICH M7. And for more information on how to determine a maximum daily dose, you can refer to Melanie Mueller's talk from yesterday's generic drug forum, where she goes into much more detail than I can here today. Facilities information in the drug master file is very helpful for us to have at the beginning. So as I mentioned earlier, it's helpful that the ANDA applicants submit all of the facilities information, and likewise, that starts with the drug master file holder submitting the information. So please do list all facilities used in your DMF in 3.2.S.2.1 and relay those facilities to the referencing applicants. It's also helpful if that section has the function of the facilities listed so it's clear what is happening at each facility. This ensures that we can have timely evaluation of the facilities related to your drug master file. These can also be submitted in FDA Form 3938, Section 9, as shown here. And again, this form really helps to assure that we know where to look for the facilities. So please consider submission. When responding to a letter, such as an IR or a CR, it is very helpful to have timely responses. Updating sections 3.2.S related to any changes that you're making in your response and FDA Form 3938 is also very helpful. So when you respond to the CR or IR, responding fully to each question is helpful, and then referring back to the 3.2.S section in which the change is being made really helps the assessor to focus, and it allows us to when 3.2.S is not updated, we will often send a letter asking you to do so. So this saves time so that we have fewer back and forth communications. And so that allows us to continue on with our assessment to support the referencing ANDA. It's also helpful in Form 3938 to indicate that it is a response to a letter. So there is in Section 7 a box that you can check to indicate that it's a CR response or deficiency or IR response. For secondary DMFs, these are very helpful for us to identify very early in the review cycle. This is a reference to another DMF, and it should be very clearly indicated. We recommend that you include this information on the cover sheet for your Drug Master file, as well as in Form 3938, Section 10. This is helpful because if the information is available to us in the beginning, we know whether we have to assess the secondary DMF as well as the primary DMF. And Madhu will talk about this later in the session. So please stay tuned for his talk. Prior assessment requests are a relatively new submission type to the agency. These are requests for assessment before the ANDA is received per GDF 3. These DMFs should meet the criteria that are listed in the commitment letter and also available in the guidance for industry that I've linked here. These are DMFs that have not had a substantive review done previously and, again, meet the criteria. In the appendix of this guidance, there's a full list of information that should be provided from both the applicant and the DMF holder, but submitted to the DMF from the DMF holder. So please do consult that appendix before submitting your prior assessment request. And the reason for this is this allows us to respond quickly and in a timely manner to your request for this prior assessment. If information is missing, then there will be some back and forth communications requesting the missing information. So if you refer to this guidance appendix, it's particularly helpful. So as we get close to wrapping up, I have a challenge question for you. So if you're in the room, please raise your hand when I say the right answer. Submission of FDA Form 3938 allows us to easily identify the need for assessment of which of the following? A, a response to a letter. B, reference to secondary DMFs. C, drug substance facilities. And D, all of the above. So who thinks it's A? B? C? Or D? Yes, so as I mentioned in the beginning, 3938 is particularly helpful to us in a number of ways. So everyone in the room got that question correct. I hope everyone online was thinking along the same lines and that you will consider to use Form FDA 3938 in your submissions to any type of drug master file that you're submitting to the agency. So as we reach conclusion, I want to provide some resources for you to look at. So in addition to the things that we discussed today, the Drug Master File website is helpful, and it links to the submission resources website. So this has things like the 3938 form, as well as the quick guide to SD file submission. This draft guidance for industry on drug master files has a lot of information that is helpful to consider in your submissions. And for information on the referencing applications and the MDDs, you can refer to the FDA labels. And then finally, I mentioned the guidance for industry, but we didn't talk about it in detail on completeness assessments for type 2 API DMFs under GDUFA. And this provides a checklist of all of the items that should be concluded in order for us to start assessment of a type 2 drug master file. So to summarize, providing explanations and justifications in your submission really helps us to assess your drug master file. It assures that we understand your thinking and can properly assess the DMF. Improving communication between the agency and the DMF holder, as well as the DMF holder and the ANDA applicant, can lead to gains in review assessment. So this increased communication can save time. And including FDA Form 3938 in each submission really clarifies multiple points and helps us to know all of the information accurately and in an up-to-date manner for your drug master file. So with that, thank you for attending my talk, and I'm happy to answer any questions during the panel discussion. And I'll turn it over to Deborah Johnson to talk about letters of authorization.
All right, thank you, Erin. So my name is Deborah Johnson. I'm a supervisor in the Office of Pharmaceutical Quality Assessment 3. and I am here to tell you what FDA wants you to know about your letters of authorization. So first, we'll cover 4 main ideas in this talk. We're going to talk about what is a letter of authorization, or LOA for short. Talk about some LOA basics. I want to talk about the importance of your LOA, and also the importance of keeping things up to date. Now, an LOA can actually be used to authorize reference to multiple items, but I'm going to stick specifically to letters of authorization that are related to type 2 drug master files. So before we talk about LOAs, I just want to talk a little bit about drug master files. So first, what is it? Well, it's a confidential submission to the agency from a DMF holder. It contains information on manufacturing processes, control strategies, facilities, specifications. So it contains a lot of information. It can support an IND, an NDA, an ANDA, or even another DMF. It is not required. You don't have to use a DMF. But very importantly, the FDA cannot access that information in the DMF without a letter of authorization. So as Aaron kind of alluded to, 21 CFR actually has some information requirements that pertain to a DMF. First, the DMF has to have a complete list of the currently authorized parties that are allowed to incorporate by reference the information in the DMF. Okay, that LOA needs to identify by name, reference, page number, volume, the information that each authorized party is allowed to incorporate. Now, an LOA can allow all of the drug products from a particular party to be included, or they may restrict it. If the LOA restricts it, then the LOA must say what the drug product name is and what the application number is. So, since the DMF must authorize someone to do it, how do they do that? It is through this letter of authorization, or LOA. So, what is the LOA? Okay, it's a formal signed document from the DMF holder. Now, the DMF is a confidential submission that is completely separate from any of the referencing applications. And the LOA is the permission that the DMF holder has given the agency to go into that DMF and access that information in the support of the referencing application. It's important to note that that letter of authorization does not allow the authorized party to access that DMF. And what that also means is if you're an applicant and you call us, we cannot disclose information from the DMF. We cannot tell the customers what is in the DMF. That letter of authorization gives the agency the right to go in and access. So, some LOA basics. Okay, 1, it must be on company letterhead. 2, it needs to identify the specific DMF number, the process, the product, the process, the facilities that are being authorized. It needs to tell us who the authorized parties are. And it's important to note that an LOA is for a specific authorized party. You can't have an LOA that gives multiple authorized parties reference to your DMF. Each authorized party needs their own LOA. So, how do you submit this LOA? It needs to be submitted directly to your DMF, and a copy needs to be submitted to each of the referencing applications, or a referencing DMF or IND. But what if the DMF holder and the applicant are from the same company? Do you still need an LOA? The answer is yes. And this is one of the most common mistakes, is assuming that just because the applicant and the DMF holder are from the same company that you don't need an LOA. An LOA needs to be submitted to the DMF and to the applicant, even if they're from the same company or if they're under the umbrella of the same organization. Okay, an LOA. If that's missing, then there's back and forth, and it slows things down. So even if you're from the same company, you still need an LOA. Okay, here's a plea from us reviewers at FDA. Please don't make a search for your LOAs. I will tell you this is particularly problematic for secondary DMFs. So a secondary DMF, you have a primary DMF, and that DMF is the one that the applicant is referencing. But then that DMF holder may reference another DMF for an intermediate. Okay, that primary DMF needs an LOA from the secondary DMF holder. We would love it if we could find it, but often when there's a secondary DMF, we get our work, we plan it, we're doing the review, you get to section 2.3 or 2.4, and whoa, there's a secondary DMF, there's an LOA, and we're like, oh, now I've got an entirely new submission that needs to be reviewed. And that can really derail us from, timelines, and it can, impact, us getting our work done in a timely manner. So please, please, please don't make us hunt. So how can you do that? Well, when you're a DMF holder, what you need to do is in your electronic submission in section 1.4, if it's an LOA for someone referencing you, put it in 1.4.1, letters of authorization. If it's an LOA allowing you to reference someone else, put it in 1.4.3. I will tell you, when we start a review, the first place we go to, these sections. It's the first place we open it up because it lets us know right off the bat how much work is involved. So if you put the information right here, we will see it, and it really, really makes our lives a lot easier. All right, this is a critical point. No LOA, no review. Okay, the best way to mess up a customer's timeline is not to submit an LOA. I will give you an example that happened to me about 3 months ago. There were 2 DMFs for a particular API. Both of those DMFs were supporting a very high priority application with a significant drug shortage on a really compressed timeline. The primary DMF referenced a secondary DMF. So we went to the primary DMF and we looked for that letter of authorization, and we looked and we looked. It took my poor reviewer about an hour, but we found it. Okay, great. Then we went to the secondary DMF and we looked for the corresponding LOA. We could never find it. So we had to send an IR to the secondary DMF holder and said, hey, you need to submit this LOA to your application or to your DMF so we know this other guy can reference you. About a day later, we get an email with the LOA. It's like, thanks, but that doesn't help. So we sent a second IR saying, yeah, great, but the LOA needs to be submitted directly to your DMF, which they did. But all in all, we burned up 3 days on a clock that was less than 8 days. Okay, so it's really important. We need those LOAs, and they need to be there when we start, because that is something that can really derail a timeline. So I want to talk to you about, what does your LOA say to the FDA? One, it says you're referencing the entire DMF. You are using every facility in the DMF. You are using every process in the DMF. But what if that isn't true? What if that's not accurate? I want to give you an example. Okay, so we have API by Scotty, who has DMF 00000 for dilithium crystals. It has an existing manufacturing facility, number 1, Acme Drugs in Timbuktu, USA. They just added a new manufacturing facility, so number 2, Acme Drugs in Sayware, USA. Now they have a referencing application from Drugs R Us for ANDA 999999. However, that ANDA actually only uses API manufactured from Facility 1. Now, they were very good stewards. Everybody's got the LOA. That's great. So we go in, we look at the LOA, and it says API by Scotty authorizes Drugs R Us to incorporate by reference information regarding the dilithium crystals from that DMF, and it's relevant to any application filed by Drugs R Us. And the DMF holder authorizes the FDA to go in and look at that information in support of that ANDA. Great. Well, it turns out that facility number 2 has never been inspected. So ANDA 999999 gets a CR because of facility 2. Drugs R Us comes back and said, but wait a minute, I didn't use facility number 2. But remember, the FDA assumes you're using all the facilities and all of the processes that are in that DMF. So what do? How are you going to get out of this pickle? Well, you can amend the LOA. So the DMF holder can go in and amend that LOA and say, look, this applicant's only going to use API from facility number 1. That's perfectly fine. Now, if you are making those kind of nuances, then an LOA needs to specify which applicants are reviewing what parts of the DMF. That needs to be specified in the LOA, because per the FDA guidance for industry in 2019, if you don't specify that, we assume the entire DMF is being referenced for every application. So the DMF holder kindly amends the LOA. And now it says here you get to authorize this, you can look at this, but the authorized party only uses API manufactured by facility number 1. Consequence? Well, that facility number 2 still hasn't been inspected, but ANDA 999999 gets approved because they only used facility number 1. So something else to consider. Nothing is forever, and that includes your LOAs. So when things change, your LOAs need to change as well. For example, in 21 CFR, it did say that your DMF needs to contain a current list of people that are authorized to incorporate by reference your information. But if you have removed that permission, you need to remove that LOA and you need to take their name off that list. Because if they're still there, then we think that they're authorized. Conversely, if you've added someone, again, a new LOA needs to be submitted and that list needs to be updated. So please keep that list updated. So companies are not in the witness protection program, and when they change their names, it needs to be updated. So if you have an authorized party and they change their name, the DMF holder needs to submit a brand new LOA with that new information to their DMF in section 1.4. Conversely, if the DMF holder changes names or companies, they also need to change the LOAs. So they're going to need to update all their LOAs with their new name change and put that in their DMF, and then they need to submit all those new LOAs to any applicable referencing party. Okay, now it is really frustrating when you pick up a DMF, because the first thing you do is you go look for the LOAs, and it says I'm doing ANDA whatever, but the name on the LOA does not match the ANDA. There's been a name change somewhere. So we're going to have to kind of stop. We're going to have to send an IR, hey, can you update your LOA because they don't match? Okay, every time we have to stop and kind of communicate like this, it slows things down. So please, keep your LOAs up as names change. Now, I know you are now all complete experts at letters of authorization, but what if you run into something that I did not cover? You have no fear. You can contact us at CEDAR OPQ inquiries at fda.hhs.gov. So LOAs are tricky things, and they can be complex. I cannot cover all the myriad of issues that can come up with an LOA. So if you run into something, please, please, please reach out to us, and we will try and help you out. So in summary, an LOA is the permission the FDA needs to reference confidential information in a DMF to support it. Without that LOA, we cannot do anything. You need that LOA even if you're from the same company. And I will tell you, this is the most common reason that we are missing LOAs. It's like they just didn't think they needed it because they're from the same company. LOAs can be specific to facilities and processes. And if you need some help with that, if you run into an odd situation where you think this little carve out needs to be happening, please reach out to CEDAR OPQ inquiries and we can help you. Your LOAs need to stay current. Okay, they're kind of like a living document. As things change, they need to change with it. And I want to acknowledge Melissa Furness. She's in our OPQ, or OPPQ, our policy group, and she's kind of my go-to person when I run into odd and strange LOA issues. So I really want to kind of put a shout out to her because she really helped me out with this presentation. And I just want to say thank you, and I will turn the time over to Dr. Madhu Gowravaram. I told him I was going to slaughter his name. All right, thank you.
Good morning. good afternoon and good evening. Okay, the title of my presentation is Regulatory Review Process for the Type 2 Secondary DMFs. In this presentation, I'm going to discuss the risk-based regulatory review process for the acceptance of Type 2 secondary DMFs, for the adequacy of Type 2 primary DMF, supporting and applications, and also some regulatory aspects associated with secondary DMFs. As we know, many Type 2 drug master files refer to a secondary Type 2 DMF in support of their submissions. So in the presentation, first I'm going to talk about what are secondary DMFs and how do they impact the review of primary DMFs. The secondary DMFs could be a starting materials, intermediate, or an API itself for the primary DMF. Hence, the scope and role of secondary DMF is a significant factor in evaluating their acceptance for a primary DMF. I'm going to go through a few case studies with the different scopes and roles of secondary DMFs and the risk-based review approach in their evaluation. At the end, I'm going to touch on few regulatory aspects of secondary DMFs. Okay, what are the secondary DMFs? Secondary DMFs are DMFs referenced by a primary DMF for the process of its final drug substance synthesis. As I said before, a secondary DMF could be for a starting material, intermediate, or an API itself. Also, there could be more than one secondary DMF associated with a primary DMF. It is important to note that when there is a secondary DMF or DMFs associated with a primary DMF, the manufacturing process of the primary DMF drug substance is evaluated based on the information presented in all the DMFs, that is primary and secondary DMFs, including facilities. Also, assessment of control of process impurities in the primary DMF drug substance is based on the overall control strategy of impurities presented in both primary and secondary DMFs as well. If the secondary DMF is referenced for a starting material, it should be justified based on ICH Q11 guidance. Also, all the manufacturing facilities of the secondary DMF, starting from the regulatory starting material, require CGMP compliant for ICH Q7. I think in this hypothetical example, I'm going to illustrate how we assess primary DMF manufacturing process when there are secondary DMFs are associated with it. In this example, primary DMF1 for the synthesis of its final drug substance API salt, that is E, refers to a secondary DMF, that's intermediate D, which itself is an API base. The secondary DMF refers to another secondary DMF3 for its intermediate C. So in this overall manufacturing process, the regulatory starting material for the manufacturing process of the final drug substance E in primary DMF1 are A and B from the secondary DMF3. Also, the manufacturing process presented in secondary DMFs 2 and 3 also considered part of manufacturing process of final drug substance E. As you look at here, the manufacturing facility of intermediate D in secondary DMF, which is nothing but an API, it's in a base, is also considered as the API manufacturing facility. Also, it is important to note that all the manufacturing and intermediate facilities should have sufficient information provided by the applicant for FDA inspection. In this slide, I would like to show some examples where the primary DMF refers to secondary DMFs, pretty much, which is nothing but an API. So, for example, a pure API primary DMF can refer to a secondary DMF, which is an crude API. A primary DMF, which has an API salt, can be referred to the corresponding base or acid salts in a secondary DMF. And we do also see some examples where salt exchange takes place between a secondary DMF and a primary DMF. A sterile API or primary DMF can refer to non-sterile API. And also, when we have mixture of two APIs in a primary DMF, many instances they refer to a single API secondary DMFs, which are nothing but an APIs. And we also have some examples where the change in physical form we do see from secondary DMF to a primary DMF. Like a secondary DMF could be an API anhydrous, and they take that material and make a monohydrate, or like a monohydrate to pentahydrate, like that. We do have examples, such examples. Also, enantiomerically pure API can refer to racemic API. And we do have some API premixes where in most cases the secondary API comes from the secondary DMF. Okay, according to the, here I would like to point one thing: when there are secondary DMFs are associated with a primary DMF, according to the GDF DMF completeness assessment checklist, subject of the DMF is a single drug substance produced by one manufacturing process. So, when there are multiple secondary DMFs associated with a primary DMF, we recommend primary DMF holder to make sure overall manufacturing process is similar in both the DMFs. That is, the synthetic scheme and regulatory starting material should be similar in the both secondary DMF 2 and secondary DMF 3 referred by in a primary DMF 1. Okay, in the next few slides, I'm going to go through a few case studies based on the role of the secondary DMF. If the secondary DMF is referenced as a starting material, it should be justified based on ICHQ11 guidance for the process of the primary DMF drug substance. If the agency does not accept secondary DMF as a starting material, the primary DMF holder may be asked to redesignate the starting material for its process. If the agency accepts secondary DMF as a starting material, so then the formal review of the secondary DMF may not be required. Again, the extent of the review of the secondary DMF is based on what is presented in the primary DMF. If most of the information is already present in the primary DMF, we may not look at the secondary DMF, but if the information is not there in the primary DMF, we do look at the secondary DMF. And also during the review, when we conduct the review, we want to make sure all the information in the secondary DMF as a starting material for this manufacturing process, whether it is present or not. Okay, if the secondary DMF is an intermediate, many of the quality sections, I would say, in the DMF will be critically evaluated during the review, including CGMP and facilities. Some sections of the DM may not be very critical as an intermediate. Again, it is the primary DMF holder responsibility to ensure proper control of all potential process impurities in the API, either in the secondary DMF or primary DMF. So when we evaluate the secondary DMF as an intermediate, so certain like ICHQ3A, ICHM7 may not be applicable. So it is the primary DMF holder responsibility, the control strategy of overall impurities in the manufacturing process. If the impurity is controlled in the upstream process in secondary DMF are higher than ICHQ3A with no downstream control in primary DMF, supportive information such as spike per study and batch data as applicable should be presented either in the secondary DMF or in the primary DMF as an overall control strategy to demonstrate their absence in the final API. Again, the same applies to the genotoxic impurities for ICHM7 as well. Okay, this is an interesting example. So if the secondary DMF is itself a drug substances, so there could be two scenarios when the secondary DMF itself a drug substance. The evaluation of the secondary DMF is based on how the material is referenced. In majority of cases, the secondary DMF is also referenced by ANDA applicants. In this scenario, the secondary DMF is reviewed as a regular drug substance because it's already referenced by another ANDA applicant and it is itself a drug substance. But if the secondary DMF, even though it is a drug substance, it is referenced by only primary DMF holder as an intermediate and is not referenced by any ANDA application, then the secondary DMF is reviewed by as an intermediate for the adequacy of referencing primary DMF. Again, it is the primary DMF holder responsibility to ensure proper control of all potential process impurities for ICHQ3A and ICHM7. Here I would like to emphasize for the secondary DMF holders that in future, if they want to reference their DMF directly to another ANDA applicant as an drug substance, then we need to evaluate the adequacy of this DMF for the newly referencing application. We have several cases where more than 2 or 3 secondary DMFs are associated with the primary DMF. In fact, we do have few examples where more than 5 secondary DMFs are associated with a primary DMF. So, presence of secondary DMFs pose a significant impact on review timelines, especially with a chain of secondary DMFs are associated. Applicant may want to be aware of associated secondary DMFs due to its consequences, which I will be touching on in the regulatory aspects. Okay, let's look at the regulatory aspects associated with secondary DMF. Secondary DMFs have to be adequate for the adequacy of primary DMF. Secondary DMF's inadequacy usually triggers a CR letter for primary DMF, basically informing the primary DMF holder that the quality review of your primary DMF cannot be completed until all DMF deficiencies, including those in secondary DMF, are adequately resolved. And this most importantly, secondary DMFs are reviewed with same timeline as primary DMF, guided by the same target date. Hence, solicited and unsolicited amendments to the secondary DMF are treated the same as primary DMF and will have the same implications and consequences to the goal date extensions. For example, if the secondary DMF holder submits a late unsolicited RS, then we may need to extend the goal date. So it doesn't have to be the primary DMF. So primary DMF holder needs to make sure there should not be any unsolicited amendments submitted to the secondary DMFs. If we receive then we may need to extend the goal date. During the DMF completeness assessment, the following will be checked: whether the primary DMF contains a letter of authorization for any DMFs reference to the support the DMF, and also we do check all DMFs referenced have been filed with the agency and are active. As Deborah indicated, letter of authorization from the secondary DMF holder should be provided in section 1.4. Also, the material sourced from the secondary DMF should be clearly mentioned in appropriate sections of the DMF. Erin pointed out, again, there are some places where the association of secondary DMF should be documented, like Form 3938, and also cover letter. Another place is 32ST1, because this is very important for us to look at the facilities involved in the manufacturing process of the drug substance. Because late identification of secondary DMF association due to lack of information, appropriate sections of the DMF and LOA may lead to delay in review timelines and issuance of IR letters. Also, if the designation of the material sourced from the secondary DMF as starting material is not appropriate for ICH Q11, it may lead to inadequate major deficiency, which has a longer good for goal date. So when a primary DMF holder reference a secondary DMF, maybe the secondary DMF may be nothing but an API, but the secondary DMF holder, what is the manufacturing process for the secondary DMF holder? If that is not appropriate in the secondary DMF, so we may need to push them to further redesignate the starting material. That leads to delay in the approvals. Okay, for the material sourced from the secondary DMF, the facility information and CGMP statement may be listed or referred to the secondary DMF in section 32S21. That is the way we look at the facilities associated with the manufacturing process of the drug substance. Having the correct facility information early in the application is critical for timely evaluation of secondary DMF facilities as needed, as Deborah, Erin indicated in their presentations. As well, again, the late cycle facility evaluation can delay the approvals. According to the FDA draft guidance, drug master files, so I just picked a phrase from this guidance. A primary DMF can incorporate information in a secondary DMF by reference. The secondary DMF holder can submit an LOA authorizing either the primary DMF holder or the product applicant to reference the secondary DMF. As stated in the guidance, I would like to emphasize that where possible, consistent with confidentiality agreements, secondary DMF holders are encouraged to submit LOA authorizing drug product applicants to reference the secondary DMF directly. That way we don't miss the appropriate facilities needed for the evaluation. I would like to thank Motang and Deborah for their feedback on the slides. And also I would like to thank Unit 9 team members for their support. Thank you for listening.
All right, I now welcome our speakers back up onto the stage for our discussion panel. And in the meantime, I'll invite anybody in the room who has questions to please line up at the microphones. As a reminder for both in person and online, we can only answer questions that pertain to today's topics. And so with that also being said, we do not answer application-specific questions. Please keep it general for us today. And if everybody's ready up top, we'll go ahead and get started if you'd like to line up.
I can scream. They know I voice question that probably involves all three of you, because I want to understand it is a situation with a primary and a secondary DMF. For example, somebody is trying to make a product, say in US, but they are buying an intermediate from another country. But the DMF for this intermediate, this intermediate company actually makes that same API. So they have a complete DMF, and all they're trying to give is an LOA saying you can reference my DMF. And just like you mentioned, Deborah, it can be a little dangerous because they are only buying an intermediate from them. The intermediate is part of the entire synthesis. So some weird situations come up here. In my opinion, if it is referenced, the reviewer should stop at that intermediate, which is like an impossibility sometimes, as we all know. And the other thing is the LOA should very clearly probably mention. But would you think that in these positions, having another DMF would have helped? But the question is, it's the same process, you know?
It's an interesting question. Now, we have actually seen instances where a primary DMF holder will reference another DMF that is actually itself an API, but they're only referencing it up to like point C. So they're really just buying it from an intermediate. Now, if that LOA just says, you know, DMF B allows DMF A to reference it, again, we're going to assume you're referencing the entire DMF. If that's not true, the LOA can be specific. We allow company A, we authorize them to reference from here to here. And then we know exactly what part of the secondary DMF is being referenced. Now that's important because let's say that in that secondary DMF, there's something in intermediate D that has a problem. So we review it. Now, this DMF is inadequate. That's going, as Madhu said, that makes the primary DMF inadequate. But again, kind of like in my example, like, but hey, we're not using intermediate D. Well, your LOA didn't say that. So if the LOA says we're only going to go from point A to point C, that's what that secondary DMF would be reviewed for. And now, if it's itself its own DMF, and it's reviewed, it could be inadequate because it has problems later on. But that secondary DMF could be considered adequate for the primary because it's only going part way. But that's where the LOAs can come in. Be specific. If you're not going to reference the whole DMF, be specific about what parts you're doing. You guys have any other questions?
Yeah, go ahead. Go ahead. Follow up on a little bit on an example where a primary DMF folder refers to a secondary DMF intermediate, only for the intermediate. The secondary DMF is adequate. But when we evaluate for the primary DMF, when at the intermediate, we had to make it inadequate for the primary DMF because when the LOA was only for the intermediate, but in their manufacturing process, whatever the control of impurities in the intermediate, they address it in the final drug substance synthesis, and the DMF is adequate, the secondary DMF is adequate. Whereas whoever the primary DMF referencing the intermediate, they did not address the control of the impurities properly from the intermediate from the secondary DMF. Even though secondary DMF adequate, we had to make the primary DMF inadequate because of the not addressing the control of impurities, which they bought from the secondary DMF.
Can I just think? That's exactly, I think, Madhu.
Mic should be on.
Yeah, I think, Madhu, that was exactly a point, because that original secondary DMF was treating this as an intermediate with, there isn't like a specification. There is a specification, but they are not, they're moving forward. They don't have a retest, things like that. So you are very right that the challenges are very interesting. And exactly, I found that challenge, that I would have liked way more control of impurities in that intermediate, which the original, that secondary DMF was doing in the finished product. And thank you very much. I think I wanted this to be discussed a little, because this will probably happen a little more, trying to bring manufacturing to US. So it's good that people hear it. Thank you.
Thank you for that. Are there any further questions? If there are, please stand up at the microphone at this time. If not, we can go ahead and open it up for the sake of time to some online questions. If you're ready, Brenda.
I am ready, I believe. Okay, Dr. Skoda, what type of DMF is applicable for drug plus excipient co-processed API? For example, an API itself is unstable and needs to have an excipient as a stabilizer.
Thank you for that question. So this is a little bit complicated, and there is some information about this in the GDF, I believe it's the completeness assessment guidance, that discusses the subject of the DMFs for type 2. So it's very helpful if you can specifically explain in your drug master file the reason for co-processing an API. And if you're unsure about how best to submit, you can submit a question to the DMF inquiries mailbox that Deborah has on her slide. But in general, type 2 DMFs are the way that people normally do that. However, there are GDUFA and PDUFA considerations, so please do look into that.
Okay, this is for Dr. Madhu. I'm taking the short route, Dr. Madhu. What are the obligations of an ANDA holder with respect to changes made to a secondary DMF that is referenced within a primary DMF? Specifically, is the ANDA applicant required to submit such changes to the FDA?
Yeah, so they need to look at their manufacturing process, whether the secondary DMF, the changes made in the secondary DMF, so how those changes are affecting the manufacturing process. So they need to look at the post-approval guidance. They need to follow, because if the secondary DMF is part of their manufacturing process, when the changes made to the secondary DMF, they need to inform the same to I think primary DMF holder should be informing the ANDA applicant. The ANDA applicant should be submitting appropriate post-approval changes.
Okay, thank you for that response. Dr. Skoda, we'll skip back to you, please. My question is related to MDD. Different formulator has different MDD, so which will be considered for type 2 DMF?
Thank you for that question. When you consider the MDDs, you should look at your specific referencing applications. And to get the MDDs, I again refer you to Melanie Mueller's talk from yesterday. It's helpful if you consider the highest MDD, because that is the most restrictive in general. However, it's very helpful if you can clearly indicate your rationale for impurity considerations and limits when you submit your drug master file, and indicate which MDD you're considering for your DMF.
Thank you, Dr. Skoda. The next question is for Dr. Johnson. Does the primary DMF holder need to submit the LOA to referencing ANDA holder or to secondary DMF holder?
Okay, if there's just a primary DMF, single DMF, the DMF holder submits an LOA to their DMF and to the referencing application. If there is a secondary DMF, the secondary DMF holder submits that LOA to their DMF, then they submit a copy to their referencing DMF. There's nothing to stop them from submitting it also to the application. I would say in general, what we see though is that chain. The secondary DMF holder submits an LOA to their DMF and the primary DMF. And then the primary DMF submits their LOA to their DMF and the applicant. That's generally how we see it. But the secondary DMF holder could choose, if they wanted to also submit it to the applicant. I don't know if that answered the question, but.
Thank you. I think Dr. Skoda has something to add.
Yeah, I just wanted to go back to the MDD question and say one more thing I forgot to mention. Your type 2 drug substance DMF will be considered for each referencing application. And so it may be acceptable for one and not the other, depending on which MDD you did apply. So please consider all of the referencing applications and clearly indicate everything. Thank you.
Thank you very much. We do appreciate the clarity of these responses and being as expansive as you can. Dr. Skoda, let's stay with you for this time.
And I do think we have one in the room.
Oh, okay, sorry.
Good morning. Thanks for sharing all the information. My name is Kamal Biswas. I'm from Regular. We help companies improve quality using AI. So all the guidelines are very clear. I think we have all the forms. We have all shared different resources. We still fail to submit the quality DMFs, right? A lot of time you need to go back to them and ask for information. So my questions for you, I mean, what do you see? What are the improvement opportunities for the companies? What are the top issues that you find either one of you or all of you may share?
So sorry, you're asking what the most common areas for improvement are?
That's correct, yes. What are the issues that you see there?
Yeah, so I think that missing information or failing to communicate are the things that I pointed out in my presentation, are things that are easy to address and have a big impact. So I think that starting there is a very good place to start. So the completeness assessment guidance for type 2 drug master files, making sure that everything is included in the submission, and then taking it further to make sure that everything is explained and communicated to the ANDA applicant and to the agency is very helpful. That puts you on a good starting place to get an efficient assessment of the type 2 drug master file.
I would say it's one of the places we probably end up issuing the most comments is probably impurities. It's tricky. Taking a good look at your impurities, and I mean, I think we're at a point now where you don't just look at it and guess which ones are genotoxic. You know, we are kind of expecting a hazard assessment so that you're classifying them appropriately per ICHM-7. So doing that up front, saying, you know, here's the data, you know, this is, you know, class 5, this is class 4, this is why, including the entire report is important. Looking at places where you control upstream, where you're controlling things upstream, making sure you have adequate justification. You know, if you're controlling something at 8% in an intermediate, you know, what's your justification, especially if it's not controlled downstream, so fully upstream, what's the justification? How do you know you can purge 8%? The other thing that we often see is, yes, this impurity indeed did not carry through to the drug substance because it turned into this impurity. So also considering, is that impurity transformed as it goes through the process? You know, those are kind of the things, you know, to consider when you're talking about fate and merge. I would say that's probably where we issue the bulk of our deficiencies. I think people have gotten pretty good. I think starting materials, I think people do a pretty good job. We still, once in a while, we'll see people pushing the envelope. But I know when I still do some primary review, yeah, it's the impurity profile where I see the most.
Good. Thank you so much.
Thank you for that response. Are there any other questions in the room? Seeing one person.
Hello. I have some question about when you just mentioned the missing communication. So no matter filing the DMF or writing the LOA, what is the most common miscommunication you meet frequently?
Yeah, so the question is, what is a common communication gap that we see?
Yeah.
So I would say it's, there is one that I didn't discuss today. So the ones that I mentioned are common. There is one that comes to mind that we didn't discuss, which is communication between the DMF holder and the applicant related to goal dates. And so it's helpful if the ANDA applicant shares their review goal dates with the DMF holder so that they can coordinate and the DMF holder can respond in time, for example, to a letter that we issue to the DMF to assure that we can keep moving with the ANDA application review. So I would say that lack of communication can cause significant delays. So again, I mentioned that the holder and the applicant should communicate, but that specific piece of information is very helpful.
Thank you.
Thank you for that. There's no further questions in the room. We can go ahead and turn it over to Brenda.
Okay, let's go with Dr. Johnson. Can I reference a type 5 DMF for sterile facility information? Can it be referenced for all application types under NDA, BLA? Question.
I'm going to stick with the ANDAs because that's the program I'm most familiar with. I don't want to step into the BLA and say something wrong. We do see type 5 DMFs, especially when you get sterile API. And they do reference a type 5 DMF. It's a DMF, you need a letter of authorization. So yes, if you can reference a type 5 DMF. They would need to issue an LOA to their DMF. They would need to issue an LOA to the primary DMF. I don't know that they necessarily have to submit the LOA to the applicant because that LOA is specific to the primary DMF. They're usually, it's a sterilization facility. If the drug product, if the applicant referenced a type 5 DMF, then that DMF would need to submit an LOA to their DMF and to the applicant directly if they're actually being used by the applicant.
Thank you very much. Dr. Madhu, is the secondary DMF submission really required?
No, it is the primary DMF holder decision if they want to refer to the secondary DMF. What do you mean by, sorry, it's an online question. Yeah, because the primary DMF holder choose to reference the secondary DMF for their manufacturing process. So they do need to LOA? Yes, of course, they need to provide the LOA. You want to go ahead?
Yeah, so if I may jump in. So the DMF is never required. That's the first thing to consider. I think maybe what they're getting at is if you are purchasing an intermediate from another company, that if you are not incorporating all of the data that we need to evaluate that intermediate in your primary DMF, then yes, you should consider referencing the secondary DMF if it's available. You may incorporate all of the data into the primary and we could assess it there. But if you do not, or they aren't sharing that information, then they should submit a DMF and you would reference it as a secondary DMF. Thank you.
Thank you both. So Dr. Skoda, this question touches upon an answer you responded to, but maybe it's phrased differently. What is your best practice recommendations for ANDA applicants and DMF holders to ensure an efficient and as seamless as possible review? I know you mentioned communications was the biggest gap that you've seen.
So what is my take-home message? Is that essentially what you're asking? I'm sorry.
What are the best practice
Best
recommendations?
practices. Okay. I think that understanding that a DMF holder and an applicant are two different entities and that you can't communicate everything is something that is tricky. However, if you can communicate review timelines, if you're aware of them, or review issues that pop up in a letter, I think that that goes a long way to ensuring adequate communication. So for example, if a DMF holder has an issue with an API that they're trying to address, it would be helpful to inform the applicant that, you know, they're working on it and, you know, give them a timeline so that they can plan for their own responses. So I think that what you can communicate should be communicated just to help the review process along and to help coordinate submission of information to the agency. And then also refer back to my slides, and I recommend that you incorporate as much as you can into your submissions.
Excellent, thank you. And I think we have two questions in the room. We'll start with the back question first.
Hi, I have a question on the API review process. So if an applicant chooses to include the API information in the ANDA or NDA instead of referencing a DMF, is the review process any different, or is it still similar to how a DMF review happens?
Yes, so again, a DMF is not required, and so we do in some instances see where all the information is submitted directly to the application, and our review is the same. It's the same quality review. It's just done in the application. I mean, the way we communicate, I mean, I think it goes out as a larger letter, where the DMF holder's like, you know, specifically, here's, you know, your API questions. If it's in the application, you're more of a team. So your responses go out in a bigger letter, but we don't change the way we review. We don't change the standards or the quality or the way we do it. We still hold to the starting materials and impurity profiles. There's just no difference in the way we review the API, whether it's in a DMF or in the application.
Just one question. How about the completeness assessment? Is that still happening if it's part of the ANDA?
It's a different, so under GDF, if the drug substance information is submitted in a type 2 DMF, there's a completeness assessment that's completed. If the drug substance information is in the ANDA, we do an assessment as part of the filing review of the ANDA. I believe it's in section A2F of the GDF commitment letter.
Thank you.
But the content of that assessment is very similar.
All right, thank you.
Yeah, as a continuum like that, people always wonder about that if your API information is in the ANDA itself, are you taking the same checklist and doing it, or is it a little different? Because, you know, one thing is DMF review is completeness is done by an expert. You know, in the RTR, completeness is a different kind of expertise. So, but I was, since the question of communication came, I thought I'll raise this. One of the confusions I see, and this is mostly with complex generics, which are complex because of the API, that, you know, there have been situations where the API manufacturer, where the ANDA holder, because they bought the API, they looked at everything, said, please refer to the DMF for the characterization, and they got a letter in the ANDA saying, no, you need to submit it here. And I have also seen the opposite, where they like literally dumped that information in the ANDA, and the ANDA reviewer said, no, this should not be here. Please go and reference your DMF. Is there a specific best practice on that? Because looks like there is a bit of a confusion based on reviewer, I guess.
Yes, so in the characterization section, they can refer to a DMF. They should probably, though, at least do an ID test. I mean, something like an IR, just to make sure that. But yes, you can refer the characterization data back to the DMF, and that is allowable.
All right, if there's no further questions in the room, we can turn it over. I think we have time for a few more questions. One, okay, online, okay.
Okay, Dr. Madhu, does a secondary DMF have to go through completeness assessment, paying a GDF fee, and being ready for reference if the primary DMF is used for an ANDA?
The DMF doesn't go through the completeness assessment, right? No, it doesn't go through the completeness assessment. I think secondary DMF doesn't need to pay the fee. Yeah.
Thank you very much. Let's stay with you, Dr. Madhu. Keep your mic on. If secondary DMF is API or any API material is available in USP monograph as well, can we use it as a general raw material to design specifications, or do we have to adopt USP monograph specifications during manufacturing process or synthesis of drug substance? Parentheses, primary DMF. Please clarify.
Okay, if the drug substance in the secondary DMF has a ESP monograph. Again, the ESP monograph specifications are kind of must or minimum for any drug substance specification. Again, the specifications and impurities depends on the manufacturing process. So if the manufacturing process does not address some of the impurities, then they need to address the control of those impurities. So USP monograph, I would say it's a minimum, but it depends on how you manufactured the drug substance.
I'm just jumping for a second. So if you've got a secondary DMF and it's not the API, let's say it's the base, and then they're going to make the salt or something. So it's not the API. The USP monograph, I think, is a good place to start, because when you go on to the next DMF, the primary, I mean, they're going to have to deal with this. So you would like to have your material be as good as possible, but it is not the API. So the USP monograph would be a good place to start. But if it's an intermediate, it's kind of also an intermediate. It's not the final. So, things like ICHQ3A don't apply, M7 don't apply, unless that is the only place the impurity is being controlled. Let's say that's the only, in the secondary DMF, it's the only place the impurity is going to be controlled in that intermediate. It's not going to be controlled in the other one. Then, things might be different. But if it's an intermediate, we treat it as an intermediate, whether it's an intermediate in the actual primary DMF or the intermediates in the secondary DMF.
Okay, thank you. I think we have time for one more, do we?
Sure, we have time for one more.
Okay. Dr. Johnson, for an API, if I have the stability testing site listed in DMF section 3.2.S.2.1, do I also need to have the stability storage site listed if different than the testing site? Or is that only required for a drug product?
See, this one always gets me. Yeah, because there's the testing facility, there's the storage facility. I want to say the storage facility still has to be in 2.1, but I could be wrong. That's a good question to send to CEDAR OPQ inquiries.
Okay, yeah.
I don't want to say wrong. I don't want to lead people down the wrong path.
So to whomsoever sent in that question, please send it to email to OPQ inquiries.
Yes.
Thank you.
That will eventually come to us. But we'll be able to look things up so we give you the right answer.
Thank you much.
Well, then I think that's a good ending place there. So we can go ahead and start the break now. So it will be a 15-minute break going until 10:45. If you'll rejoin back in here, we'll start at 10:45 on the dot. Just a reminder that if you did purchase a lunch here on site, you can pay at the kiosk. That helps just get ahead so you can pick up your lunch later and make that a bit more expedited. Bathrooms are also, if you go down this hallway past that main registration area, there will be a signage that points you to the bathrooms over that way. Thank you. All right, we are at 10:45, so I think we're good to get started here in the room. Everything sound okay? Sounds good? Okay, excellent. Well, we are good to go ahead and get started with our second session of the morning here. First, we are going to hear from Dr. Sherry Bai, a chemist in the Office of Product Quality Assessment 1 within the Office of Pharmaceutical Quality, or OPQ. Then we will hear from Dr. Arlene Chen, pharmaceutical scientist in the Division of Pharmaceutical Manufacturing 3, or DPMA 3, in the Office of Pharmaceutical Manufacturing Assessment, OPMA, within the Office of Pharmaceutical Quality, Pharmaceutical Quality. Then we have Amit Kokate, Dr. Amit Kokate, senior biologist in DPMA 3 within OPMA and OPQ. Then we have Vicky Hay, senior chemist in DPMA 3, followed by Dr. Kay Wren, Deputy Division Director within the Division of Bioequivalence 3 and the Office of Bioequivalence and the Office of Generic Drugs. If you'll now join me in welcoming Dr. Sherry Bai up to the stage. Thank you.
Good morning, everyone. Thank you for joining us for today's session. And I'm going to discuss about pre-ANDA meetings for complex drug products, specifically focus on do's and don'ts from the quality perspective. So overview, I'm going to show you what I'm going to talk about for the next 20 minutes. So I will touch on the strategic use of the right communication for pre-ANDA communication, and then define what makes a drug product complex, and go through some pre-ANDA meeting package contents and stages. And we'll discuss something about like do's and don'ts for pre-ANDA, for the pre-dev and pre-sub meetings, basically pre-ANDA meetings. And then I will wrap up with takeaway message. So first, I'd like to show you what makes a drug product complex. This topic maybe looks familiar to you because I think Dr. Yan Wang already touched on this yesterday for the PSG topic. But I would like to go over this because my pre-ANDA meetings are mostly currently limited to complex drug products. So I'd like to review this concept. And so under GDF 3, the complex drug product categorized in 5 categories. And first, complex active ingredient. So these active ingredients are not simple single chemical entities, such as glutamate acetate, protamine sulfate, and also complex formulation. So for this, the drug is generally delivered via sophisticated formulation or in some like physical form, like iron sucrose, propofol. And also some drug products with complex route of delivery, such as locally acting drugs like desonide emulsion, prednisolone acetate suspension, brimonidine gel, and also complex dosage form, such as transdermal systems like fentanyl patches, and meter dose inhalers like albuterol. Also, the complex drug device combination. For this drug product, generally it consists of both drug and a device. So whether the device is integral to the drug delivery, safety or effectiveness, such as epinephrine, autoinjector, and as also like Dr. Yan Wan mentioned yesterday, all these complex drug product may fall into multiple categories. So, okay, with this concept in your mind, I'd like to show you some pre-ANDA communications. Actually, from yesterday, you may already, there are already some topics on PSG, control correspondence, CC, and also suitability petition. And for me, because I'm focused on product quality, so the product quality is generally outside the scope of petition, suitability petition. So I don't have that suitability petition in this table. But if you like to get details about like PSG, CC, or suitability petition, you can go to yesterday's talks. There are several excellent talks there, and I learned a lot from them. And okay, so for this table, generally, I just want to give you some like guidance, how to navigate all this helps you got from the agency. For the PSG, as mentioned yesterday, it is basically a blueprint. It is your fundamental resource. From here, you can find the agency's current thinking, the established recommendations on bioequivalence, pharmaceutical equivalence studies. And for when you have some target scientific questions, it is not answered in the PSG, you can submit controlled correspondence. It is best for the narrow specific scientific or regulatory questions and do not require in-depth data review or discussion. But if you have like complex issues, may not be addressed effectively in the CC. So these formal meetings are reserved for this when the control response is not adequate or the meeting would improve assessment efficiency. Then this time you can request formal meetings. And here I'm going to, my topic is going to focus on the pre-ENDA meetings, basically the pre-development meeting and also pre-submission meetings. So these slides, I'm going to show you the meeting package contents. This is information required to be in the package, including product identification, meeting request information, and development background. And there are several sub-bullets here. You can find all this information from the guidance I showed here. That is the formal meetings between FDA and ENDA applicants of complex products under GDF. So from here, you can get all the detailed information because these are required. So you need to pay attention to this. Otherwise, you may get denied. Your request may get denied if the package is not complete. And here, I'd like to show you the key features of the product development meeting and the pre-submission meetings. So for PDEF meeting, it is eligible for complex drug product without PSG, or the product with PSG but alternative equivalence evaluation, just like what mentioned in yesterday's PSG meeting. For what recommended in the PSG meeting are not requirements, they are like recommendations. So if you have alternative approach, you can always submit request for product development meeting to discuss about it. So as I mentioned earlier, this is requested when the control correspondence cannot be adequate to address the questions, and the meeting can significantly improve and assessment efficiency. And for this meeting, generally the format is face-to-face, video conference, or written response only. And I put the grant denied decision notification date. It's within 14 days here. And for the meeting, generally will be scheduled like 120 days from the grant. For pre-submission meeting, it is for the applicants who have already submitted PDF meeting before for the same drug product. And generally, we recommend you to submit requests like 6 to 8 months before anticipated ENDA submission. And this meeting generally is like face-to-face or video conference. And the grant denied decision will be notified within 30 days. And for both meetings, you will get official meeting minutes like 30 days from the meeting. Okay, so let me talk about like PDEF meeting, the do's and don'ts. We encourage you to focus on your developed strategy and data, present some preliminary data with adequate justification to support your proposal. You can ask a series of specific questions requiring like targeted scientific advice on the B or P study design. What we don't want to see is like if you question meeting like for non-B or P specific elements, the PIG is about B and P. So if you ask question not B or P, generally this is not what the PDEF meeting for. But also you are not encouraged to ask for like review issues, but this is not for review meeting. Generally, if you have a review issue, generally will be dealt with during ENDA review. And also for the package, please do not bring the new data or surprise in the last minute after you submitted the package. And also, like as I mentioned, this is not for assessment, so this is you cannot really expect like definitive approval of your approach. We'll give you guidance, but not like approval for the meeting. So basically, to summarize, generally the common reasons for PDAP meeting denial generally is like some questions are too broad or vague, not specific, not clear, and also questions seeks assessment decision, like I mentioned earlier. And also questions is actually can be better answers through control correspondence, like simple questions or like not complex questions. You can get quick question, quick response through control correspondence. And also like insufficient data provided to support your proposal. And product do not meet the criteria for complex drug product. And also questions not really related with B or P issues. So for pre-submission meetings, do's and don'ts, and we like you to provide information per the PSA meeting presentation outline template. It is in the guidance, and present some like unique or novel data or information that will be included in the ENDA submission. Here I mean like unique or novel because we already have some like a product development meeting before. So if you have some new data you like to let us to look before the submission, yeah, you are welcome to provide information here. And also in your packet, please include regulatory history or summary of generic drug development. And again, you need to have like prior PDAP meeting for the same product. So what you don't do is like for PSA meeting, it is not question-based meeting. It is not like opportunity for you to determine whether the application is acceptable or for receipt. And it's also for product. You should have, if you don't have a PDAP meeting, then PSA meeting will be generally denied. And also for non-complex drug product, it will be denied also. So here, I'm going to show you some like appropriate PDAP questions just to give you like sense what should be asked in the, would be appropriate for the PDAP meeting. Like this question, can the agency clarify whether proposed physical chemical characteristics and analytical techniques or methods are considered acceptable for demonstrating sameness between the proposed test product and the ROD? So this is generally, we receive this kind of question a lot in the PDAP meeting. And from the response, this is also like a generalized response you will see from the agency. We'll comment on your general approach. If it is reasonable, we'll say, okay, your approach proposed for the comparative characterizations appear to be consistent with the draft guidance, that is PSG. However, then we'll give you some like recommendation for improvement, like the adequacy of the characterization method is an assessment issue and will be determined during the ENDA assessment. We have the following additional common recommendations for you to consider. So you will receive like a list of recommendations for you to improve your the quality of your ENDA. Please note that this question is appropriate because it seeks clarification on approach, not definitive approval. Okay, so I prepared several questions just to let you know, get some sense like what are the appropriate questions. So I will give you a test of this. So this is like a practice. So the first question, does the agency agree with our approach for comparative physical chemical analysis of the test product and the ROD? The second question, can the agency confirm that the choice of sample preparations for comparative physical chemical analysis of the test product and the ROD are considered suitable? What do you think? For these two questions, it's kind of borderline questions for whether it's appropriate for the PDF. It's kind of borderline. But for this question by itself, actually, you can submit it in the control correspondence. That would be more effective for you because you will get response quickly, like written response quickly. But if this question is like you have like some others, multiple interrelated questions, the most effective way would be in the PDF meeting. So it really depends. And I have like a few more questions for you to practice. For this one, the first one, does the agency agree that the proposed parameters for the product specification are sufficient to control drug product characteristics and release and throughout the shelf life? For this one, basically this is like a clarification to see if the agency agree with their control strategy. For this one, generally we'll see, yes, we'll address this question in the PDF meeting. And then the second question, does the agency agree that at least A is performed only as part of the comparative categorization between the test product and ROD and not as part of routine release and stability testing? This, whether one us to confirm or not, this generally I would put it into the control correspondence. It will be efficient. And for this one, last question is like clarifying questions, like whether in use testing parameters are sufficient to confirm compatibility with a diluent. Then for this one is because the strategy, while speaking, that is good to be in the PDF meeting. For the agree or not confirm, all this question is like will be most effectively to be in the control correspondence. Okay, so this here is the challenge questions. Hopefully this is obvious to you. The applicant is developing complex generic drug product. Which question is not suitable for PDF? A, does the agency agree our proposed approach of using these techniques to evaluate secondary structure of peptide is in the test DP or RS with respect to the active ingredient sameness? B, can the agency clarify whether the proposed in-use stability study protocol for implant devices is considered reasonable to demonstrate compatibility? And C, does the agency agree on the proposed limit for impurity X? Hopefully, I made it clear to you. The question, we says the right answer would be C, because C is for the question it's like an assessment issue, not really, not for clarification or not for discussion. So this is the reminder. Generally, we will in the response, we'll always give you the advice that the final decision regarding adequacy of any supporting data or information will be determined once ANDA is submitted. And additional information may be requested during the application review process. So this slide shows the resource, from all this reference I talked in the slides. In summary, so the key takeaway is, we have several pathways for you to use that is better for the strategic and effective FDA engagement. And it will help you prevent you now for the meeting request. And for the PDAP meeting is for B and P approaches during development. And the PSAM meeting is to present some unique and novel information that will be included in the ANDA submission. So remember, still the final decision on adequacy of data and information are made during ANDA review. With this, I'd like to acknowledge my supervisor, Dr. Cameron Smith, and my dean director, Bing Kai, who helped me prepare the slides. And hopefully my talk will be helpful for you while you navigate with this pre-ANDA meetings. Thank you. And with this, I'd like to introduce our next speaker, Arlene Chen.
Hi, my name is Arlene Chen, and I'm a food pharmaceutical scientist in OPQ. My background is in microbiology, and so my work is focused on the quality assessment of both sterile and non-sterile drug about some of the common microbiology deficiencies for ophthalmic suspension drug products. So this is just our disclaimer stating that all opinions expressed in this presentation are based off of my interpretation of current regulations and guidance documents, and they don't necessarily reflect the views or policies of FDA. So an overview of my presentation. First, I'm going to give some background on manufacturing processes of ophthalmics. And secondly, we'll dive into some of the top issues that are focused or that are found during a quality microbiology review. So as I'm sure many of you are involved in the manufacturing of ophthalmics already know, the manufacturing of ophthalmic solutions can be quite complex. The majority of ophthalmic suspensions are filled using aseptic processing because terminal sterilization is not really feasible. To add to the complexity, the manufacturing process typically includes a combination of different sterilization processes to account for the soluble and insoluble phases that are later blended together to create this sterile suspension. This means that there's a combination of sterilization processes, including filtration, steam sterilization, gamma radiation, that each need to be optimized and validated for each component in order to get your final drug product. In addition, if there's a sterile drug API, additional care needs to be given that the API powder might be exposed to the surrounding environment during powder addition. Not only that, because we're using aseptic processing, the containers, closures, and equipment contact parts that come in contact with the various drug substances, the excipient phases, as well as the final drug product, they need to be sterilized separately and validated accordingly. And then similarly, because we're using aseptic processing, an environmental monitoring program with controls should be in place to ensure appropriate environment for aseptic filling to occur. And this can mean that such equipment, such as isolators, are used to improve the overall environmental control. So this is just a flow diagram showing some of the different components and how they would need to be treated differently to achieve a sterile product. So if you have an insoluble API, they're typically sterilized at a contract facility using gamma radiation or ethylene oxide, or if the API is heat stable, it can be created into a slurry, which then is sterilized in a small vessel using steam sterilization. Vehicles such as water for injection or WIFI or your purified water or other insoluble or soluble excipients, they can be sterilized by filtration and collected into a mixing vessel or which is then sterilized by moist heat sterilization. Also, viscous components, that are difficult to filter, you may also sterilize them with moist heat sterilization in an autoclave or within a manufacturing vessel. Or they can also be filtered at high temperatures. And then lastly, your container closure system can be sterilized in a variety of different ways, including gamma radiation, ethylene oxide, and moist heat sterilization. So for my talk, my colleagues helped to compile some microbiology information requests that were issued from 2019 to 2025 for ophthalmic suspension ANDAs, and an analysis of the 108 IRs that revealed some critical sterility gaps. As you can see from this pie graph, there's a decent range of issues, including IRs related to the overall sterilization validation, hold times and process controls, media fill, aseptic process simulations. And then just as a side note, a lot of these types of IRs are common to all types of sterile drug products. It is not just focused on ophthalmics. It's just that a lot of the times these ophthalmic products use a combination of these different types of sterilization techniques. So let's get into more details on some of these IRs. So let's start with container closure integrity testing. So typically the gold standard test is microbial immersion. method. So in this method, your container closure system is filled with a growth medium, and then it's submerged into a microbial suspension with added pressure or vacuum. The container closure system is then removed and then incubated and checked for growth. Another method that is used is dye ingress, where the container closure system is filled with water or the drug product and then immersed in a dye bath. And this is typically like methylene blue and also exposed to pressure and vacuum. For this method, the amount of dye that is entering into the container closure system is measured. And that's either objectively using a method like spectrophotometry or subjectively, such as through visual detection. And that's like looking for a visual color change. So some of the deficiencies that are issued regarding these methods are due to method sensitivity and limit of detection of the method that is used. So the question is like, is the method as sensitive as the gold standard microbial immersion method, or is the analytical method subjective or objective? When you're visually looking at a breached unit, is there going to be a different impact based on which analyst is actually looking at the container closure system? Similarly, there's some advanced technologies that might be used for CCIT testing, and this could include high voltage leak testing, helium leak testing, and vacuum decay leak testing. And all of these have different types of limitations in these methods as well. So another common area of deficiencies relates to antimicrobial effectiveness testing, or AET. So ophthalmic suspensions are typically multi-dose, and that means that they contain a preservative. And so a lot of the times when we're looking at the stability data, the preservative content that was not at or below the minimum stability specification. So this data is important to ensure that the preservative is working throughout the proposed shelf life of the product. Therefore, we want to ensure that at least one primary stability batch is tested for AET at or below the minimum preservative specification. Component bioburden. So common deficiencies related to component bioburden include performing bioburden testing after filtration or other sterilization processes that have occurred. The pre-sterilization bioburden testing of all phases should be performed in order to get an accurate depiction of the overall bioburden of the product. And additionally, if excessive bioburden limits are used, a rationale should be used to justify the limits that were proposed. Hold times and process controls. So common deficiencies issued were related to not describing the maximum hold time for each manufacturing stage. And similarly, if an excessive hold time was proposed, supporting microbiology studies were not provided to confirm that unwanted microbial growth did not occur during the proposed hold times. So ensure to include those studies with your submission. The sterilization of container closure. So a lot of the time, sterilization of these systems are sterilized by gamma irradiation or ethylene oxide. And these studies, the common deficiencies that were issued were related to not performing them according to the standards recommended in ISO 1137 or ISO 11135. In addition, the IRs were issued due to the lack of recent requalification studies, dose audits, and bioburden results. So if you're using these methods, make sure to review the ISO standards in order to make sure that you have the appropriate dose and also the number of samples that are used during your validation. In addition, if there's been some time since your initial validation was performed, make sure that you include a recent requalification. Sterilization of manufacturing equipment. So one of the main deficiencies is that the validation was not performed with an appropriate number of runs for all equipment. And this is all equipment that comes in contact with a sterile product. So that could be your vessels holding the sterile components, the filling equipment, etc. And then also, as I mentioned earlier, not providing recent requalification results. So for moist heat sterilization, heat sterilization may be used to sterilize heat-stable excipients or API if it's in a slurry. And similar to what I said earlier about the sterilization of equipment, the excipients and API also need validations that have an appropriate number of runs within that validation. And then if there's been some time between the initial validation, to make sure that you include a recent requalification. Another IR that is commonly issued is that if the volume filled for that vessel is not representative of the commercial production, you want to make sure that you include that. And that might mean that the studies that were provided were using the exhibit batch size instead of the commercial batch size. So the volume filled is important because you want to know the impact that the heat and how heat is transferred throughout that entire container. If the API is sterilized by gamma irradiation, you want to ensure that the studies are conducted per ISO 11137. And this means for the dose setting that you're either using method 1, which is taking the average bioburden of multiple samples, method 2A and B, which is a fraction of positives, or the VD max 25 method, which is typically what we see. Many of the deficiencies issued under these categories are because the ISO guidelines were not followed. If the sterilization of the API is done by ethylene oxide, again, they should be conducted per ISO 11135. And this means that either it is done via the biological indicator bioburden approach, which is found in Annex A, or the overkill approach in Annex B. And if you are having a contract facility performing these validations, you need to make sure to include those in your submission. So some components, such as the Wi-Fi or purified water or other soluble excipients, may be sterilized by filtration. Note that the drug product formulation and or manufacturing condition may also impact the filter effectiveness. So that means that if there is a change in your formulation, like in a supplement or such, that filter may be impacted. And so a new validation study may be warranted. In addition, some deficiencies that were issued were due to the lack of viability study results. If your product is bactericidal, you still need to include those viability study results so that we can look at them. In addition, another common IR is that if the specific, at least the product-specific bubble point, or BP, was not provided. So a lot of the times the manufacturer will include a bubble point, but that is within water. And so, as I mentioned earlier, sometimes the drug product formulation can impact the filter components. And so a product-specific bubble point determination should be included. In addition, at least one filter with the pre-filtration bubble point at or near the manufacturer's minimum specification was not used, and therefore received an IR. If isolators are used within your manufacturing process, some deficiencies that are included are not, again, not having the appropriate number of runs. And I know that I've said that a bunch, but typically that means 3 or more. And also a recent requalification result was not provided. And also when you're including your validation of isolators, you want to make sure that you are putting your biological indicators in areas that are worst case. So media fill. When performing media fills, you want to ensure that an appropriate number of aseptic process simulations using the subject container closure system is provided. And typically, you want to make sure that if you're using a bracketing approach, that at least your subject container closure is within that bracket. In addition, if the maximum proposed filling duration is longer than what you actually demonstrate in the media fill, you want to make sure that you include a media fill that extends the length of the proposed hold time. So that means that if your hold time is 8 hours, you want to make sure that your media fill is not 7 hours and 50 minutes. Another common IR is that all the production operations were not simulated. And this is particularly true for aseptic powder fill. And this is because if that powder is sterile and it comes in contact with the environment that could cause it to become non-sterile, we want to make sure to encapsulate that. And then again, as I said in a bunch of other slides, if your initial validation has been performed quite a while ago, you also need to provide a recent requalification result. And typically for media fill, this would be every 6 months. So in conclusion, as I stated earlier, manufacturing ophthalmic suspensions is complex. And after compiling some of these common microbiology deficiencies, it's found that a lot of these submissions contain incomplete information regarding the different sterilization processes essential for demonstrating sterility assurance. And some of the common IRs include an excessive hold time or acceptance criteria, studies that were not performed according to ISO standards, missing or inadequate validation and or requalification data, and then overall media fill insufficiencies. So this was just a brief overview of some of the common deficiencies you might run into, but be aware that there's always going to be unique circumstances that could lead to other types of deficiencies. But in the end, make sure to check your FDA guidance and references to aid in your validation of your manufacturing process. So these are just some of the references that should be addressed when thinking about your submission. And then just a thank you to my coworkers, Dr. Judu and Jesse Wells, who helped prepare this presentation, and my supervisor, Ching-Ji Hu, and Division Director, Sharmista. And our next talk will be by Dr. Amit and Vicky Hayes.
Good morning, everyone. My name is Amit Kokate. I'm from the Office of Pharmaceutical Manufacturing Assessment in CEDAR, and I'm joined by my colleague Vicky Ha from OPMA as well. The topic of our presentation today is it too loud? Okay. The topic of our presentation today is common manufacturing process deficiencies that we've observed for complex dosage forms. Sure. Is this better? Okay. Specifically, transdermal delivery systems, TDS, metered dose inhalers, MDIs, and dry powder inhalers, DPIs. So this will be a joint presentation. I'll cover TDS, whereas Vicky will cover MDIs and DPIs. So this is the standard disclaimer slide. I'll start off with a brief overview of the manufacturing unit operations and the control strategy for transdermal systems, followed by the commonly observed process-related deficiencies and considerations for TDS. Vicky will then take over and she will go through DPIs, MDIs, the unit operations process risks for these dosage forms, as well as the commonly seen process-related deficiencies for the DPI and MDI dosage forms. Finally, we'll wrap up with the overall conclusions and recommendations. The manufacturing process for transdermal systems typically involves 3 unit operations: mixing, coating, drying, laminating, or simply CDL, and converting. So during the mixing unit operation, the drug substance and excipients such as adhesives, permeation enhancers are blended in the presence of solvents. This drug adhesive matrix is then applied onto a moving substrate, such as a release liner, during the coating process. And during drying, the thin layer is dried by evaporating the solvent in a multi-zone oven. During laminating, the individual layers are combined into a single laminate. The single laminate is then slit into individual smaller rolls during the slitting process. And during converting, this laminate is die cut into individual patches of the final size and shape. And finally, the individual units are heat sealed into pouches. This slide summarizes the typical process parameters that are observed during the transdermal manufacturing. However, in the interest of time, I'm going to skip this slide as well as the next slide. However, I think you will have these slides handy later on for future reference. The in-process tests during TDS manufacturing are summarized here. Again, I'm going to skip through the slide. However, we will be covering some of the in-process tests during in the next few slides. This table here represents a tree map of the most frequently identified process deficiency topics that we have assessed for about 16 applications in the last 5 years. As you can see, a majority of the process deficiencies centered around the serial and mixing unit operations. For example, in case of mixing, we had process deficiencies on bulk viscosity, mixing parameters, percent solids. During the serial unit operation, there were deficiencies on coat weight, the serial parameters, laminate defects. We also had deficiencies on other topics like converting topics such as the parameters used, yield, hold time, equipment compatibility, and inaccurate documentation. Now let's go over some example deficiencies, process deficiencies for the mixing unit operation. I'll also cover a few considerations that can be used to resolve these deficiencies. One common deficiency is we notice an increase in temperature during mixing as per the submission. However, an adequate control strategy is not included in the submission. So in this case, our recommendation is to implement a temperature control and upper limits for your process parameters to ensure that the temperature rise is not significant. Another issue we noticed is with redispersion of multi-phase systems where development data is missing to demonstrate that the remixing process parameters are adequate. So in this case, consider performing development studies to demonstrate that remixing or the remixing process restores the dispersion to its original state. Also consider implementing appropriate mixing controls and maximum hold times. One common topic is around bulk viscosity test, in-process test for the drug adhesive matrix, which is either not proposed or in some cases the limits are not adequately justified. So our suggestion here is to implement a bulk viscosity test with meaningful limits in your in module 3. When it comes to percent solids or solvent contents, which are kind of unique in process tests for transdermal systems, we've noticed that the solvent content in the adhesive matrix is not adjusted, or sometimes the percent solids have a very wide range. This could result in potential variability in the composition, which in turn could have a significant impact on API solubility or processability. So our suggestion is to adjust the solvent content in the batch formula so as to achieve a target percent solids within a tight limit. Also for dispersions, there is a risk of aggregation, as most of us are aware of it. Like there's a risk of aggregation, risk of phase separation. So in this case, the risk of these issues should be evaluated during your product development. And also appropriate process controls such as visual appearance, microscopic globule size distribution, or particle size distribution tests should be considered. Now moving on to the CDL unit operation, which is probably the most critical unit operation during transdermal and topical delivery system manufacturing. We see most of the deficiencies in this particular unit operations. I mean like process deficiencies. So one common deficiency is where the critical process parameters like line speed, zone temperature, airflow rate are either not defined or they're simply listed as TBD, to be determined in the batch record. So consider evaluating these process parameters during development studies. Also propose appropriate ranges which are justified based on development studies. Moreover, sometimes the critical process parameters are established or are proposed in the submission. However, when we go through the product development report, we realize that the CPP ranges were established based on a prototype or a development formulation. So in this case, you should verify that the process parameter ranges that are being proposed are still applicable to your final commercial formulation. Because sometimes we've noticed that the commercial formulations are drastically different than the development formulation. One other issue is coat weight, which is one of the most important in-process tests for transdermal system manufacturing. The sampling strategy or specifications are inadequate. Although this is an application-specific topic, the minimal expectation when it comes to coat weight sampling is that the sampling should cover the entire CDL process, and it should like enough samples should be taken from the rolls to evaluate the variability in coat weights throughout the manufacturing process. Moving on with few more deficiencies during CDL, certain transdermal products contain rate controlling membranes. Not all of them consist of a rate controlling membrane. But the ones which do have a rate controlling membrane, as it's a critical component, the defect of assessment of that particular membrane should be discussed in the submission, because having a pinhole or a tear or some other sort of defect in a rate controlling membrane may result in dose dumping or drug delivery issues. So adequate controls, which include visual inspection of the raw material, the membrane itself, should be included in your submission. One more issue is the presence of significant defects, such as streaks or voids or bubbles that are observed in the laminate during the exhibit batches. If that happens, the defect should be identified and the root cause should be addressed in your package. Most of the times we've noticed that these kind of defects are a result of either the mixing or coating process, where like during the mixing compounding process, maybe the mixing is either inadequate or the deaeration is inadequate. When it comes to coating, the coating equipment setup may not be appropriate, or sometimes we may even have defects in the coating equipment, like wear and tear. Volatile components are commonly present in transdermal systems. And the formulations which do have these kind of components, and an example of that is volatile permeation enhancers. A big challenge is evaporation of these components during the drying process. And this could result in CQA variability, like variability in the assay, variability in the drug release. So a common development strategy that we recommend here is to evaluate the risk of evaporation of these components. You may consider performing development studies like design of experiments to assess the impact of the coating, drying, laminating process parameters on the evaporation potential of these components. And additional considerations include, like especially if you're justifying excess excipients to compensate for the loss, enough data should be provided, like adequate data should be provided in the submission to justify the excesses. Moreover, consider humidity of the drying air during the coating process, because that could be a potential source of variability. This is my last slide. Like when it comes to converting, I have one deficiency here, common deficiency that we see where IPCs for release liner functionality and pouch integrity are missing. We consider these to be important IPCs during the converting unit operation, so our recommendation would be to include these. When it comes to scale-up equipment change, there could be an equipment capacity utilization difference between the exhibit batch and the commercial batch. So the impact of the change in equipment capacity should be evaluated, and also appropriate in-process control should be implemented. In terms of incomplete solvent removal, this could result in poor or variable adhesion. So our suggestion is to revise the residual solvent limits based on the formulation and process capability. For yields, when you have low or variable yields, although it's not specific to TDS, suggestion is to provide justification and mitigation strategy. This concludes the TDS portion of my presentation. I'll hand it over to Vicky. She'll go through DPIs and MDIs. Thank you.
Okay, so I'm going to switch gears and talk about dry powder inhalers and metered dose inhalers, and both are oral inhalers. Long button, sorry. So as illustrated by this schematic here, many types of dry powder inhalers or DPIs has been commercially available. The blister and capsule DPIs have been the main generic submissions to the FDA since 2015. So this section focuses on these DPIs. So for the capsule and blister DPIs, the key manufacturing unit operations include powder blending, powder filling into the capsule for the capsule DPI, and then powder filling into the blister strips for the blister DPI. And then for the blister DPI, the blister strip is assembled with the device during the commercial manufacturing process. Versus for the capsule DPI, the fill capsule is primary packed in a blister car. And then during usage, the patient would just take that capsule out and put it in the device. So some examples of process risks on drug product critical quality attributes can include the following. For example, high product uniformity risk due to the low drug loading and potential blend segregation, because normally for capsule, it's a very small capsule filled with a very small amount of powder, and same with the blister strip, it's a very small amount of powder filled into a very small cavity. So, you know, and then the drug load is low, so product uniformity is definitely a key risk for this type of manufacturing process. And then also the, you know, the physical damage of the blister strip during filling and sealing, misalignment of the components during device sub-assembly, also misalignment of the blister strip with the final device during assembly
can also affect the drug product CQAs. So one common deficiency for the powder blending process is missing justification for the blend uniformity sampling size. So a key consideration is to provide the details on the BU test sampling location, the size, and the acceptance criteria. Many times during the huge dilution of the excipient versus the drug product in the formulation, and also trying to sample a very small amount of powder from the blender, sample variability may occur. So if a sample size greater than the typical 1 times to 3 times unit dose weight is required to minimize the sampling error, make sure you provide a justification. So for filling, a common deficiency is insufficient individual net field weight control information. Now, field weight control is critical to ensure accuracy and reproducibility of the dose, especially we are talking about microdosing here. So a key consideration is to provide the details on the individual net field weight test method, whether it's an inline automated weight verification system based on capacitance, detection of the powder falling, or it's a manual offline net field weight check. Make sure you provide the details of which one you're using, and then if it's a inline automated field weight verification system, make sure then there's adequate calibration for that method. Then again, of course, the acceptance criteria for the net field weight limit should be justified and supported by the exhibit batch and development data. So also for filling, another common deficiency is insufficient in-process stratified CU test information. So, provide the in-process stratified CU test data with the statistical sampling plan and acceptance criteria. So, FDA has published a guidance on BU-CU testing for oral solid dosage to replace the withdrawn guidance, and ASTM it's recommended for for the sampling and acceptance criteria. But if you are using an alternate method, make sure you provide a justification. So the data is required to demonstrate that product uniformity is achieved throughout the filling run, and then a blend segregation has not occurred, and that's why due to the potential segregation during filling, just providing the BU data by itself is not enough. So let's now take a look at the meter dose inhalers. So we are moving from powder inhalers to liquid inhalers. So for the MDIs, the key manufacturing process steps include liquid compounding, the valve crimping and filling, water bath stress testing, layover or quarantine period, functional testing of the valve performance, and then finally, the fill canister is assembled with the inhaler. Examples of manufacturing process risks on drug product CQAs include propellant evaporation to the pressurized manufacturing tank, to that headspace. Due to the evaporation of the propellant, then the concentration of the drug and propellant in the both solution then changes, so that could affect your drug product CQA. Improper crimping pressure, right, could then result in valve damage or propellant leakage. And then, of course, poor mixing and sedimentation can lead to high product CU risk, especially on the low drug loading suspension systems. So for a common deficiency for compounding and filling, I mentioned a key manufacturing process risk being a propellant evaporation to the pressurized tank headspace, and that's actually also a common deficiency that we see is inadequate justification for the propellant compensation. So a key consideration is, the proposed propellant compensation during the compounding and filling should be supported by theoretical calculation, and also it should be verified by in-process test data before and after each propellant compensation point. Also for compounding and filling, another common deficiency we see is incomplete critical process parameter information. So the CPP information is required to demonstrate that quality is built into the manufacturing process, and it's not just based on product testing. So make sure to provide the necessary CPP information in the submission, and the limit should be supported with, with development data or exhibit batch data. So this slide summarizes other common deficiencies for the MDIs. While they're all important considerations, I want to highlight the second one, which is insufficient product uniformity data. Now, the MDI manufacturing process is a big unit that sampling from the compounding vessel is very challenging due to the pressurization of the system. So, then it becomes very important to provide in-process test data with representative sampling throughout the filling run or the packaging run to verify that uniform mixing and filling have been achieved during the process. So in conclusion, the goal is to implement a control strategy that can assure consistent manufacture of product with desired quality and also mitigate any manufacturing and scale-up risk. I may talk about the transdermal systems. I mentioned briefly the MDI and DPI manufacturing lists and common deficiencies. The key consideration is to provide enough data to support the proposed process parameter ranges and implement appropriate in-process controls to enhance detectability of any variability, any off trend, and mitigate the risk. And the goal and the take-home message is really to provide the necessary information, and this really helps the submission process and also the review process in terms of minimizing the information requests and also and eliminating unnecessary review cycles with going back and forth with the IRs. So with that, thank you for your attention. I'd like to acknowledge everybody, all the colleagues, including the supervisors and the division directors that have contributed to this content. Next, I'm going to turn it over to Dr. Kiran. Thank you.
Good morning, everyone. Thank you for joining this morning's section. So I will be presenting data integrity and the data quality issues in abbreviated new drug applications. So this presentation has 4 key learning objectives. So you will understand the fundamental principle of data integrity in ANDA submission. And you will recognize the responsibility of sponsor, investigator, and CRO have in maintaining the data integrity through the study life cycle. Through the case study, you are able to identify the common data anomalies and the integrity issue we encounter during our bioequivalence review. At the end, you will comprehend how FDA responds to data integrity violation from regulatory perspective. So let's start with the foundation. What exactly is data integrity? Data integrity refers to accuracy, completeness, and the reliability of the data throughout its life cycle. At FDA, we follow the principle of ALCOVA, which stands for the attributability, legible, contemporaneously, original, and accurate. Those principles can ensure the data you can able to trace the original source, is clearly readable, it records what exactly happened during the activity, and it exists in the original record or have a truly verified copy, and is free from errors. So ensure the data integrity is fundamental to maintaining the quality, compliance, and trust in the regulatory process. So you can refer this kind of guidance in our published in April 2024, data integrity for in vivo bioavailability and bioequivalence. So why does the data integrity matter? Let me explain from two perspectives. From positive standpoint, the strong data integrity ensures the safety, efficacy, and the quality of the drug product. It supports the FDA ability to evaluate drug application and protect public health. Maintaining data integrity represents the key responsibility of pharmaceutical company and the CRO. It's essential, it's not an option. On the other hand, the consequences of the poor data quality are severe. The poor data quality undermines the FDA ability to conduct appropriate regulatory analysis. It may result in FDA issuing untitled letter to the CROs. It can lead to medication failure and delay in patient access to generic drugs. It causes loss of credibility for the organization involved, along with financial burden from having repeated the study or third-party audit. To illustrate the scope of the data integrity, let me share some historic context. So this table shows FDA notification regarding the data integrity concern at different CRO across the past 15 years. This timeline demonstrates that data integrity is constantly concerned. That requires a constant rigorous at the different from the different stakeholder. So how do we ensure the data integrity in application submission? The responsibility is shared between investigator and the sponsor. Investigator must allow monitoring, auditing, and regulatory inspection. They must provide a direct access to the study records and maintain procedure to ensure the integrity of the study activity. They must update their process continuously to maintain the compliance. On the other hand, the sponsor must implement quality assurance and quality control system along with written standard operation procedure. They need to ensure bioavailability and bioequivalence study complying with protocol and regulation. They must maintain access to the sites, source, document, reports, and apply quality control across all stages of the data handling. So these requirements are provided in the ICH E6, the Good Clinical Practice Guidance. So when FDA conducts inspection, OSIS reviews the inspection findings to assess compliance and potential data integrity concern. Based on the evaluation, the inspection are classified into three categories. The first no action indicate, NII, means no objectable condition or practice are observed. The facility is in the acceptable state of compliance. The second one is a voluntary action indicate, VAI, where objective condition and practice are observed, but facility can voluntarily correct deficiencies. The third one is official action indicate, the most serious one, where regulatory and the administration action are recommended because the facility is in the unacceptable state of compliance. So OII's classification indicates significant corrected action and remediation effort to address identified deficiency, include data integrity issue. So within the Office by Equivalence, our review division have responsibility to evaluate data integrity assessment. Our responsibility is to evaluate whether the data submitted in the application are reliable. If we identify potential concern, we will request for cost inspection to OSIS for further investigate. So what triggers this kind of concern? There are all types of the data anomalies. This include overlapping pharmacokinetic profiles or unusual PK trend across the subject. We also alert the situation where we are suspicious of switching the test and reference the standard study sample or reuse the study sample or make the dilution to make the study pass the BE criteria. We also examine questionable the repeats that did not follow the appropriate procedure. So this flow chart illustrates we used our systemic approach to investigating the study data. It also shows our collaborative process among the review division, BISTATS, and the OSIS. First, we use the software tool and other bioanalytical method to detect the data anomalies. If we raise the concern, we will request for cost inspection. Secondly, we carefully evaluate analytical reports, especially reanalysis sample and ISR, and check whether consistent reporting the data across the documents. In addition, in order to understand the nature of the similarity or unusual PK trend for this particular drug product, we also use the concentration data from other ANDA for the same drug product conducted at different sites as a control. So when we found the concern, we definitely will issue deficiency to ask the applicants to explain. So case study. The case study 1 involves data discrepancy. So in this case, in the fasting study, we suspect potential bias in the study due to discrepancy in reported concentration value across different reports. For example, let's look at this subject A with the reference treatment. We identify discrepancy reported value in analytical raw data, in the SAS dataset, and in the analytical report from sampling time 0.25 hour to 1 hour. When we use correct the concentration in the data analysis, the study failed to meet by equivalence criteria. So this case underscores the critical need for CRO to implement the robust quality management system that's able to detect and correct this kind of deficiency, discrepancy before regulatory submission. So case 2 involves a complex data anomalies. So in this fasting study, the subject were dosed in 2 groups, with 1 week apart at the same clinical site. When we conducted the data analysis, we suspect switching of the subject test and the reference sample between two groups, either during the dose, sampling collection, or sample analysis. When you look at the PK parameter in this table, the dosing group 1 from subject 1 to 30, the geometrical mean ratio, T and R, is 2.86 for C maxima and 2.77 for AUC. Look at the dose group 2 from 31 to 86. The geometrical mean ratio is 0.32 for the C maxima and AUC is 0.36. So this dramatic reverse have highly suspicious. When we issue the complete response deficiency letter to the applicants to ask them to explain this such significant difference, the applicants did not provide the explanation and voluntarily proposed to repeat this study at a different CRO. And later on, they withdrew this application. So this plot of C maximum ratio in the order of the subject ID, you can see a clearly pattern. You can see that in the group 1, the subject consistently have higher TR ratio. In the group 2, have a lower TR ratio. The situation become even concerning after when OSIS conducted inspection. Overlapping PK profile were observed across the subject from 2 dosing groups. The OSIS found a notebook at the analytical site and documenting subject pairs that had nearly identical concentration time profiles. So additional cases of unusual PK trend and overlapping PK profile were identified at this analytical site. So OSIS recommend our review division further look at the order application, use this site as an analytical site. So the FDA, the site, the classification was OII. So the review division conduct a comprehensive evaluation. So we evaluate all the ANDAs we study conduct at this site. We use the software tools to identify potential data anomalies. We verified that the similar observation was not presented for the same product in study conduct at other site. We carefully evaluated the bioanalytical report, especially repeat analysis, and check any discrepancy in reported concentration across different reports. Interestingly, we also observed some studies that failed to meet bioequivalence criteria with a larger number of the subjects at a different CRO site, but passed the B criteria with small sample size at this affected site. So the review division carefully evaluate explanation provided by the applicants and the CRO. Based on totality of evidence, we conclude that the reliability of the data generated at the CRO site A could not be ensured. FDA issued an untitled letter stating that the study conducted by CRO A was not acceptable. We publicly notify NDA and the ANDA sponsor that the study conducted at the CRO site A was unacceptable due to data integrity concern and must be repeated. The affected application include approved and tentative approved and pending applications. So applicants were required to repeat the BE study, use different CRO. For approved ANDA, we change the therapeutic equivalence rating to BX, where this means they are no longer considered by equivalence to the reference list drug. So case 3 involve in vitro permeation study, IVPT. So in the left panel, it describes how we conduct IVPT study. So T represents test treatment, R represents reference standard. So each block corresponds to one replicates the skin section. From this plot, you can see 5 replicates the skin section for the test treatment, and the 5 replicates skin section for the reference treatment are evaluated. So both test and reference will apply to the same donor, use the same method. So this setup allows us to compare the drug permeation, comparing between the test and the reference. So we found that the study 1 contained 12 donors. The study 2 had 20 donors. Each donor had 10 replicates of the test and 10 replicates of the reference product. So we identified nearly identical IVPT profiles between 8 out of 12 donors in study 1 and 8 out of 20 donors from study 2. So this overlapping was observed between 2 studies and from 2 different ANDAs conducted at the same analytical site. So basically, for IVPT study, due to nature of the high variability, overlapping profile are challenging to observe, even with the replicates within the same treatment at the same donor. So this kind of observation raises our highly suspicious. So this plot is showing the matching profile. The donor 15-1 from study 1 and the donor 8 from study 2, we examine the IVPT profile separately within the study 1 and within study 2. We did not find any overlapping profile from each individual study. When we combine those profiles, 2 studies, we observed the overlapping profile across certain donors between those 2 studies. So OC for cost inspection was requested, and the final OC's inspection classification was OII. So FDA requires the applicants to repeat the in vitro B study, use different CRO, and change the therapeutic equivalence rating to BX for the approved ANDAs. Summary. A multidisciplinary approach is crucial in identifying systemic data integrity concern. So FDA expects that all the data submitted in the ANDA be reliable and accurate. The poor data integrity imposes a significant cost and burden on the agency through increased review time and follow-up. The data integrity violation have serious consequences. So applicants and CRO should implement robust quality management system and maintain compliance to prevent data integrity issue and ensure the reliability of submitted data throughout the study life cycle. So at the end, I would acknowledge our colleagues from Office of Bioequivalence, OCs, and the Office of Biostatistics for their essential contribution. And thank you for your attention. I will take questions during the panel discussion. Thank you.
All right. And with that, I will now welcome our speakers up to the panel discussion. as they take their seats. Again, as the standard reminder for anybody in the room that has questions, please stand up to the microphones at this time. If there's not, we will then go to the Q&A online. Please, if you're online, feel free to upload your questions to the Q&A portal so that we can have a chance there. We will be going a little bit over time for the lunch break, so we'll have about 10 minutes for this Q&A panel for this. And with that, we'll go ahead and as everybody gets settled, if anybody wants to come up to the mic within the room, please feel free to do so at this time.
Hi, this is Aloka Srinivasan from Raha LLC. My question is for Dr. Chen. I saw in your micro-related document, which was incredibly nice, you repeatedly, for several situations, talked about not enough lots being used. And I was wondering how that becomes a common, as a former FDA, it was the Holy Trinity. I mean, as long as people did 3 lots, we used to be okay. But has anything changed in there? Or are you seeing that more and more people are being adventurous and not doing the right work?
Yeah, it's the latter. Yeah. So yeah, typically we always want to see things in triplicates, and a lot of the times when we have issues related to that, it's due to the triplicates.
All right, thank you for that response. We have another question within the room. If you'd like to go ahead and go, sir.
Good morning. This is Sharif Ahmed from Avon. A disclaimer, I'm not a microbiologist, but I do have a question for Dr. Chen. So when you have a product which cannot be sterilized, either terminal sterilization or aseptic filtration, and the most viable option is a gamma radiation, and even at that, a full dose is not viable, has the agency accepting a lower SAL level? And what kind of justification we need to provide with that?
That's a complicated question. So typically if we're looking at injectables or things that are being put into eyes and things like that, we want the product to be sterile. And so in order to justify those situations, that there's a bunch of different studies that would need to be conducted. Just because a product cannot be terminally sterilized, there are also bioburden-based sterilization techniques. So if you are calculating how much bioburden is within a drug product, that could be used to justify not being terminally sterilized. But again, those could be questions that are brought up during meetings in order to determine what type of studies would be needed.
Thank you. So these will be more appropriate for a pre-development meeting or pre-ANDA?
Yeah, because if there's a review issue based on how that product is getting validated in the submission, there probably would not be enough time to address those issues during the submission process.
Thank you.
Thank you for those responses. If there's no other questions within the room, we can turn it over for online questions.
Okay, this question is addressed to Dr. Bai, please. Following the RS agreement reached via CC, can the agency clarify if a pre-ANDA meeting is available? Although the product itself is non-complex, the current RLD shortage and the use of RS in a different dosage form have introduced significant complexities into the BE study design.
Okay, thank you for the question. Generally, the pre-ANDA meetings are reserved for complex drug product, but for some special cases, it will fall into May grant category. And as I see the question also talking about BE approach, so I'd like to have our OGD folks to address it.
So, given you already have established the conversation with the agency through CC, and still have complex, like the RD not available, and you want to use the different dosage form to as a bridging study to support your develop the generic drug. Yes, you can submit the PI in the meeting, but in the submission, you need to clearly describe what is the complex issue, what is the alternative, the B approach you want to use, and outline the detailed statistical approach, like you want to use the bridging, like what kind of information you can support your proposed method. Thank you.
Thank you very much. The next question is for Dr. Chen, please. If we implement a slight reduction in the concentration of preservative to improve ocular tolerability, will the agency accept USP 51 on the finished product at the end of its shelf life, or is a new stability study required?
So the reason that we're asking for the preservative content at or below the minimum specification is because we want to ensure that no additional microbial growth has occurred. So if there is, like during the whole time of the sample, so we would like to see new stability studies that are showing your preservative content at or below your new formulation.
Sorry. We still have some time. We're not almost done. Thank you.
And it does look like we have another question in the room if we'd like to go there.
Thank you.
I guess I have a follow-up question regarding preservatives. So when you have a product which has a combination of preservative and antioxidant, we believe there may be some difficulty in doing the preservative effectiveness test. Is there any guidance, or can you suggest how should we handle, or what, if you are aware of any acceptable way to study this whole system, or the primary preservative study should be good enough?
So if there is some sort of competition between the oxidant and the preservative, if you include that study within your application, that would be helpful, but the primary preservative testing should be sufficient.
Thank you.
Mm-hmm. All right, thank you. I think we have time for at least a few more questions. Brenda, do you have more?
Okay, this is one for Dr. Kokati. Regarding the coat weight sampling strategy on slide 10, can you elaborate on the recommended test frequency and sampling locations?
So coat weight is an important IPC test for transdermal systems. One suggestion we have in terms of the sampling, testing frequency and locations is the testing frequency should cover the entire CDL process. That's one thing. And although it could be application specific, our suggestion is that the minimal requirement is at least one role should be tested. And if you have multiple roles, then for each role, you should the applicant should consider taking samples from throughout the length of the role as well as the width of the role. So both dimensions need to be considered for sampling locations.
Thank you very much. Next question will be for Dr. Ren. Are you considering the ISPR? Oh, I'm sorry. Include subject period reanalysis.
So your question is ask whether we include the subject period.
Include subject period reanalysis.
Reanalysis. So in our evaluation, we definitely carefully evaluate the subject sample reanalysis, whether it follows their SOP or whether this kind of analysis criteria is appropriate identified in the SOP, whether this kind of analysis could impact the BE outcome. Yeah, we do.
Okay, I think that may be all the questions that we have time for so that we can stay on board with our lunch break for now. So we will go ahead and break at this time. If you have any remaining questions, you can always contact us via email and we'll get those addressed or at least distributed appropriately there. If you are on site, you can go to the kiosk there to pick up your lunch and pay for it if you have not already done so. If you're online, please rejoin us at 1:20. We'll resume again at that time. That should be an hour for everybody. And then we will begin with our afternoon sessions. See you all then. All right, it is 1:20 on the nose. So I think for those to that does send out those thank you emails for our participants of these events. And it does look like it's currently getting resolved. We expect for it to be resolved by tomorrow. And we will be sending a cumulative thank you to all attendees for both days there. So that link is coming your way, but also on SBI events, our main one-stop shop page, you can find the link to go ahead and do that certificate and survey right away if you prefer to do it then. We do solicit any feedback we can have on that survey. We go through all of it there and appreciate any feedback on how we can improve. So thank you in advance for going ahead and doing that. And with that being said, I will go ahead and jump into today's sessions. It is my pleasure to introduce our presenters for today. First, we will hear from Dr. Zhen Fu, Senior Pharmacologist in the Division of Pharmacology and Toxicology Review within the Office of Safety and Clinical Evaluation in OGD. Then we will hear from Dr. Min Guo, pharmacokineticist in the Division of Bioequivalence 1 within the Office of Bioequivalence in OGD. Then we will hear from Paramjit Kaur, lead pharmacokineticist in the Division of Bioequivalence 2 in the Office of Bioequivalence in OGD. Then we will hear from Dr. Wanling Liang, senior chemist in OPQA3 in the Office of Pharmaceutical Quality. Then we will hear from Dr. Dia Shaklea, senior research scientist in the Office of Pharmaceutical Quality Research in OPQ. Next, we will hear from Dr. Zhen Zhu, senior pharmaceutical quality assessor within the Division of Product Quality Assessment 9 in the Office of Product Quality Assessment 2 in OPQ, alongside Dr. Vincent Crowley, Senior Pharmacologist within, as well, DPTR in the Office of Safety and Clinical Evaluation in OGD. Then we will also hear from them in our Q&A panel afterwards. So now please join me in welcoming Dr. Zhen Fu.
Hi, good afternoon, everyone. I'm sound okay? Can you hear me? Okay. And also welcome back, everyone, online and in the room. Today, I'm going to talk about nitrosamine safety assessment in generic drug from a PharmTox perspective. This presentation reflects my view, not represent FDA's will or policies. So in this presentation, I'm going to talk about an overview of nitrosamine safety assessment, and then followed with common pitfalls identified during our PharmTox review of submitted nitrosamine safety assessment. At the end, I'll provide some summary and recommendations. The term of nitrosamine refers to both small molecule nitrosamines and complex nitrosamines. Complex nitrosamines, also called nitrosamine drug substance-related impurities, is a structure unique to each drug substance and may contain activating and deactivating features and often have a higher molecular weight than small molecule nitrosamines. And importantly, complex nitrosamines do not have compound-specific carcinogenicity data. In contrast, the small molecule nitrosamines are known potent rodent carcinogens and probable human carcinogens. Largely based on study of small molecule nitrosamines, it's believed that nitrosamine require metabolic activation, specifically through alpha hydroxylation, to form reactive intermediate that can interact with DNA, form DNA adduct, and cause gene mutation and ultimately tumor formation. Nitrosamines belong to cohort of concern in SHM7 and require tighter control than typical mutagenic impurities. SHM7 apply compound-specific acceptable intake to limit nitrosamine cancer risk. The acceptable intake is allowed exposure that causes 1 per 100,000 cancer risks over the lifetime. So here I want to emphasize FDA's recommendation for nitrosamine safety assessment are based on science. FDA formed partnership with Health and Environmental Science Institute has actively contributed to advancing nitrosamine science. Within FDA, there are multidisciplinary group of experts. They support nitrosamine-related decisions and guidance development. Among them include research scientists from FDA's National Center for Toxicological Research. They conduct nitrosamine investigations on their mutagenicity, as well as metabolism activation of complex nitrosamines. Their research and the findings support and guide regulatory decision and guidance development. Within CDER, there's a nitrosamine safety team. They include farm tox expert in both generic and new drug groups, and also include computational toxicology group. They not only coordinate farm tox review effort on nitrosamine safety assessment, they also engage CDER's quality, compliance, drug shortage staff, regulatory policy expert for enhanced coordination. In addition, FDA participates in international harmonization effort for nitrosamine impurities. For example, they participate in nitrosamine International Technology Working Group among different regulators, as well as participate ICH M7 nitrosamine addendum development to improved harmonization on nitrosamine assessment. When you ask an FDA farm tox reviewer how to conduct a nitrosamine safety assessment, the simple answer will be follow FDA guidance for nitrosamine impurities. FDA recommends 3 approaches to determine the AI limit for nitrosamines. The first is carcinogenic potency categorization approach, also called CPCA. Also can be determined based on AI limit based on compound-specific data. The third approach is read across from a surrogate with robust carcinogenicity data. FDA provides 2 important nitrosamine-related guidance. First is the quality guidance for control of nitrosamine impurities in human drugs. The second one is the safety guidance, is the recommended AI limit for NDSRIs. We also refer it as real guidance. In addition, FDA established nitrosamine webpage that include comprehensive information related to nitrosamines. So in this nitrosamine webpage include updated guidance, the list of recommended AI limit for hundreds of nitrosamines, and also interim limits for certain nitrosamine impurities. The webpage also provides recommendations on analytical methods and the safety testing method. It's also offered initial guidance on emerging issues and provide direct communication with applicant to help agency to collect data and better understanding those issues. The webpage also provides implementation timelines for FDA recommendations and the request. The real guidance is really important for farm tox reviewer to conduct nitrosamine safety assessment. It describes the use of a CPCA framework to establish an AI limit for NDSRIs without compound-specific data. Only the structure of nitrosamine are needed. And based on the structures that can affect alpha hydroxylation, which is critical for nitrosamine mutagenicity and carcinogenicity, they assigned numeric score for those features such as alpha hydrogen, activating and deactivating features, and which lead to the final potency score that corresponding 5 categories with acceptable AI limit from 26.5 nanogram per day for compound under category 1 up to 1,500 nanogram per day for those nitrosamines under potency category 4 and 5. The real guidance also recommend approaches to justify alternate AI limit. For AI limit up to 1,500 nanogram per day, FDA recommended negative enhanced amines assay, in vitro mammalian mutation assay, and in vitro metabolism study. For AI limit above 1,500 nanogram per day, additional negative in vivo mutagenicity assay using transgenic rodent model is recommended. Another approach is use read-across to a surrogate that's structure relevant with robust carcinogenicity data. Here, I want to point out for the nitrosamines, it tests negative in vivo mutagenicity. Currently, FDA does not automatically accept control limit at a level equal to ICH Q3A or Q3B qualification threshold. So with all those, the approach and recommendations on nitrosamine safety assessment, the pharmacology reviewer in OGD reviewed the submitted assessment and identified some common pitfalls. And this slide is listed top 3 pitfalls we identified. They include inadequate enhanced amines assay to assess mutagenicity, and you propose alternate AI limit solely based on the negative enhanced amines assay. And the third is improper use of a surrogate. I will discuss each of those pitfalls in a little bit more detail in the following slides. And following that, I will provide additional discussion of additional pitfalls identified in our reviews. When talking about inadequate enhanced amines assay, one common issue is did not test nitrosamine at a sufficiently high dose. The standard guideline recommend to test up to 5,000 microgram per plate if the test article did not cause severe toxicity. And we often see the submitted assays that did not test the high level, even though without cause significant toxicity. Some nitrosamine did cause severe toxicity and cannot be tested at sufficiently high dose. That make those enhanced amines assay inadequate to really fully characterize their mutagenicity potential. Another issue is select the solvent. And some solvent, especially at a higher concentration, can interfere with metabolic activation, which is critical for nitrosamine to cause mutation, and therefore, if the solvent interfere with metabolic activation, then make these assay inadequate. Also, we see assays did not include proper nitrosamine positive controls, and also did not include sufficient metabolic activation conditions. We also see a lot of justifications for alternate AI limit only based on negative enhanced amines assay. As I mentioned previously, FDA recommend negative enhanced amines assay and also in vitro mammalian mutation assay, as well as in vitro metabolism study focused on alpha hydroxylation in order to support the AI limit up to 1,500 nanogram per day. Here, I want to acknowledge that FDA's recommendation differ from the recommendation from other regulators. And we proposed this recommendation at a time, this started 2023. At a time, the information is limited. And the enhanced MSA is still under optimization. And there's a lot of uncertainties with the data. Therefore, we recommend additional assays to reduce the uncertainties. And since then, we receive a lot of data and also gain a lot of knowledge, but still there's a lot of a data gap. And also we see discordant data. So currently, we are actively assessing the data we have and also actively working with other regulators for harmonization. So talking about using read-across with surrogate, it is important to select the surrogate, the structure relevant. When you consider the relevance of structure, it is important to consider that the nitrosyl alert in the surrogate should in the same chemical environment as the subject nitrosamines. In addition, they also should have a similar degree of substitution and steritic box features. In addition, electronic influences, metabolic activation potential, and stability and reactivity of the resulting metabolite should also be considered in addition to the overall molecular weight. And also, very importantly, the surrogate should have robust carcinogenicity data. So in addition to these top three common pitfalls, we also increasingly seeing the applicant applying approaches that are currently still under investigation. For example, they apply molecular weight or less than lifetime approach to adjust the AI limit, or set the AI limit based on in vivo mutagenicity data. Since those approaches is still under investigation, including the assessment by ICH-M7, so currently FDA does not accept those approaches. And also I want to emphasize, if the nitrosamine test positive for mutagenicity is required, we recommend to use the CPCA-based AI limit to be protective. Also, it's not acceptable to submit the general toxicity data to justify the AI limit. The general toxicity data is not designed to adequately characterize mutagenicity and carcinogenicity of the nitrosamines. Currently, the mutagenicity evaluation is the most critical evaluation for nitrosamine safety assessment. If in vivo study is necessary, we recommend to use transgenic rodent gene mutation assay. As I mentioned previously, FDA currently does not automatically accept control of non-mutagenic nitrosamine at the level of ICH Q3A, Q3B, QT level. FDA considered the outcome from compound-specific assessment, such as determination of root of cause, and also applied the mitigation approach to reduce the level in the products. And therefore, FDA, based on those assessment and the batch testing data, to set the control limit for those kind of nitrosamines. It's also not acceptable to set other regulators' AI recommendation without providing the supporting data or the right of the reference for those data. It's also generally not acceptable to control nitrosamine at the level observed in the RLD. And also, it's not appropriate to consider interim AI limit as the final product control limit. So here is the summary. So to provide a successful nitrosamine safety assessment to FDA, we recommend to follow FDA nitrosamine guidance and use the CPCA-based AI limit for the nitrosamines without the compound-specific data. Also, we recommend to monitor FDA nitrosamine webpage for the latest update. And also, I hope my presentation demonstrates that farm talks in OGD conduct nitrosamine assessments based on the science through collaboration and harmonization. And if you have questions with those nitrosamines issues specific for your product, you can submit control correspondence and we can provide the product-specific response. So this is my last slide and provide some useful reference can help with nitrosamine safety assessment. And before I complete my presentation, I'd like to have a acknowledgement and highlight for the Farm Talks group in OGD for their hard work and commitment to protect the safety of generic drug. And without their work actually make my presentation possible. So next, let me invite Dr. Kao for her presentation. Thank you.
Thank you. Good afternoon, everyone. My name is Paramjit Kaur, and I'm joined today with Dr. Men is joining me today, both from the Office of Bioequivalence within the Office of Generic Drugs. Today, we will be talking about bioequivalence recommendations for bridging pre- and post-mitigation formulations of nitrosamine impactor drug products illustrated through 4 case studies. Before we begin, I need to provide the disclaimer. This is our standard disclaimer. Let me provide the overview of my presentation. Today, we will start with the background on nitrosamine impurities and the mitigation strategies available to the drug product manufacturers. Then we will dive into bioequivalence approaches for drug products reformulated to control nitrosamine impurities. Next, we will talk about 4 case studies, which are hypothetical case studies designed to illustrate these principles in practice. Then we will conclude with key takeaways. I will be covering the background and the first 2 case studies. Then Dr. Min will take over to discuss next 2 case studies, followed by summary and acknowledgements. Starting with the background, since 2018, unacceptable levels of nitrosamines have been detected in commonly prescribed medications, which have become a significant concern both for the agency and the drug product manufacturers. Nitrosamines are potent genotoxic agents in many animal species, and some are classified as probable or possible human carcinogens. They are considered as cohort of concern compounds in ICH M7 guidance, as earlier speaker pointed out. This issue has a widespread impact affecting both new drug applications and abbreviated new drug applications. The majority of impacted applications are generic drug application because they are anyway more, and it impacts all kinds of drug products and the dosage forms. The nitrosamine impurities can be categorized into 2 categories: small molecule nitrosamines that can be present in the API or in the drug product, including NDMA, NDA, and related compounds. They do not share any structural similarity to the active pharmaceutical ingredient and are found in many different drug products. Second category is nitrosamine drug substance related impurities. They are structurally related to the API or API fragments. For more details, you can refer to FDA guidance control of nitrosamine impurities in human drugs. In my subsequent slides, I will be referring to this as nitrosamine guidance. In the nitrosamine guidance, various regulatory strategies are recommended to mitigate the risk of nitrosamine impurities. For today's presentation, we have categorized it into 3 categories. First is API source process modification. The applicants can modify their route of synthesis, select new API suppliers, or optimize their manufacturing process, or control nitrile levels in raw materials. Second is drug product reformulation strategy. This is the most common strategy taken by the generic drug applicants. It involves addition of an antioxidant that has been studied in FDA-supported research, which include ascorbic acid, alpha-tocopherol, propyl gallate, and cysteine hydrochloride. It can also involve incorporating pH modifier or replacing problematic excipients with safer alternatives or different grades having low levels of nitrite. And third is you can control is manufacturing process control, where you can modify process equipment or implement packaging changes. Well, now the most important question is what kind of bioequivalence data is needed to support the reformulation of the drug products to control nitrosamine? Well, it depends on the type of BCS class, the dosage form, and the reformulation strategy taken by the drug manufacturers. Nitrosamine guidance outline the alternative B approaches, which are summarized in this table for BCS class 1, 2, and 3. Drug products reformulated with FDA-researched antioxidants at an amount no more than 10 milligram per dose or maximum daily exposure, whichever is less, or using a pH modifier. We recommend comparative dissolution data on pre and post change products. The dissolution data should be provided in quality control medium as well as in 3 pH 1.2, 4.5, and 6.8. In other cases, BCS class 1 and 2 and 3 with FDA-researched antioxidants or other antioxidants. In addition to dissolution data, we may request additional studies. For BCS class 4, with any reformulation and all modified release drug products, IVIVC, PBPK modeling, or in vivo BE studies are recommended. Alternative BE approaches can be considered on a case-by-case basis. We encourage the applicants to engage with the agency earlier to get inputs on their alternative proposed BE approach. It is important to note that this alternative BE approach, it does not apply to narrow therapeutic index drugs. It also does not apply to sublingual, buccal, orally disintegrating, or chewable tablets. Now let's turn to hypothetical case studies. First two case studies are related to pre-approval changes, which I'm going to present, and then Dr. Min is going to present next two case studies, which are related to post-approval changes. So for first case study, the drug product is available in multiple strengths. BE studies were conducted on the highest strength, and the waiver was requested for the lower strengths. The BE studies met the BE criteria. Dissolution data was fine. All the strengths were proportionally formulated. However, there were nitrosamine impurities issues, but additionally, there were CGM compliance issues at the API facility. If there are CGMP compliance issues, then the quality of biobatch cannot be assured. If the quality of biobatch cannot be assured, then the BE studies and the waiver request cannot be deemed adequate. To address this issue, the applicant changed to a new API source. No other formulation changes or process changes were made. They submitted the dissolution data comparing the pre and post change drug products. Based on the agency's evaluation, original API DMF was closed without resolution of CGMP deficiencies. This is the key point. If the original API is not adequate, it has quality issues, then the API source change to address nitrosamine impurities cannot be supported by dissolution testing only. Therefore, all the data generated using the batches manufactured with original API were considered unreliable, including in vivo BE studies and dissolution. The applicant was recommended to conduct new BE studies and dissolution data using the batches manufactured with a new API source. In case study 2, the drug product is an IR tablet. It is available in multiple strengths. Similar to the previous case study, the applicant conducted BE studies on the highest strength and requested waiver for the lower strengths. BE studies were deemed adequate. However, the overall application was deemed inadequate due to nitrosamine impurities. In this case, the applicant's mitigation strategy was to reformulate. They did the reformulation by changing the multiple excipients and the manufacturing process change, which would fall under level 3 category, which will require in vivo BE studies. In this case, they submitted the BCS class waiver 1 request. They conducted their own solubility and dissolution studies per ICH M9 guidance, and in support of high permeability, they submitted the data from the approved reference listed drug labeling. Based on the agency's evaluation, the submitted data was found adequate to support BCS class waiver 1 request, and the waiver was granted. Now I would like to hand over to Dr. Min. Thank you.
Hi, thank you, Parajit. And my name is Min Guo. I will be transitioning to two post-approval changes in the applications. So the hypothetical case study 3, this is a delayed release capsule, a DR capsule product with multiple strains, and the BE studies has been done on the highest strains. So apparently the nitrosamine issue has been identified, and the applicant proposed mitigation strategy, which is relatively straightforward by adding a antioxidant, a pH modifier, and other excipients has been reduced just to compensate and to keep the capsule weight the same. No other significant changes. So the applicant proposed the data to support such change, bridging the pre and post change products to conduct a permeability study and to prove the antioxidant does not affect the bioavailability absorption, and also in vitro dissolution study between the pre and post change products. So because this is a DR product, belong to the modified release category, based on the SUPAC MR guidance, the proposed formulation change is classified at level 3 change. And also the nitrosamine guidance generally does not recommend the alternative BE approach for the MR products. But on the bright side, so the addition of the antioxidant is within 10 milligram per dose. It's also a FDA research, one of the 4 FDA researched antioxidants, and also the pH adjuster modifier or antioxidants only add to the drug core, the IR layer, as the DR product is generally relatively simple in terms of other modified release product. So there's no change to the delayed release coating. So then we can think if there's anything can be think of as an alternative BE approach, because the formulation change is not expected to compromise the delayed release coating. And also the applicant provided data to prove the drug substance is highly soluble. So we consider it as a putative BCS class 3 drug substance. So by the nitrosamine guidance, it's a BCS3, and also the characteristics resemble the IR product. Then we're thinking we can the whole formulation change presents risk similar to an IR product. So overall, our recommendation is neither the in vivo BE study nor the permeability study is necessary. And based on the nitrosamine guidance, we recommend the applicant submitting comparative dissolution data for both the pre and post change products under the QC and the multimedia conditions. And another thought is not just applicable to this drug product, but also overall to generally all the post-change product. If you have any new excipient or increased excipient, we're gonna evaluate the safety of the excipient. Just because the guidance, nitrosamine guidance recommend those antioxidants, or you added some pH modifier, doesn't mean there's still a risk is over the IG limit of the excipient. So you should also evaluate and submit the corresponding data. And in this case, the the patient population, including the pediatric population. So you need to evaluate the intended patient population, including the pediatric. So the next hypothetical case 4, which is an IR tablet with multiple strains again, and the B study, of course, down in the at the highest strains, waiver on the lower strains, with the nitrosamine impurities identified. But this one, the mitigation strategy is somehow unique because the nitrosamine issue has only been identified in the let's say the lower strains, not the highest strains, and not all the strains of the drug product. So the mitigation strategy is also unique accordingly. So what the applicant plan to do is to reduce the excipients, all the excipients in the lower strains significantly, let's say 40–50%, and compared to their original formulation. And then because these excipients have been reduced to the similar extent, so overall, if you look at the weight by weight percentage, it generally been kept the same. That's why the applicant argue that it would be based on the SUPAC IR seem to be a level 1 change, and only the QC dissolution would suffice the change. However, is that really so? So the main crux is to identify the correct level change and the risk, and then accordingly we can decide what data to support such change. So the key issue is there is a significant weight reduction in the capsule in the tablet weight, and the SUPAC IR does not directly address such total dosage form weight change. But if you recall, as we know, it's mostly just talking about the weight by weight percentage, right? And that's the basis why the applicant says level 1. But in the SUPAC IR Q&A, there's a question 9 saying if the new target weight is within the range of the original approved application, then it's a level 1. Otherwise, it's a level 2 or level 3. So from there, we have the concern it's not just a level 1. It's reasonable for us to ask for more data to prove that. And also, if you look at the formulation, the composition, because the significant reduction of each excipient, so the lower strains is no longer proportionally similar to the highest strains. So you cannot just go by a simple QC biowaiver pathway. So we ask for other supporting data like multimedia dissolution. We're thinking about if we can go by a BCS-based waiver approach. And the applicant approved, again, this drug substance is highly soluble, which is the purity BCS class 3. And then based on the M9 guidance, we need to look at the Q1, Q2 criteria and also the multimedia dissolution. But unfortunately, this drug product is Q1 the same, but not Q1 the same, Q2 similar to the reference product. Also, it's not a rapidly dissolution across all the multimedia conditions. Therefore, it doesn't really meet the BCS3 criteria, biowaiver criteria. With all of that, we think the impact of the proposed formulation change, particularly the reduction in the weight, significant reduction in the weight, cannot be fully understood without the in vivo study. So therefore, we recommend applicant conducting an in vivo BE study on one of the higher strains among those low strains, or alternative approach if they can propose alternatively. So in summary, so nitrosamine issues, as we can see, has impact on both the approved and the pending and applications. And the applicants has been adopting different mitigation strategies, one or more, involving the API manufacturing change and even reformulation. And the nitrosamine guidance offer us the alternative BE approach to lessen the industry and the regulatory burden. However, the still is just a general guidance, like old cliche, is still case by case because every drug product is unique. And in principle, the guidance applicable to both the pre and post-approval changes. And if the changes are beyond the scope of the guidance alone may not constitute the adequate resolution. And finally, we encourage the applicants have early engaging with the FDA through the meeting requests and the control correspondence. And finally, I want to thank all the OB management and also, most importantly, the review team of those applications. Thank you. And I'll hand it over to Dr. Liang.
Oh, hi, good afternoon. Okay, my presentation is mainly talking about the CMC-related nitrosamine update and some issues I observed during the under review. Same declaration letters, everybody has it, so I'm not going to repeat it. So my presentation will include a full part, basically how to use the recommended AI limits tables and the timelines for submit the implementation changes or the progress update, and some emerging issues recently published on the nitrosamine webpage. And the last one will be some common issues I have been observed. First, I would like to say the importance of the nitrosamine web pages. The nitrosamine web page is the central place for FDA to provide to update the current FDA thinking about the nitrosamine controls. So it updated very frequently whenever the new information become available, and it can be implemented immediately. So the FDA recommendation is that you check it regularly and to assess any kind of impact to your products and your applications. Now let's talk about the recommended AI limits. On the web page, there's 3 recommended AI limit tables. Table 1 provides theoretical AI limits. It was calculated using the CPC approach. So these are for those nitrosamines they don't have available tox data. So table 2 provides the compound-specific AI limits. Those are calculated based on the available tox data or suitable surrogates. Table 3 provides an interim AI limit. And this AI limit is only applicable to those marketed medical necessity products. And this was provided when the immediately comply of the recommended AI limits in table 1 or table 2 could cause drug shortage. So this serves as the same function as a temporary regulatory discretion to prevent drug shortages. And the thing is, although there are so many nitrosamines was listed on the web page, but the nitrosamine listed on the table does not mean it will definitely exist in your drug products. So you need to do the risk assessments. And also the AI limit is just a safety threshold. It's not the actually observed level. Another thing I want to remind is for all the nitrosamines listed on table 1 and table 2, a nitrosamine risk assessment report is required for the new ANDA application. And for the approved ANDA applications, implementation changes or a progress update is required. And here are some key points for the 3 interim limit AI tables. The table 1, as I said, this is the theoretical values. It was calculated based on the molecular structure because we don't have reliable tox data. So it's not all inclusive. So new nitrosamines has been observed frequently and reported. So the table has been updated and it will continue to be updated. When enough tox data becomes available, the nitrosamine in table 1 can be moved to table 2. So when the nitrosamine were moved from table 1 to table 2, the recommended AI limit in table 2 should be applied. So don't use the previous one in table 1. The table 3 should be paying more attention. Table 3, as I said, is only for the interim limit. The only purpose is a temporary release to prevent drug shortage. So this was published when multiple manufacturers was involved, and majority of drug products cannot meet the currently recommended AI limit. So it has durations, the application durations. By the end of duration, it can be expired or it can be extended. However, this interim limit, as it's only temporary, it cannot be used, or it's not a permanent solution, and it cannot be used as a permanent solution for the mitigations. And it cannot be approved as specification limit, and also you cannot use it to justify a specification limit. And these interim limits do not replace the long-term control and mitigation strategies. So basically, it's not eliminate your obligation to implement a sustainable control strategies to meet the AI limit recommended in Table 1 and Table 2. Now, for those drug products that have identified nitrosamine risk, initially, FDA expected the confirmatory testing and submissions for the strategy mitigation changes to be submitted by August 1st of last year. Later on, FDA realized that some of the mitigation changes may take longer time. So if you cannot finish these confirmatory testing and mitigations by that time, a progress update should be submitted on the same due date, with the same due dates, August 1st of last year. So here is the requirement for what should be included in the progress update. You can find detail from the web page. So if you cannot provide all those required information, scientific justification is required for those missing part. So if you just submit a partial update without explanation, it will be not considered as sufficient. And here is how to submit the progress update. You can also find it from the web page. What I want to point out is for those products which are currently released using the interim limit to prevent drug shortage, and a progress update is required for these drug products or applications. And in addition to all those required information, you also need to submit the batch data, the batch release and batch stability data for those batch you currently released against the interim limit. So FDA can base on the data to update the duration period. As I said, the new nitrosamine issues has keep arising. So FDA using the web page to alert industry to the newly identified nitrosamine issues and to provide possible root cause and information mechanisms, and also provide mitigation recommendations. The recent issues published like leachable NDBA or small molecule nitrosamines from the infusion bags and the newly identified NDSRI in some returnable drug products. So the expectation is you monitor the white page regularly, and for the newly issues to do the assessment to your products and implement the recommended mitigation strategies promptly. Next, I'm going to present a few issues I've been observed during the ANDA review. First one is the inadequate control strategies. We often see some control strategies solely based on the release testing result without addressing the formation mechanism or its lack of justification for the effectiveness and sustainability across the product lifetime. Please be noted, the FDA expectation is to prevent nitrosamine formation, not just detected in your product. Another inadequate justification is the inadequate justification for an AI limit exceeding the recommended AI limits. This, I think, Dr. Gao just talked about. FDA do not accept the following recommended justifications. First one is a less than lifetime approach. Second one is solely rely on other agencies' recommended AI limit, like EMA, Health Canada. That's we always see. Another one is using the interim limit to justify a permanent drug product spec limit. The issue is the incorrect filing categories. And here I just gave a summary as a quick reference for the supplement filing categories. When you're using the interim limit in Table 3 to release your drug products to prevent drug shortage, no supplement is needed. However, a progress report should be provided, and also to use the interim limit does not prevent your obligation to control your drug products, like to test at release and stability. So the progress update should be submitted with the batch data. If you have questions, you can contact the drug shortage staff. When your risk assessment concluded as no risk, then you don't need to send any submission. However, for the NDSRIs listed on the web page, a progress update is required. So you need to submit a justification and supporting document and data to support your no risk conclusion. You cannot just say, I have no risk. And another is a formulation and a manufacturer process change. That will be the PAS. And for changing of a specification based on your confirmatory data, it can be an annual report when it's less than 10% or between 10 to AI limit. It can be CBE 30 or over AI, it's be the PAS. And for the NDSRIs, which are not listed on the web page, 26.5 will be the most conservative AI limit. So if your control is less or equal to the 26.5, it's be the CBE. But if you're over that one, even though you're using the CAPA, the CPCA approach, it will still be the PAS. And here's the takeaway. So the white page is a dynamic regulatory communication platform for FDA to communicate all the nitrosamine policies. It's updated frequently and it can be implemented immediately. So you always apply the correct and most recent AI limit. Interim AI limit are not permanent strategies. It do not replace long-term control. So it's just temporary to prevent drug shortage. Submit required change and detailed progress update on time, and implement effective, preventive, and sustainable mitigation strategies. Pay attention to the emerging issues and also submit the changes and the correct filing categories. And here is the results you can contact. And next, and thanks to my colleague. And next, welcome to Dr. Shakir.
Yeah, thank you, Lynn. So hello, everyone. Good afternoon. So today, after my colleagues talk, I'll be discussing our research efforts at the FDA focused on advanced nitrosamine risk mitigation in pharmaceutical manufacturing. And this work reflects the research conducted within the Office of Pharmaceutical Quality Research at CDER, which is important to note. And this nitrosamine contamination has posed significant scientific and regulatory challenges in recent years. And our goals here has been better to understand the mechanism behind their formation and evaluate the practical mitigation strategies using real drug product case studies. So this is the message that we need to show, that the pharmaceutical quality is assuring every dose is safe and effective and free of contamination as nitrosamine and defects. So this presentation is not intended to convey any formal FDA policy, but just relying on our research work that has been done in our laboratories. So this is the outline. First, I will be identifying what are the risk factors and some of the control strategies. Then showing the mitigation strategies that we have applied in the lab, and then also presenting two case studies, that pimetanide and metformin drug product that we completed and presented and published in peer-reviewed journals for the industry to use. So important to understand first what are the risk factors that affecting your formulations.
So nitrosamine formulation requires three main elements, and I'm sure all of you know that. That's the amine, the nitrosating agent, such as nitrite, and the favorable reaction condition, which is related in the other presentation, like acidic pH. Also, manufacturing steps, blending, drying, granulations, along with the storage and packaging can further influence the formations of nitrosamine. So our primary objectives of this studies was to evaluate the inhibitors, particularly the antioxidant, and also the pH modifiers in mitigating the NDSRI formation using two model drugs, pimetanide and metformin drug products. So what are the possible control strategies that can be implemented? First is the API, which is the main source of the nitrosamine formation. So to highlight that mitigation must be a multi-layered, which that's we concluded in from our research. It begins with the API manufacturing controls and the robust supplier quantification or qualification program to monitor nitrite impurities in excipients. And additional strategies include antioxidant, pH modifier, and tighter manufacturing controls, which I will be explaining in the next slide. And also the inner packaging materials, which my colleague alluded up to the, for those nitrosamine that can leak from, or the precursor that can leak from the packaging. And also important to say that effective mitigation requires addressing risk across the entire product life cycles. So there are several mitigation strategies that can be implemented. What we did in our lab, that's what I'm going to present here. Acidic conditions favor nitrosation reaction, and that's from our research and from the literature that it says that the acidic pH or nearly acidic pH can add those to the nitrosation reaction. So maintaining a neutral pH can be protective. Also from our experience that heat and moisture significantly increase nitrosamine formation, and particularly during wet granulation and drying. We also evaluated the antioxidant, as I mentioned, and we selected three main excipients as antioxidant, ascorbic acid, caffeic acid, and ferulic acid, based on literature and recommendation from our assessors and reviewers. And that's what we demonstrated, that varying degrees of inhibition effectiveness from different concentration of those antioxidants. I'll just walk you through some of the manufacturing process that we attempted in modifying those drug products, and including some of those strategies, mitigation strategies. So the process began with the sieving and geometry dilution to ensure uniform blending of API and antioxidant. It took us some time to understand that when you mix your API with antioxidant initially, that helps in preventing or mitigating some of those nitrosamine formation. Also, the nitrite was intentionally spiked during granulation to model contamination risk. This step allowed us to simulate realistic manufacturing scenarios where nitrite may be introduced through excipients. To continue granulation, granules underwent a semi-drying, which is one of the important factors, as I mentioned previously, the heat. And we use here, they commonly use in industry, the fluidized bed dryer, moisture testing, and final blending before compression. Since drying is a critical step for nitrosamine formation, controlled temperature and moisture was essential to understanding formation dynamics. And here I'll just highlight some of the results that we got from the first model drug, which is pymetanide nitro. So pymetanide was detected in this drug product. And as I see here, there are so many profiles, just highlighting one of the important, that different concentration of antioxidant may change the mitigations over the, we did the study here for 6 months, and we found out that ascorbic acid, the vitamin C, was shown to have at 1% showed to have better mitigations. And also we demonstrated also in this published paper that sodium carbonate or bicarbonate can also influence the mitigations of the nitrosamine and pymetanide drug products. And the second example here is the NDMA mitigations in metformin tablets. And we wanted to understand also the effectiveness of those antioxidants. It can be same as the previous drug, or it is different. And we saw that the ferulic acid at 0.1%, it shows different compared to the caffeic acid and ascorbic acid, but at higher concentration, all of those antioxidants showed mitigations. And finally, this is my summary, which I highlighting the importance of the findings. So antioxidant can effectively mitigate nitrosamine formation, but they must be fit for purpose. Their effectiveness depends on drug substance chemistry, which is important, formations, formulation design, and manufacturing conditions. Increasing antioxidant concentration generally improve mitigations in metformin and pymetanide drug product. Additionally, neutral or slightly basic pH condition were protective too, while we understand that acidic environment facilitate the formation of nitrosamines. And with that, I just like to acknowledge my OPQR colleagues, OPQR management, and the OPQ nitrosamine working group, and finally the organizer for the invitation. With that, I would like to invite my next speaker, Jinju, to present his talk. Thank you.
Good afternoon. My name is Jing Su. I am a senior chemist reviewer from the Office of Pharmaceutical Quality. So along with my colleague, Dr. Vincent Crowley today, we'll be talking about extractable leachable. I will be focusing on qualitative perspective, and Dr. Crowley will be talking about toxicology assessment. So this is standard disclaimer. You have seen this many times over. So this is the outline of my talk. FDA colleagues, including myself in the past, have presented to industry how to perform extractable leachable study. But today, I'm not going to talk about how to perform an extractable leachable study. Instead, we're going to present to you some strategy how to not to perform a leachable study. We recognize that extractable and leachable study is very time-consuming and costly, and hopefully after this talk, you might be able to save some resources and time. So, introduction, we all know what extractable leachables are, and we know that extractables are actually compounds that are extracted into a solvent in a laboratory conditions. And leachables are actually present in your drug product. So in that sense, leachables are actually more clinical relevant compared to extractables. And there are quite a number of valuable references available to you for your study. Obviously, USP 1663 and USP 1664 are very valuable references. They have inputs from FDA, so they are good references. And there are also technical references from PQRI publications. And FDA published a guidance document for CCS in 1999. And just for information, that guidance is being updated and could be published any day. Sorry. So one document I did not include in this slide is ICH-Q3E because ICH-Q3E is not finalized yet for adoption, so I did not include in this slide. But that document now is available for public consultation, for public comment, and I believe that is the public consultation period closed at the end of May next month. So that's a very, very important upcoming document guidance for you. So this is a good time for you to provide comment, and we appreciate your comments as well. So the first step before you perform a extrapolable study, obviously you need to perform a risk assessment for and each risk assessment is product specific because you will know each product has different formulation and you could use different CCS as well. So the first step, you need to contact the vendor of the CCS to understand what materials are used in that CCS and also what kind of processing aids that are used in the manufacture of the CCS. Prior knowledge, very, very good information. Many of you may manufacture more than one product. Some of you may manufacture maybe 10, 20, 30 injectable product, for example. And in those cases, you may want to look into your internal database to see if the same CCS has been used in a approved product or not. And in those cases, you might be able to use a approved product to support the new application currently under review. Route administration is also important. We know that some routes are more critical, more risky than others. And for example, an injectable product will be considered more riskier compared to oral route. Duration, we all know that the longer exposed to the drug, obviously the higher the risk. Maximum daily dose is not an important factor. The higher dose, the exposure to leachable will be higher, chance will be higher. And also formulation composition. Some product contains organic solvent to help solubilize the API, and for those formulations, obviously the chance of leaching out leachables is higher. There are some leachables that can interact with the drug substance, also depends on the structure of the drug substance. In those cases, there are a chance that there will be a product coming out from the reaction between the leachable and your drug substance. And that's another factor you should consider. And also the storage condition shelf life. Some product that have a longer shelf life. For example, a 48 months compared to 24 months. The one that is 48 months will have a higher chance and higher potential higher concentration of leachable. So how do you leverage prior knowledge in your initial assessment? In these cases, you might be able to not doing an actual extrapolable study using your prior knowledge. For example, using the product you already have that already have been approved by the FDA. For example, if the product obviously having the same route of administration to injectable products that use the same CCS, and the new product will have the same or similar product composition. For example, the new product under review contains less organic solvents compared to the one that have been approved. And also, in this case, the one that has less organic solvent is expected to leach out less leachables. And maximum daily exposure is very important. You can use the one that has a higher daily exposure to support the one that has lower maximum daily exposure, but not the other way around. So for oral drug, we know that oral drug is relatively lower risk compared to other parenteral product, for example, injectable product like that. And we encourage you to look into the potential that use this approach so that you do not need to conduct a extractable leachable study for oral drug. And this approach is to provide documentation and test results to show that the CCS actually comply with integrated food additive regulations 21 CFR 174 to 186. These regulations have very specific information that, for example, the construction materials of the plastic, rubber, and the use conditions, and also the specifications and test method. So if you are using this approach, make sure that you need to provide proof that the material construction of your CCS are consistent as those listed in the regulations. It cannot be different, have to be the same. Okay. The use conditions are the same or less stringent compared to those listed in the regulation. And also the leaching propensity of the oral formulation should be similar or less than the food listed in the regulation. And this could be tricky. You're talking about one drug versus one food, right? So, but there are, in some cases, you can compare them. For example, some food product that contains a lot of oil or organic component. For example, in the regulation, there are some plastic that are suitable for packaging, beverage containing alcohol up to a pretty high level. Then those kind of material, if your product contains less organic component compared to this kind of plastic material listed in the regulation, then you have a point to argue that your product is less has a lower extraction propensity compared to the food. And also the regulations contain specific test method as well as specification. You need to provide the specification as well as the test result to show that the CCS you're using comply with the regulation. So the second, the last point I want to talk about is the post-approval changes. As we speak now, we do not require a extractable leachable correlation to be submitted in any ANDA, but we encourage you to do so. And the reason for that is that the correlation comes with many benefits. For example, if you decide that you need to control leachable, then if we have the correlation, you can control the extractable at the QC stage of the CCS. So you do not have to control leachable in the drug product during its shelf-life. And that's one good benefit. The second benefit is post-approval changes. If you have the correlation, then it is a lot easier for you to, for example, make later changes to the composition of the CCS. Then you can compare just the extractable data to say that the changes will not increase the change of leachable in the drug product. So we encourage you to submit the extractable leachable correlation if possible. For group supplements, as I said earlier, that many of you may make more than one product, could be 10, 20, 30 injectable product. And all these products may use the same CCS. For example, rubber stopper may different sizes, but the composition of the rubber stopper could be the same. In those cases, you might be able to use the highest risk product to support the group supplements, and that will save you a lot of work, and that will save us work as well. Okay, so if you decide to do so, make sure that you perform a assessment on each and every single CCS, each and every single product, because each product is different. And then use the one that has the highest risk to support the entire group supplements, and take consideration that maximum daily dose in product leaching propensity. Okay. With that, I thank these supervisors and colleagues for support. With that, I would like to invite Dr. Crowley to come to the podium. Thank you.
Hi, I'm Vince Crowley. I'm a senior pharmacologist in the Division of Pharm Tox Review within OGD. Here's the disclaimer for the fourth time this afternoon. And so today I'm going to talk about extractable leachable safety assessment in generic drugs, outlining some of the key factors that should be considered when compiling your safety justifications, and then some of the common pitfalls that we see that lead to deficiencies being issued. And so to begin, extractable leachable studies identify compounds that may migrate from either manufacturing equipment such as tubing or filters, or the container closed system as vials or stoppers. And these extractables are identified under exaggerated laboratory conditions, either high or low, very high or low pH, increased organic solvent content, or a number of other variables. And then leachables are migrants that do show up in the drug product under typical storage conditions for the drug product. And like Jen mentioned before, the quality review team confirms or reviews the assessments, and then they confirm the adequacy of the study design and the analytical methods, including the sensitivity of the methods. And then OGD Pharm Tox, which is the division that I'm in, we evaluate the extractable leachables that are observed above the safety thresholds. In our assessments, we consider the mutagenicity, general toxicity, and local toxicity of the observed compounds based on the context of use specific to that drug product. And so from the outset, a lot of extractable leachable study assessment is based on thresholds. And so you want to make sure that the thresholds are identified properly at the beginning. And so one of the main thresholds is the safety concern threshold, which is listed here. For most routes, that's 1.5 micrograms per day for chronic use drug products, and for shorter use products, it's 5 micrograms per day. But for some more sensitive routes, such as ophthalmic, which is listed below, or intrathecal, where there's exposure to sensitive tissues, lower safety concern thresholds may be warranted. And in either case, if there's any question on what the appropriate threshold is, or how to what the maximum daily dose is for a given drug product, then a good opportunity. This is a good place where you could submit control correspondence to OGD to receive guidance related to a specific drug product. And so after setting up the thresholds for your study, you can go ahead and perform the study, and the extractables or leachables that are identified over the safety concern threshold over here on the top left. Those two peaks should be identified by both their CAS number and or their chemical structure and name as shown in the bottom left. And so once you've identified all the chemical, all the extractable leachables over the threshold, you want to identify what types of safety assessments are required. And so the thresholds that we use in our current practice are for general systemic toxicity is 5 micrograms per day. Mutagenicity thresholds are based off of the duration of use for the drug product, which is described in ICHM-7. And then local toxicity is generally assessed at 5 micrograms per day, along with the general toxicity, but may be lower for more sensitive routes of administration, such as intrathecal, as I mentioned earlier. And as Dr. Zhu mentioned previously, we know that Q3E draft guidance has been published, and there are safety thresholds listed in there. However, these are not finalized, and so they're not used in our current review practice. And now we move into compiling the safety assessment of the leachables identified above the safety thresholds. And so the easiest case are ones where there are compound specific data available for the leachable observed above the safety threshold. However, many of these extractables and leachables, there are no compound specific data. And so typically a read-across approach to identify surrogates with available data is used in order to inform the safety of the observed leachables. And so when you perform this type of assessment, you need to strike a balance between structural similarity for a surrogate versus what data is available to support the safety of the observed leachable level. And so some considerations in your surrogate selection and potential groups. Number one being the overall structural similarity. to the observed leachable. The functional groups within that leachable should be, or the surrogate, should be in a similar chemical environment, both for sterics and electronics. In the chemical structure, they should undergo similar metabolism to yield yield products that are the same in vivo, and along the same lines, they should possess similar physicochemical properties such as polarity, solubility, ionizability, and molecular weight, which would be indicative of a similar behavior in vivo absorption and distribution and metabolism properties. And then lastly, if you identify a number of leachables that are structurally similar, it's reasonable to group them into one assessment. If you need to, use a surrogate. But it is important to note that all the individual members of that group should be identified by their structure and/or name. And then when assessing the safety, you want to assess the total exposure for the group against whatever safety data is available for the surrogates. And so now we'll move into a few pitfalls that we observe commonly and results in a few issuing deficiencies. And so one of the more common ones that we see is not considering the worst case scenario maximum daily dose, or MDD. And this is in cases where there may be multiple dosing regimens or patient populations for a drug product. And so you want to consider the worst case scenario for those when calculating the maximum daily exposure. And the second point for not considering the worst case scenario is when there are multiple time points, say in a leachable study across the shelf life of the product, you have multiple time points, or in an extractable study where you have multiple conditions, you want to consider the highest, generally consider the highest exposure levels when performing your safety assessment. Another common pitfall is that there's no safety justification provided, and the onus is going to be put back on the applicant to justify the safety of the observed leachables. And then lastly, for here are if the extractables and leachables are not adequately characterized, then it makes it difficult for us to try and perform a comprehensive safety assessment. And then I'll also note that when surrogates are used, surrogates can only be used when the extractable leachable are adequately characterized, because if they're not, then we can't tell if it's a good surrogate or not. A couple of other pitfalls more related to toxicity assessment is the use of in silico methods for non-mutagenicity endpoints, or using Kramer classification. These endpoints these methods are not validated for regulatory assessment, and so we do not use these in our assessments and are not adequate justifications. And the next is when the appropriate context of use for a given drug product is not considered when performing the safety assessment. For example, this would be if you're relying upon an acute toxicity study to inform the safety of a leachable in a chronically used drug product. There's going to be some more information needed in order to justify the safety. And lastly, as I mentioned before, the use of ICH Q3E draft guidance thresholds are not acceptable at this point, because there's harmonization is still ongoing and these thresholds are not finalized. And so in summary, OGD PharmTox works closely with our colleagues in OPQ to evaluate the safety, or to evaluate extractable leachable studies and the safety justifications for the manufacturing equipment and container closure system and their changes. Compounds exceeding the SCT should be characterized by the chemical structure and/or CAS number. And then safety justifications should include mutagenicity, general toxicity, and local toxicity assessment based on the exposure level and the context of use specific for that drug product. In silico assessments are only acceptable for mutagenicity, and other endpoints other endpoint assessments are not acceptable from in silico assessments. And then when selecting a surrogate, we should be finding a balance between structural similarity, physical chemical properties, as well as availability of reviewable toxicity data. And then lastly, just as another reminder, control correspondences are a great resource to get feedback for specific maximum daily doses or for extractable leachable study thresholds. And so with that, I'd like to thank Dr. Zhu, my OPQ counterpart, for this presentation, and then Drs. King, Dorsum, and Gupta, who helped put this presentation together. Thank you.
And with that, we will go ahead and invite our speakers up to the stage to join us. We will also be joined today by Dr. Andre Raw, Associate Director for Science and Communication in the Office of Pharmaceutical Quality and Assessment 1 within OPQ. You'll all take your seats, go ahead and get settled. At this time, I will also invite anybody in the room who has questions to please line up at the microphones. Okay. Let's see, I saw the gentleman in the back. Go first, and then we'll go kind of bounce back and forth there. And then as well, go between the online questions. If you are online, go ahead and submit your questions at this time. We'll balance between those, do as many questions as time allows. So if everybody's ready up front, we'll go ahead and start with the first question in the back, okay?
Good afternoon all. My question is related to nitrosamine group. So what kind of data or supporting document is expected by FDA if the physical standard of NDSRA is not available or cannot be synthesized, or the chemicals, because of the chemical instability, that standard is not stable because of the steric factors. So in such situation, what kind of data you are expected during the ANDA filing? For example, ticagrelol and the NDSRA, if you see, that physical standard is not available because manufacturers cannot able to synthesize that.
Would you mind repeating the question, please?
Mm-hmm. I mean, I can.
Yeah, go ahead.
I can sort of start, okay? But so first of all, if you really cannot manufacture it, you know, so basically, if you can prove that the molecule cannot be formed based upon stress studies, you know, using, you know, various conditions, you know, aqueous conditions and organic conditions, you know, and you can prove that it cannot be formed, then you don't have anything to worry about. It will never exist. So that's one way of doing that. But if it can really form and you can't form that standard, you're going to have to make some assumptions about relative response factors and all that. So there's sort of the two scenarios that would exist. But if your product is if it's very unstable but it exists, it's probably it may not be of tremendous concern. But we would have to see the data and the application to see that.
Just to add to that, ticagrelor does not form. And last year, during the meeting, it was discussed that again, the table 1 is a hypothesis. Ticagrelor doesn't form, and I think it's pretty well known. You can probably go with that. I know, yeah, but Andre responded to that. So you have to show it, I guess. I'm so sorry. Because ticagrelor was discussed last year a lot. My question is to Dr. Yu. While CPCA has made a huge difference in our lives, but we all know that it's still limited because you know, the nitrosamine, as a student of Dr. Lepke, I can say that after 1990s, very little work got done. Organic functional groups like ether, sulfoxyl groups, even amides esters are not, you know, adequately addressed. Is FDA thinking of doing some studies to like expand the CPCA a little more, add more functional groups? Esters and sulfonils come to mind. I used to work a lot with sulfoxides. They're not carcinogenic, believe me, I'm here. I'm not dead.
I don't think I'm really capable to answer that question because we have a specific expert working on that questions. I can collect the question and forward to the right person to address it. So,
Thank you. I mean, I do agree that CPCA solved a lot of problem, but there's still a lot, you know.
I know they are working on to further refine and based on new information. But again, this is not the area I'm really expert on. And so,
I mean, if you can. The other question I have is more on the communication.
Time, we do have many questions. Would you mind if we go ahead and allow the other people? And then
This is a continuation, though. Can I just?
Because we've had these other questions, we'll circle back around if there's time, okay? I appreciate it. Thank you.
Good afternoon. I have a general question regarding the filing categories that are not for NDSRIs that are not on the list. Lately, I've been receiving conflicting information or I guess statuses from the agency. So for example, I have a split of even 50-50 where they reject the PAS and say that it's unreportable, and then another one they say that, okay, it's approved. In all these cases, the commonality was there was no control needed because it didn't exceed 10% of the CPCA limit. So is there a shift here? Because I'm seeing it's putting us in a precarious situation.
Okay, for the supplement, you're talking about the one not on the list or the one on the list table?
Not on the list.
So for the one is not on the list, basically the 26.5 is the conservative one, it's the fundamental one. So if it's less or below 26.5, basically it's CB30. If you go above the 26.5, and then you say, oh, I calculated based on CPCA, that will still be the PS, because that still need a farm task to review for the CPCA estimation. But if you get data, your confirmatory data said, okay, I'm less than 10% of the 26.5, then that's an annual reportable.
All right, so it's based on not the CPCA, but the actual value you're getting.
That's true. For these CBE or PAS, it's all based on your confirmatory data. It's not based on your CPCA.
Yeah, I just want to clarify one point here. So if it's published in the list, in our table 1 and or table 2, and you're less than 10%, it's an annual reportable. But if it's a brand new nitrosamine that has never been reported, it's not on our list, okay? And you're proposing a limit that's greater than 26.5 nanograms per day, we need to get an assessment of your CPCA calculation or other limit that you need. And per the guidance, you have to submit that as a prior approval supplement. Does that, so it depends whether it exists, it should be a supplement, okay? Generally, if you're posing greater than 26.5, if it's not on the table, okay? Do you see what I'm saying? But if it's already on the table and you already show that it's below those thresholds by less than 10% or cannot form, then you could be annual reportable.
All right, I appreciate it. Thank you.
Thank you for that. And at this time, we'll go ahead and take some questions from the online in order to balance.
Thank you. This question is directed to Dr. Fu. If a nitrosamine tests negative in the enhanced Ames test, what is the control limit for that nitrosamine? What is the control limit conversely for nitrosamine that tests positive in the EAT? Does FDA communicate externally when it is aware that a nitrosamine poses mutagenic risk?
If I hear the question right, the first one is if it's tested negative.
Yeah.
And can it be controlled?
What is the control limit for that nitrosamine if it tests negative?
Well, it's depend on what is the CPCA, what's recommended CPCA AI limit. That is the default AI limit. So if they want to control above the CPCA-based AI limit, as I mentioned, if only have a negative enhanced Ames assay is not sufficient. FDA require additional 2 tests. It's really depend on
whether they have what's the established AI limit.
Okay.
The second question,
is what if it, what is the control limit, excuse me, if it tests positive in the AET?
If it's test positive, then it's considered mutagenic. And then we, by default, would recommend based on CPCA-based AI limit if there's no already established AI limit, because sometimes there's established AI limit based on surrogate is kind of acceptable, or maybe some, like small molecule nitrosamine, they have a compound-specific AI limit. So that's the case. But if there's no other compound-specific data, then go back to the CPCA-based AI limit.
Okay, and the last component of that, does FDA communicate externally when it is aware that a nitrosamine poses mutagenic risk?
I'm not sure, actually. We generally...
Hold on one second. I think we have an answer here. If you can make your clear for us.
My name is Shruti King. So if there is a mutagenic risk, FDA may let the applicant know that the appropriate control limit is the CPCA-based limit.
Yeah, I thought they have to public, no, that's what I'm not certain.
Yeah. Do you know whether they make it externally to the public?
So if it's a published citation, if NCTR, for example, has identified that positive risk, FDA may refer you to that. Alternatively, if FDA is aware internally of a signal, a positive signal, we may communicate that on an application-specific basis.
Okay, thank you, everyone.
All right, thank you. I think we have a question here in the house, sir, if you'd like to go.
Stephen with a V, last name SUR. I'm working for a company based out of the San Francisco area. I think one of the mitigation strategies was adding antioxidant, and I think one of the slides said vitamin C and vitamin E to mitigate the risk of the contamination. Is there specific ones that may be recommended by the FDA, or is it generalized? Is there ones that work better? Thank you.
Yeah, thank you for that question. So as we mentioned in multiple presentations, that it's a fit for purpose. We tried vitamin C. We tried the other antioxidant. Some of them works, some of them doesn't work. So you need to do your method development initially. You need to test them and see, because sometimes they may work initially, but over life cycle, they may not. So you need to test each drug product with the specific antioxidant. But there are a number of those antioxidants were published by FDA that can be used. Thank you.
All right, and it looks like we have another individual stepping up to the mic here in person.
Hi, I was just wondering if the FDA had a progress update on the NDSRI progress reports that were due August 1st, and if and when we can anticipate to receive any feedback on those.
Start. So, we did request that people provide the progress reports on August 1st, 2025. And be honest with you, we are looking at all those progress reports. I can guarantee you that. But, we're going to compile it. We're compiling all the information. And then, based upon that, we will hopefully make a public announcement based upon the next steps.
Okay, so no tentative date yet as to when we can anticipate.
No specific date yet.
Okay, thank you.
Excellent. Then in that case, we'll go ahead and take a few more from online.
Okay, this is for Dr. Kaur. In hypothetical case study 1, why is it necessary to conduct a new bioequivalence study with a new API source, considering that the concern regarding nitrosamines is primarily related to toxicological safety rather than pharmacokinetics or bioequivalence criteria?
Thank you for the question. In the first hypothetical case study, it was not only the nitrosamine issue. So there were CGMP issues also. the deficiencies about the CGMP issues were communicated to the applicant, but they never addressed those deficiencies. Because they never addressed those deficiencies, then the quality of the batches could not be assured. So that's why in that case, the dissolution data only was not enough. If the quality of the bio batch could have been assured, the CGMP issues would have been resolved, then the applicant submitted dissolution data would have been enough, which is the case in most of the cases that when you're changing the API source, all you need is dissolution studies if there are no other issues. Thank you.
Okay.
Okay.
More.
Thank you much for that answer. Next question goes to Dr. Goh. The nitrosamine guidance only recommends alternative BE approach for IR product. In case study, it involved a delayed release capsule that was treated similarly to an IR product. Does it mean the agency generally treats DR product as IR product? Thank you.
Yeah, thank you. Thank you for the question. So this is still a unique case. So it doesn't mean we universally treated the DR product as the IR products. And also it's case by case. For example, in this one, it's only formulation change happening in the IR, the sugar sphere. But what if the delayed release enteric layer, although the formulation didn't change in that case, if the manufacturing change happened to the delayed release layer, then we would ask for additional data. So it's still case by case. We cannot generalize that.
All right. Thank you for that response. Another call for any questions in-house this time. All right. We have another one here at the back mic.
Again, related to nitrosamines. Is strength-based nitrosamine specifications are allowed for same product? Suppose there are 4 strengths in a product, 25 mg, 50 mg, 75 mg, and 100 mg. In any case, strength-based specifications are allowed. Instead of taking directly maximum daily doses, can I use an alternative justification for lower strengths, higher limits?
Oh, I'm sorry.
What's the question? I did not get it. So I did not get. Can you repeat your questions? I did not get what's your question.
Is the strength-based specification is allowed for a product?
Strength-based, how?
Strength.
Like, I know, just it means different. Your one product has different strength, right?
Yes.
So you want to have a different specification limit for each different strength.
Yes.
Normally, we are not allowed because your specification limit is against the API. So it doesn't matter what's your strength, your nitrosamine will still be the same. Because your API, the strength different is the API content is different for each strength. But you're against the API, so the API will be different and your nitrosamine level will be different because your strength different. But relatively go to the ppm level, they should be all the same. So I don't know why you want to go to, how can you go to different way?
If you see the specification limit calculation, it depends on the AA limit divided by maximum daily doses. That maximum daily doses is of course common for product, but of course if smaller strengths, if clinically not possible for taking that much maximum daily doses, say for example, lower strength only 2 doses is possible, that higher limit is allowed. Of course, recently FDA also accepting with the proper justifications. If at all that is acceptable, why don't you put in some Q&A sessions or in a published guidance so it will be clear to the industry?
So you're talking about like different strengths have different maximum daily dosage. That's really, I did not see much and has a different MDD for different dose. All dose have the same MDD, except I think one end they have from like 5 milligram all the way to 100 milligram. In that case, sometimes the farmhouse allow you using different MDD for different dose when you have a big range. But for majority of drug products, they're not big. It's just like 20, 30, 50. So their MDD will all be the same. So in that case, you will have one value. But when you have a product have a 10 different dose with the MDD from like low to high, then you could be, we do allow you have a different dose, have a different like spec limit when you have a wide variety of the MDDs. But that's the case. It's still case by case. You have to see your product to see if tox allow you have different MDD for different strengths. So that's based on it's nothing to do with stress. It's just for the MDDs. Thanks.
Thank you.
If I could add just one additional piece, so I agree with what Dr. Liang is saying. If there are questions about adjusting the AI limit based on your strength, definitely consider submitting a control correspondence to OGD with that specific question. It is a question that we have addressed directly, so please do that for your application.
Excellent. And I think that seems like all the questions that we have time for. We've reached our time here. So thank you, everybody, for a great conversation with this one. I think for the break coming up now, just a moment to first thank our speakers and everybody on the panel today. And then if everybody's aligned, I think to stay as close to schedule as we can, because we're about 5 minutes past, would it be all right for a 10-minute break instead and be back by 3:15? All right, 3:15 it is. We'll be back in time for then. Okay, and the kiosk should be open out front for afternoon snacks. We've double checked that. So if you guys do need anything there from the kiosk, that will be open out front, okay? All right, thank you, everyone. All right, so we will go ahead and resume with our last session of the afternoon here. And so I appreciate everyone for sticking with us. I hope today has been educational and you've learned something new and been able to take away, as well as our whole event these past 2 days. We will first jump in today with Dr. Suman Dadamunde, lead pharmacologist in the Division of Bioequivalence 3 in the Office of Bioequivalence within OGD. Then we will hear from Dr. Yang Liu, Senior Staff Fellow in the Division of Bioequivalence 3 in the Office of Bioequivalence within OGD. Then we will also hear from Commander Steve Rao, Supervisor within OPMA within the Office of Pharmaceutical Quality. Then lastly, we will also hear from Dr. Allison Aldridge, Consumer Safety Officer within the Division of Pharmaceutical Manufacturing Assessment 5 within OPMA and OPQ. At this time, if you will join me in welcoming Dr. Dadamunde.
Good evening, everyone. I know this is the last session. First of all, thank you all for joining today, both in person and online. Okay. So today I'm going to talk about ICH M13E guidance and its implications on the BE assessment, specifically focusing on the high-risk products and non-high-risk products. Let's skip the slide, standard disclaimer. So the learning objectives or outline of my today's talk is I'll provide an overview of ICH M13E guidance with respect to the scope and objective of this guidance. And then I'll jump into the FDA's framework that we have established in classifying these solid oral dosage forms, whether it's a high-risk or non-high-risk products. I'll also explain the bioequivalence study recommendations that we have provided for these products. And at the end, I'll highlight some of the case studies showing the impact of this ICH M13 guidance on the ANDA assessment, specifically related to bioequivalence. So ICH M13A guidance, this is the significant step forward in harmonizing the bioequivalence study approaches across the various agencies, specifically for solid oral immediate release dosage forms. So we all may have seen this guidance. It classifies or it provides binary classification for the drug products as high-risk products and non-high-risk products. What are high-risk products? These are the drug substances which specifically, let's say, low-soluble, combined with the formulation complexity or the manufacturing processes that may lead into differences in the bioequivalence outcome when compared with the varying GA conditions. That is, if you compare with a fasting study versus fed study, where in the sense that if you show bioequivalence in fasting study, it may not always be possible to extrapolate this results to the fed conditions as based on the differences in the test and RLD formulations. So if it's not a high-risk product, the other one is a non-high-risk product with the predictable outcomes where the drug may be highly soluble and there are no complex formulations or manufacturing designs. Now, when ICH M13 also provides the outline of once you know what the risk classification, what BE studies have to be conducted to demonstrate bioequivalence for these kind of products. So let's begin with, in this slide, I'll show the framework that FDA has established in classifying, determining the risk category for the drug products. So first, the biopharmaceutics properties of the drug, taking into account the solubility and the BCS class, as this tells us information on how drug behaves in various GI conditions. Then moving on to the complexity, whether it's coming from the formulation design itself together with the manufacturing processes. Some of them are highlighted here, like solid dispersions, microemulsions, or nanotechnologies that may have significant impact on the food effect modulation or solubility or in vivo PK performance. The other aspect we also look at is the critical excipients that the formulation may have. Specifically, we start with the RLD product because that's the information that's available for the agency at the initial stage. And if the RLD formulation has any critical excipients, like, for example, surfactants, solubility enhancers, anything that can affect the in vivo PK performance, we take that into account in classifying the drug product. Last but not least, the dosing instructions that are provided in the RLD label with respect to whether it has to be taken with the food or if there is significant food effect based on the RLD product or the magnitude of food interaction. All these properties are considered in first classifying the drug product, whether it's a high risk or a non-high risk. Now moving on to BE study conditions. So ICH M13A guidance provides recommendations for us on what type of studies have to be conducted once you categorize or classify these drug products into high risk and non-high risk products. So let's start with non-high risk products. Like I said, these are simple products in the sense of solubility wise or the formulation design wise or manufacturing process wise. So the typical recommendation is to do one study. More generally, we recommend fasting study because fasting condition often provides sensitive formulation differences, if ever exist between test and RLD formulations. However, which I don't want to go into detail, but if you look into the ICH M13A guidance, there is also alternate, instead of fasting studies, sometimes FED study is recommended if there are safety considerations or if there are significant PK variabilities or PK performance improvement. In such cases, instead of fasting study, we may recommend FED study, but it's all case by case. Now moving on to high-risk products, both fasting and FED-B studies are recommended for these products. As I stated before for the previous slide, the reason is that if ever there are any formulation differences that are obtained between test and RLD formulations, fasting study alone may not tell you how it behaves test product similarly or differently under the FED condition, under varying GI conditions. That's why the FED study is recommended to ensure that in all the conditions, the test product demonstrates bioequivalence with the RLD product and the switchability is not a concern. So now we have this ICH M13A guidance is implemented in 2024. Since then, what is FDA is doing? Based on this guidance, now we have classified the products as high risk and non-high risk. Now we are revising a lot of drug products based on this risk classification. So the previously already posted PSG recommendations were revised. They revisited to see what is the category. And if it's a non-high risk products, in the past, based on the PK endpoint guidance, we used to recommend fasting and FED study. Now moving on to single study of mostly fasting, but again, depending upon the safety considerations. However, for high-risk products, we continue to recommend both fasting and FED-B studies. One point I want to make is FDA is always open for alternate approaches. And if there are any significant advances in the scientific technologies, we adopt that. And we encourage applicants also to demonstrate if there is scientific rationale behind alternate approaches. One advancement I want to highlight here is repotrectinib tablets. Our initial classification is this is a high-risk product. As such, 3 studies are recommended for the both low strength and higher strength together. Fasting condition, FED condition on higher strength, and fasting on the lower strength. However, there is also alternate approach provided for this. In the case that FED condition study can be waived if in case fasting study is acceptable and the dissolution shows similarities between the test and RLD products. And the most important is comparison of the qualitative and quantitative way formulations between test and RLD, specifically if there are critical excipients, because we are aware that this RLD formulation has solubilizing agents. So we want to make sure that the test formulation is similar to RLD formulation, so the risk of bioequivalence under FED condition is minimal. So with that background, with that information on the ICH M13 guidance, now I want to move on to the case studies. I want to make a note here that these are hypothetical cases and any data I'm showing is the mock data. So the case studies highlight the importance of ICH M13 guidance and how it gets implemented during the BE assessment when the ANDA submissions are received. Let's go into the first one. So this is a class 4 drug, which is a low soluble drug and low permeable drug. And the RLD label indicates that the food has no influence on the extent of absorption of drug. That means the product can be taken with or without regards to the food. And when we looked at the RLD formulation, the initial process where which we go on the risk classification, this is a non-complex formulation, or no complex methodologies are involved in manufacturing this RLD product. Based on this initial risk assessment, we have classified this as a non-risk, non-high-risk product, and as such, only fasting BE study is recommended in the PSG. Let's say we received an ANDA, and during the generic drug development, the ANDA applicant tried to develop two formulations to see how they behave, and the two formulations differ in the polymer that is utilized in making amorphous solid dispersion. First of all, I want to make a point here that RLD formulation is simple. However, when it came to the test product, it involves complex methodologies. This is an amorphous solid dispersion. So the two polymers are pH-dependent polymer 1, and the other one is pH-independent polymer. As you can see in this slide, the dissolution profile, that's the first comparison to be made. When they made with the RLD product, there is significant differences in the dissolution profile between the test and RLD. And also the point is there's differences between the two formulations that generic drug manufacturer is trying to develop. And the question that comes now is how this translates in vivo performance. How this difference, if ever these dissolution differences, can have significant impact in vivo. So pilot studies are conducted in this case, which is less than 20 subjects. And I understand this is a busy slide, but what I want to convey the message is that depending upon the formulation they choose, pH-dependent or pH-independent, the BE outcomes are varying. And not only that, the fasting study is not predictable of the FET study. The results are varying between the formulation and also between the condition. Again, this is going to the point that together with the drug substance properties, in this case, this is a crystalline form which has low solubility, and that complex methodologies are involved. So the food formulation interaction may vary between the test and RLD. So it points that FET condition, FET study is important in these cases, in these products. So for this ANDA submission, even though initial classification is a non-high risk, but based on the test product, this is considered as a high-risk product. As such, both fasting and FET BE studies were submitted. So let's go to the case 2 study. So this is a fixed dose combination product. It has two APIs. First API is a highly soluble. I did not mention it here, but I want to highlight the second API, which is a class 2 drug, which has a pH-dependent solubility. Again, the RLD labeling states that food has no influence on the exposure of both the drugs, and this can be taken with regards to the food. The recommendation that comes in the RLD label is based on the RLD formulation characteristics, because they might be excipients that may modulate the food effect. So the RLD formulation has complex methodology, which is amorphous solid dispersion. So based on this, our initial risk assessment is this is a high-risk product, and as such, we recommended both fasting and FET BE studies in the PSG. Now, ANDA came in-house, but at the time of submission of this ANDA, there is no PSG. So the generic manufacturer is unaware of the PSG, the BE study recommendations. However, based on their test product formulation characteristics, they've conducted fasting BE study, but in case of FET BE study, they proposed an alternative approaches of modeling data. They also understand that their formulation is complex, so FET BE study is needed for this. However, they proposed alternate approaches. So as I mentioned before, FDA is always open and encourage applicants to provide alternate approaches only with proper scientific, rational, and validation to show that the FET study, under whatever data they provided, they can actually show that there is no differences between the test and RLD performance in vivo. In this case, we did accept, in the sense the methodology modeling data is considered. However, there are some deficiencies related to this modeling data. So a letter was sent out to address those if in case they want to choose this alternate approach pathway, or alternatively conduct a FET-BE study to show or to demonstrate the bioequivalence between both the products. So now let's move on to the third case study, which is my final case study for today's talk. This is a class 4 drug. It has low solubility, low permeability, and the RLD labeling does indicate that food has no effect on the drug bioavailability, and the product can be taken without regards to the food. Now, the RLD product has critical excipient, which is a pH modifier. I want to make a note here that having just one pH modifier or solubilizing agent, we do not consider it as a high-risk product. However, if the magnitude of the food interaction or together with the properties of the drug substance and the methodology, we may consider it as a high-risk product. Initial classification, we made it as a non-high-risk product, but finally, based on the data that we have received during ANDAs, the classification was changed to high-risk product. Now, if we see the current PSG for this drug product recommends both fasting and FET-BE studies. In the next few slides, I'll go through the scientific rationale, how this recommendation came into. So we received an ANDA submission. There's no complex formulation. There's no manufacturing method, complex manufacturing method, and also there are no pH modifiers in the formulation. So the applicant submitted both fasting and FET-BE studies. The fasting BE study passed, whereas the FET-BE study failed. Now FDA recommended the applicant to conduct the FET-BE study as well, and also reformulate their test product because of the IAG limits and other purposes. So we looked at the ANDAs that were submitted across in-house that we have received for this drug product. So we have made based on that few observations. So generic formulations, if contains pH modifiers, pH adjusters, then consistently we saw fasting and FET-BE studies being passed. However, if the generic formulations do not contain pH modifiers, then it exhibited not only inconsistent food effects, the PK variability also varied depending upon the formulation, and also there is no consistent pattern in the acceptance or the passing or failing of the FET-BE studies. So based on this, we understood that pH modifiers are playing a significant role in the in vivo performance, specifically under the FET condition. So together with the RLD information that we have available, that RLD contains pH control agents for the stability enhancement and the PK variability that we have observed across all the generic formulations with respect to the differences in the formulation, specifically for the critical excipient, and based on the inconsistent food effects, now the current PSG recommendation is to have both fasting and FET-BE studies. So with that, I want to summarize. The key takeaways is that the ICH M13A guidance for the non-high-risk products, typically a bioequivalence study recommendation is based on a single study and mostly fasting, whereas for the high-risk products, it necessitates both fasting and FET-BE studies. I want to make a note also here that the initial risk assessment that FDA makes, whether it's a high-risk or non-high-risk product, is based on the available information of the RLD labeling, RLD formulation characteristics, and the biopharmaceutics properties of the drug substance. However, once the ANDA is received in-house, we also evaluate the generic formulation. If the generic formulation has any complex methodologies, as I have described in previous slides, or we think that there is a magnitude food effect or food interaction is significant, then we also recommend fasting and FET-BE studies in such case. And again, I want to reiterate the point that PSGs are guidances. They are not requirements, and applicants can propose alternate approaches. However, there should be a strong scientific rationale behind these approaches if any in vitro is provided in lieu of conducting any in vivo studies. With that, I conclude my talk, and I would like to thank our OB office management, Dr. Partha Rai, who is in house room today, and Dr. Bingley, along with my division management, Dr. April Brady, Dr. Kiran, Dr. Ezang, and all the BESSs who have provided me these case study examples. With that, thank you all for your attention. Now I will hand it over to Dr. Yanglu. Thank you.
Good afternoon, everyone, and greetings to our online audience. My name is Yang Liu from DB3 OB-OGD. So in my talk, I will kind of switch gear from like the high-level BE study design and data integrity and the guidance to a little bit more specific on bioanalysis. So in the past 2 days, very informative session, my OB colleagues has been talking about how to proper design a BE study, how to interpret the data, how to make sure the data integrity is assured. However, at the end of the day, in my opinion, the BE establishment is based on numbers, is based on the drug concentration seen in the BE samples, and that is supported by reliable, accurate, and valid bioanalysis data. So that is what I'm going to be talking about today. So this is the disclaimer. I'm going to skip it. So for today's presentation, hopefully I can cover the following things to describe the bioanalytical challenge of endogenous drug in ENDAs that we receive and review. And I will try to summarize different approaches recommended by SHM10, how to handle the bioanalysis of analytes that are also endogenous molecules. I will try to explain the surrogate matrix approach, which is the focus of my talk today. And I will try to use a couple of mock examples based on real cases, the evaluate of analytical data acceptability regarding matrix effect and everything that is mentioned, but not very specifically in M10 guidance. So first of all, endogenous drug. Endogenous molecule play very, very important physiological functions and pharmacological roles. And some examples are sex hormones, thyroid hormones, fatty acid, etc. The endogenous molecule in ENDA land or in generic drug land, most of the time is the active pharmaceutical ingredient or APIs that we are actually actively looking into for the concentration after drug dose. So the post-dose endogenous drug concentration are the fundamental of PK assessment, and which is used to demonstrate BE in our ENDA. In other words, it will be used to determine whether a drug is BE or not to the reference drug. However, bioanalysis of endogenous drug concentration can be very challenging compared to regular drugs. And part of the reason is actually shown very actively in this figure. This is an example chromatography of endogenous drug after drug dosage. We can see in the figure, the blue one is actually the concentration peak observed. However, this is not just from the drug that a patient or subject are taking. It also includes the endogenous concentration, which we call the baseline. There are certain ways when we are actually handling the study data, for example, baseline correction as recommended in most PSGs for endogenous drugs. However, the bioanalysis of such drug can also be very challenging. That is because it is almost impossible to find a analyte-free authentic matrix. So in other words, we can actually consider in one way that the endogenous concentration of the target analyte in the subject matrix can be seen as an interference to our bioanalytical methods. There are recommendations about how to handle and how to conduct bioanalysis of endogenous drugs, specifically mentioned in SHM10 section 7.1. There are several recommendations, recommended approach for handling this type of drugs, including surrogate matrix, surrogate analyte, background subtraction, and standard addition. So the benefits, actually, based on our in-house ENDA review experience, most of the ENDAs, related to endogenous drug, actually use the surrogate matrix approach. Why? The reason is this approach can provide quite a few benefits. That includes the analyte-free matrix because we can design and we can screen, we can design what surrogate matrix to use and make sure there's no interference to impact the quantitation result. And also this kind of method is also usually very, very robust, very easy to handle, especially for chromatography methods. They provide very promising sensitivity, and I will be talking about that in detail in one of my later slides. And also, there is always a discussion in the industry as well at the agency that is that acceptable to actually try to screen and find an interference-free authentic matrix, which is the same matrix type as the BE samples? Is that kind of interference-free authentic matrix screening acceptable and recommended? And I'll also mention that in my next example, case study 1. So let's just call this drug endogenous drug A, and the matrix type is human plasma. Based on the literature and empirical data, the human plasma, the mean human plasma of the endogenous drug A is about 0.2 to 3 nanogram per mil. And that is actually expected to be the baseline that we will see in the BE subjects. So in one of the applications we reviewed, we have experience working with is the applicant actually trying to get rid of the interference in the blank matrix, and they set the lower limit of quantitation, or LQ, to a relatively high level of 2 nanogram per mil. By screening some of the loss, the applicant successfully identified some loss with a lower concentration that is even lower than 20% of the LQ. Per SHM10, if the interference peak or the interference peak area is lower than 20% of the target LQ, the matrix is considered analyte-free. However, that can be problematic. Again, just like Suma mentioned, the case example in my presentation are mock example based on real cases, but the numbers are not real. So the applicant's method design and rationale are as follows. Authentic matrix was used and screened. They were used to prepare all the calibration standard, quality control samples, etc. And the human plasma, some loss used in the validation and sample assay were identified interference-free, which is 20% lower than 20% LQ. And the method was validated and used to support the BE study sample analysis. So this method by this applicant was actually treated very similar to a non-endogenous drug, very similar way. Actually, they are not considering the endogenous level anymore. However, there are concerns about this current approach. And the reason, as I said, the matrix, the authentic matrix the applicant used is not really analyte-free, is man-made analyte-free because they raised the LQ level. And what problem can that cause? Just go to the observation in our review is pretty much all the pre-dose samples were below the limitation, below the quantitation limit or BQL. Or in other words, they were all reported as 0 in the PK concentration data set. And that caused no baseline correction can be performed because there's no baseline detectable. And also, not only the pre-dose samples, but there are insufficient measurable concentration in the elimination phase because ever since the drug concentration dropped below this relatively high LQ level, it was reported as 0. One example, in one case, in one subject, the Cmax in one period was observed as 7 nanogram per mil. Only, if we do the math, only after 2 half-lives, the concentration, theoretically, it will drop to about 1.75. But using this current method in this ENDA case, this 1.75 cannot be detected because it's below the 2 nanogram per mil LQ. And that makes not only we lost a lot of data that can be collected using a different approach, but also this can make it very, very hard to make a conclusion whether the drug is BE due to the limited data size. This figure actually is showing the comparison in the same case if the concentration was detected using a surrogate matrix, which is shown in the blue curve, or using the current method used by this applicant, which used authentic matrix with a higher or elevated LQ level. We can see we kind of lost a big chunk of the AUC due to the limited sensitivity. So the not only the selection of the matrix or a surrogate matrix is important, but also when a surrogate matrix is actually used, it provides good sensitivity, not like the previous case. However, it also requires additional consideration and validation during the validation work. One very important part of the validation, if a surrogate matrix is used, is a surrogate matrix effect. Because in this case, the general, first of all, let's go back to one step. The surrogate matrix, according to SHM-10, can vary very widely. It can be a very simple buffer. It can be a depleted matrix of the same species, or it can be a matrix from a different species that is lack of the interference. So how to prepare our standard and quality control sample to just try to eliminate the impact of interference? The calibration standards are always prepared in surrogate matrix that can bring the LQ to a certain level that can suffice the purpose of the analytical work in the BE study. However, for quality control sample or QC sample preparation, is the current thinking at the OGD is for method validation, the QC is expected to be prepared in both sets, both surrogate matrix and authentic matrix, for direct comparison, and try to spot any potential issues ahead of time before we actually touch the BE sample. However, for BE subject sample analysis, the surrogate matrix may not be needed. However, the QC should be prepared in authentic matrix at least. It is recommended to still have 2 sets of QC in both matrix types, but at the minimum, the QC in authentic matrix is required. That is because per M10 guidance and by the general thinking, the QC sample, the purpose of QC sample is to mimic the whole lifetime of the incurred sample. So they should be prepared in the same authentic matrix type. Because there are 2 different types of matrices used in such surrogate matrix approach methods, surrogate versus the authentic, we can expect there will be some matrix effect difference between the 2 matrix types. So let me go into my second case study. In this case, let's call the analyte endogenous drug B, and the calibration range, let's set it to 5 to 250 nanogram per mil with an LQ of 5. So the blank matrix concentration, and that also bring up the question that if I'm using the calibration standard is prepared in a surrogate matrix, I don't need to worry about because there's no interference. But for a QC sample, which is prepared in the authentic matrix, there is an endogenous concentration. Do I need to consider what lot to use? Does the endogenous level, high or low, does it matter? Actually, the answer is yes. And in this case example, we can know why. In this case, the blank plasma used by the applicant contain a relatively high endogenous concentration at 60. And these lots or pooled lot of plasma was actually used in both validation and sample analysis. And that gives the authentic low QC a nominal concentration of 75, 60 from the endogenous, and 15 from 3 times LQ spiking, which is a very standard QC preparation approach specified in the guidance. However, that brings up a big issue, and this is the calibration range. We can see is ranging from 0 to 250. The 3 red circle marks the concentration levels of the authentic QC samples. We can see the lowest concentration is, as we said, is 75. However, whenever we are analyzing the BE samples, if not all, but most of the pre-dose samples and most of the elimination phase samples are actually located in the green box, which is lower than the authentic, sometimes much lower, close to like 10 or 15. This brings up a concern from a review perspective. The concern is there's insufficient data from validation and in the assay. Both authentic low QC are relatively high comparing to some of the observed concentration in BE samples. And that brings the concern that there's insufficient data to support the accuracy of the BE sample concentration on the low end. And there's the proposed justification in this case is to reperform a partial validation trying to screen and select some authentic matrix lots and with lower endogenous concentration and use those lots, save them and use them to prepare authentic low QC. In this case, the applicant actually screened and selected a couple of lots with the mean endogenous concentration of 10 nanogram per mil. And we can see we still spike the sample with 3 times LQ. Now the normal concentration is only 25. Comparing to the previous QC set of 75, this can cover most of the pre-dose sample, and it gives not only the applicant, but also the reviewer more confidence that the samples are bracketed. However, in this case, it also say this issue can actually be avoided if a certain approach was taken. That is, before preparation of the authentic QC sample, it is highly recommended to screen more lots of authentic matrix comparing to other methods to try to identify and use the lots or lots with lower endogenous concentration level, just in favor of the authentic low QC preparation. Another thing that the surrogate matrix approach should focus on is the parallelism. I know parallelism is sometimes the idea actually comes from the ligand binding. However, in M10 guidance, it doesn't actually specify parallelism is only required or recommended for ligand binding study. So that is why in chromatography methods, it's also recommended to have the parallelism data to demonstrate the sameness of between the surrogate and the authentic matrix. And missing parallelism data sometimes can cause inadequate method validation by the reviewers. In addition to just setting up the parallelism to see if the two types of matrices perform similarly, a parallelism study can also provide additional data. And that is if we set the two sets of curve, one in the surrogate matrix, which is in the blue curve, one in authentic matrix, which is in the orange one. If we extrapolate the authentic matrix curve, we can actually get a more accurate endogenous concentration. I know in a lot of validation report, the very first thing to be validated is to measure the concentration of the pool blank plasma, authentic blank plasma. However, this is a little bit too fast in a reviewer's eye because the method has not been fully demonstrated. The determined blank concentration may or may not be accurate. The scientists who actually work on this method may have more confidence in this method because this has already been done in method development or based on previous experience. However, for reviewer, we do not have access to method development data. So it is recommended to have one set of parallelism validation in the report. The last focus, there are so many things, but the last thing I want to focus on today is the matrix effect assessment. So surrogate matrix may present different matrix effect, which is expected, but most of the cases it's acceptable if certain justification is provided by validation data. For example, sample extraction should be optimized to mitigate the potential impact of matrix effect difference between the surrogate and the authentic. If this is not done correctly or properly, it can cause some problem. Another thing that can help mitigating this potential impact is the using of stable isotope label internal standard, which can also provide a lot of benefits. So the matrix effect, first of all, after all, this is a quantitation method, it must not impact the quantitation accuracy. However, the recommendation for matrix effect validation is more than just QC accuracy. Let's say, the QC in both sets of matrix actually passed the criteria. And in the last case study I want to share with everyone today is the viable matrix effect and its potential impact. This table actually shows the method validation data, and in here we can see the IS response actually varies between the surrogate matrix and the authentic matrix. In this case, the surrogate matrix is BSA buffer. We can see whenever the authentic matrix human serum was used, the IS response rate dropped significantly, which is understandable. And because of the use of the stable label internal standard and the demonstration for accuracy of QC sample in both sets, this is not perfect, but it is justifiable. However, when things moved to the BE study, things start to change a little bit. First of all, I want to emphasize the order of the sample I'm showing in this figure is not the actual order of sample in the run, but to demonstrate my point, I just resort them. So the first set of data on the far left is the standards, and which is and the QC, which are prepared in the buffer in the surrogate matrix. We can see a pretty high IS response. The second set is the serum QC, which is prepared in the lab. And the third set of QC is real subject SPE samples. We can see this time the signal suppression, it increased a lot. In the previous case, validation case, there was about a 50% signal suppression, which is not ideal, but understandable. But in this case, the signal suppression raised all the way to more than 95%. Not only that, this raised a question and caused a lot of sample that has the internal out of range, but also it caused a concern from the reviewer perspective about the accuracy of the data itself. One of the reasons is the chromatography. These two samples, or these two chromatograms actually are showing two samples with a very, very similar observed concentration, but in different matrix types. The left side is the LQ sample, and the integrated peak is the target analyte. On the far right side is actually the BE sample, giving a very similar concentration. We can see the chromatography now is actually in the middle of a lot of interference peak, and it actually make it less confident that this is actually representing the true concentration in the BE sample. As I said, this actually raised concern from different aspects, and the proposed justification in this case is actually to conduct an investigation in the lab to demonstrate the accuracy of QC prepared using actually the pre-dose sample of these BE subjects. However, this can also be, the best effort should be also made to prevent this kind of issue. This include the best effort to try to mitigate the matrix effect by improving the extraction procedure, by improving the chromatography conditions to actually separate the target peak and the potential interference peak, etc. So at the end of my presentation, I would like to wrap up with a couple of challenge questions. The first one is, which of the following is considered a surrogate matrix if the subject sample of BE study is actually in human plasma? There are 4 options: BSA buffer, depleted human plasma, or rat plasma, or all of the above. Apparently, the answer is D, all of the above. Anything that is different or treated differently as the target BE sample matrix is considered a surrogate. The second question is a true and false question. If an endogenous drug analyte method using the surrogate matrix approach, it can still be considered inadequate even though all the QCs prepared in both surrogate matrix and authentic matrix met the accuracy and precision criteria. Is it true or false? This is true because, as I mentioned in one of the slides, accuracy and precision is not the only thing that we focus on whenever we're developing a method or validating them that is using a surrogate matrix. So to summarize my presentation, surrogate matrix approach can offer a lot of benefits, but also it requires additional consideration, mostly that resulted in more validation parameters in the work. And additional experiments and data in the method validation are essential to support not only the cleanness, but also the sameness of the surrogate matrix used in the method. And the potential impact of using the surrogate matrix on bioanalytical data reliability may also require additional scientific justification if it's not covered in the original validation and sample analysis. Last but not least, I would like to give my sincere thanks to the management team at OB, Dr. Partha Roy, and the DB3 management, Dr. April Brady, Key, and E. And also, I would like to thank SBIA committee for inviting me here. Thank you. Now I will turn this to the next speaker, Commander Ryu.
Good afternoon, everyone. Thanks for joining us from today. in both in person and virtually. My name is Steve Ryu. I'm a commander in the United States Public Health Service, working in the Office of Pharmaceutical Manufacturing Assessment at CDER. Today, I'm going to talk about lyophilization in pharmaceutical manufacturing, specifically the common deficiencies we see, effective control strategies, and what FDA expects from a regulatory standpoint. So what are we going to cover today? First, we will walk through lyophilization as a manufacturing unit operation and why it matters. Then we will dive into the common deficiencies we are seeing in drug applications. We will also discuss the control strategies that manufacturers are using. And finally, we will look at some real inspectional findings to give you a sense of what we are seeing in the field. Before we get started, let's get a quick poll to see where everyone's at. Which of the following best defines lyophilization according to the 1993 FDA Guide to Inspections of Lyophilization of Parenterals? Is it A, process where solid ice turns into liquid water? Or B, the process where liquid water turns into water vapor? Or C, a process where water is removed from a frozen product under vacuum, allowing ice to change directly from solid to vapor without passing through a liquid phase? Or D, none of the above? Yes, the answer is C. So with that definition of lyophilization in mind, let's look at some trends. According to the LyoHub annual report from 2025, we are seeing steady FDA approvals and a recent uptick in demand for lyophilized drug products in many different therapeutic areas, especially oncology and infectious disease. You can see from this pie chart that generics took up 65% of the lyophilized drug applications approved in 2024. Now, lyophilized drug products accounted for about 5% of the roughly 800 generic applications we review each year. That might not sound like a lot, but here's the thing. The most frequent process-related deficiencies we encountered actually come from lyophilization. So even if it's a small percentage, it's an area where we really need to focus our attention. Let's put lyophilization in context. When we are manufacturing lyophilized drug products, we are typically looking at a sequence of unit operations. We start with compounding, mixing your ingredients, then you've got filtration to remove particulates and microorganisms. Next comes aseptic filling, where you're filling vials in a controlled environment, followed by lyophilization or freeze drying, and then crimping and packaging. Lyophilization sits right in the middle of this process, and problem here can impact everything downstream. Maybe your cake collapses and you've got appearances issues. Maybe you didn't dry completely and you've got some stability problems. So getting lyophilization right isn't just about that one step. It affects the quality of your final product. So what are we seeing go wrong? There are several recurring issues. First, formulation-specific problems that aren't adequately addressed. Every formulation is different, and what works for one product might not work for another. We see applications where manufacturers haven't adequately characterized how their specific formulation behaves during freezing and drying. Second, poor lyophilization cycle development. Basically, the cycle hasn't been properly designed or optimized. This is a big one. The lyo cycle, your freezing, primary drying, and secondary drying phases needs to be carefully designed based on your product's characteristics. We see cycles that are just copied from somewhere else or haven't been properly optimized. How do you know when your product is actually dry? If you can't answer the question with data, you've got a problem. Cake collapses. this is when your nice, elegant, lyophilized cake turns into a shrunken, melted mess. It's usually a sign that your product temperature exceeded the critical temperature during drying. It's a quality defect, and it tells us your cycle isn't under control. And finally, we are seeing applications with no data or justification for scale up from development to commercial manufacturing. These are all preventable issues if the right work is done upfront. From a regulatory perspective, what are we looking for? We want to see manufacturers reduce uncertainty in the lyophilization process. A key part of that is proper endpoint determination, knowing when your product is actually dry. We are encouraging the adoption of process analytical technology, or PAT tools, to help with this. We also want to see increased detectability of failure modes, which should be aligned with quality risk assessment principles outlined in ICHQ 9. The bottom line is understand your process, control it, and be able to demonstrate that control. And let's talk about control strategies. One approach is direct measurement using thermocouples. The idea is here simple. You stick thermocouples directly into some of your vials to monitor product temperature during the cycle and identify the endpoint of drying. You can see exactly what's happening in those vials. However, those vials with thermocouples aren't representative of your entire batch. Why? The thermocouples can serve as a nucleation site for ice crystallization from freezing, leading to higher nucleation temperatures and larger ice crystals. This can result in lower product resistance and faster sublimation. They are different from the rest of your product, so you are not getting a true picture of what's happening across all your vials. In addition, inserting thermocouples requires human intervention, increasing the risk of contamination, which is not suitable for commercial manufacturing. So while thermocouples are useful during development to understand your process, you can't rely on them as your primary control strategies for commercial manufacturing. Other common PAT tools include comparative measurement using Pirani gauges and capacitance manometers. Pressure measurement with Pirani gauge is based on thermoconductivity, which is sensitive to gas type. In the beginning of primary drying, they read higher than absolute pressure measured by capacitance manometers. Why? Because during the primary drying, the majority of gas species in the lyo chamber is water vapor, whose thermal conductivity is higher than nitrogen gas, which is continuously bled into the chamber to replace the removed water vapor and control chamber pressure. At the end of primary drying, the pressure read from both sensors becomes comparable. These can be useful, but they come with their own challenges. They require frequent calibration to remain accurate, and Pirani gauges in particular aren't always robust enough to withstand repeated sterilization cycles. So while these tools can help, you need to think carefully about their limitations and maintenance requirements. This slide shows the common control strategies we are actually seeing in drug applications. You can see the distribution of different approaches manufacturers are taking. It's worth noting that the most popular method used to determine the endpoint of drying for generics is thermocouple-based measurement, followed by a trial and error approach. The key is choosing the right strategy for your specific product and process, and then validating it works. Now look at what we are finding during inspection between 2015 and 2019. Visual inspection issues accounted for 27% of findings. What are we seeing? Inadequate defect libraries for training. Think about it. If you're training someone to inspect lyophilized products, they need to know what defects look like. They need reference materials. We are finding facilities where that doesn't exist. We are also seeing lack of proper training programs. People are doing visual inspection without adequate training on what to look for. And in some cases, there aren't even SOPs in place. No written procedures for how to conduct visual inspection. This is your last line of defense. If a defective vial makes it through visual inspection, it could make it to a patient. This has to be done right, and it has to be done consistently. Cleaning and sterilization issues made up 23% of findings, nearly a quarter. And these are really fundamental CGMP issues. We are seeing improper clean in place and sterilize in place procedures, especially for intricate components. Lyophilizer have a lot of nooks and crannies. If your CIP and SIP procedures aren't designed to reach all those areas and identify cold spots, you've got a contamination risk. We are also finding lack of integrity testing for gas filters. And we are seeing inadequate leak testing for the lyophilizer itself. If your chamber isn't holding vacuum properly, your entire cycle is compromised. Loading and loading issues represented 22% of findings. Common problems include inadequate or completely absent smoke studies to demonstrate proper airflow. We are also seeing first air being breached during vial transportation or while loading partially stoppered vials into the lyophilizer. Remember, these vials are only partially stoppered during lyophilization. They need to be open to allow water vapors to escape. That means they are vulnerable to contamination during handling. If you are not protecting them from these risks, and that's a problem. So in conclusion, what should we take away from today? First, you absolutely must identify the endpoint of drying. This isn't optional. Second, you need proper control strategies in place that are appropriate for your product and process. And third, that is critical, your manufacturing facilities should be ready for inspection and fully compliant with CGMP requirements at the time you submit your application. Don't submit an application thinking you will fix things later. Don't wait until we schedule a pre-approval inspections to address deficiencies. Be ready from day one. So before we wrap up, I'd like to take this moment to acknowledge my colleagues who contributed to this presentation. And with that, thank you for your attention, and I'll hand it over to the next speaker, Dr. Aldrich.
Thank you, Commander. Well, congratulations. You made it to the last one. Excuse me. So this disclaimer you've been seeing most of the day, these are my thoughts and only my thoughts. And you've seen pieces of this slide today too. Everyone deserves confidence in their next dose of medication. And pharmaceutical quality assures availability in addition to safety and efficacy for every dose. And people think about when they get to deficiencies. I was in industry at once. I promise you, we're not trying to make your lives miserable. I'm only speaking about myself. I'm not trying to make your lives miserable. I'm trying to give you information to help you get your applications approved as quickly as possible. So you've heard several people from my sub-office today, the Office of Pharmaceutical Manufacturing Assessment. We do have a mission, and that is that quality pharmaceuticals are consistently manufactured over the product life cycle. And our vision is that we're the best at holistic assessment. So a little more about OPMA. You've heard from our microbiologists today. You've heard from Commander Reed. We do process, facilities, and microbiology assessment. And we do ANDAs, NDAs, BLAs, and we also help with INDs, which is not on there. And our work goes across the entire life cycle of the products. So we talk about one quality voice. We're all working on an integrated quality assessment with the other disciplines. And so we form teams and we all write chapters to this integrated quality assessment. And that's where everything that I'm going to talk about today comes from, is from these IQAs, as we call them. So our learning objectives, what are some of the things that FDA has done to expedite drug approvals? We're going to look at the common manufacturing deficiency trends. And then we're going to review the top 10 deficiencies. And then we're going to talk about some documents to help you with your submissions. So one of the enhancements that came about in GDF 3, and that was sending information requests and discipline letters late in the assessment cycle. So for example, if we send a DRL and there's still some things that we aren't satisfied with, we'll go ahead and do another round of information requests in hopes that we can go ahead and approve it in this same review cycle. Now, in some cases, the review cycle might be increased by a few months, but it's still trying to get the drug approved as quickly as possible. With GDF 7, they introduced 4-part harmony, and that's for all the information requests. And 4-part harmony has, as you probably guessed, 4 pieces to it. They acknowledge the information provided. It identifies the issue, in other words, what's missing, what we need for us to make a regulatory decision, and then we try to provide a specific reference or information to support our request. So I'm going to read you an example of one. We acknowledge that nitrogen is used during the cup filling operation. However, the filling operation is part of the manufacturing process, and the cups are blanketed with nitrogen. As stated in 21 CFR 211-186b3, a complete list of components designated by names or codes should indicate any specific quality characteristics, and then revise the batch formula to include nitrogen as a processing aid. I don't know if you could tell by the language that this was actually a second IR for the same issue, because the applicant felt that the filling was not part of the process, but we believe that it is part of the process. So all the products I'll be talking about today are liquid drug products. So that includes injectables, oral dosage forms, topicals, and insufflates. And I actually take an insufflate for my allergies. And it comes in a new device because there is no generic at this point, so I encourage you. But anyway, the difference with this one is when you blow it into your nose, more of the drug is supposed to be going into your sinus and not just your nose. So I'm appreciative of that product, but I really would like to pay for a generic. So our poll question, which unit operation do you think has the #1 deficiency? Raise your hand for mixing. See a couple of hands. How about filtration? Few hands. Filling and sealing. Few hands. Packaging. Few hands for packaging. Oops, it didn't. Okay. The answer is C, filling and sealing. Okay, just to talk about how we gathered our data. We looked at more than 2,000 IQAs, which are assessment documents from our database. We took 1,850 of those from January 2019 to June 2023. This resulted in more than 6,500 IRs, or deficiencies, depending where we're at in the review process. Now, one thing I would like to say is some of the IRs may have been counted twice, and I'll tell you why. We categorize the IRs by keywords using Visual Basic Script. And the categories included unit operations and sections in our IQA. In other words, things that we evaluate for every single application. And some examples include filling and packaging, which are unit operations, and in-process controls. So here is the big picture by deficiencies by keyword search. And you see the largest one is filling, 20%, followed by batch record, mixing, filtration, drug substance attributes, and then packaging is small, certification, you heard about 21 CFR 177 earlier today. That's what we call certification. That's one of the certifications. Compatibility, sampling plan, hold time, and Commander Reed gave a great presentation on lyophilization. And then batch formula and process diagram. We look at all of these things for every application. So when I show the results, we will show you mixing, filtration, filling and sealing, lyophilization, and packaging. And a different IQA section is drug substance attributes. We'll look at that as well. So for mixing, the most IRs we see have to deal with a batch record. So, and then it follows by in-process controls, or IPCs. We have a other when it's full of IRs that we couldn't put in other buckets, or they happen at such low numbers, we couldn't have a big bucket. Followed by process parameters. And these process parameters are typically critical process parameters that we're thinking about. And then we heard two great presentations on extractables and leachables, and then sampling and then certification again. So I'll give you one example. We acknowledge that proposed ABC release specifications for final release testing of the XYZ injection and results from the exhibit batches. However, you failed to establish an IPC for the ABC assay for commercial manufacturing. And so, of course, what we want you to do is to establish an IPC for assay. And then the reference that we cited is 211.110C. There is a draft guidance for 211.110. I don't know if you guys got a chance to comment on it, but in the future, look out for it. The next one is filtration. And for filtration, the biggest issue is flush volume. And so if you have a filter, you may need to flush it because it may be holding up things in the filter, or it might be absorbing the API, any of those things. So you need to do some work in your development to determine if you need a flush volume. You may not need one, but if you don't need one, you need to put that in your submission saying why you don't need one, a justification. And then we have other, again, a lot of different IRs that won't fit in the other buckets. And then extractable leachables, certification, IPCs, process parameters, and again, batch records, and hold time. You heard a little bit about hold time from the micro talk this morning. Filling and sealing. 30% of the issues are the batch record. 30% are the IPCs, although we rounded to 30%, but batch record was a little higher than the IPCs. And we see flush volume again. And we have an other here, sampling, process parameters, gross contact, certification, and again, hold time. But the big ones here are the batch records and the IPC. So lyophilization, can't say much more about what Commander Reese said, except for the fact process parameters, as he discussed, are the biggest issue, 40%. And then other was a big chunk, 25%. And then batch records, IPC sampling, extractable leachables. So we took a deeper dive into the other to see what it looks like. And 45% of that is the lyocyclograph. Commander Reed showed one in his presentation. We want to see those when we're looking at your application for the exhibit batches and possibly even the other batches. Because if you made other batches, we kind of like to know about those as well and how they turned out. And then the equipment load, he talked briefly about the load. And then there's still 18% of other things we couldn't put together, and then stability. Packaging. The other was really huge here because there's so many things that were mentioned, but not enough to put into one bucket, the other buckets that we had. So behind other, we had batch record at 26%, followed by combination drug product comment. And then IPC, sampling plan, process parameters, and uniformity. So we looked at this other as well. And 34% was the device component. Then visual inspection. You've heard about visual inspection. Then labeling, and then there's still some we could not categorize. So drug substance attributes. So we look at the drug substance from a point of view of how it's going to behave during the process. Is there anything in the process that we need to be concerned about based upon the drug substance? So we've seen the most issues with pH at 16%, solubility, oxidation, light, hygroscopicity, which means, it could absorb water over time, and then stability, temperature, assay, and polymorphism. And so if you have a drug that is sensitive to oxygen, we hope that you take controls, that you have a strategy to minimize contact with oxygen of your drug substance throughout the process. So now we're going to go into the top 10 deficiencies. Number 10 was drug substance attributes solubility. Number 9, drug substance attributes pH. Number 8, mixing process parameters. Number 7, filling, sealing, extractables and leachables. Number 6, filling, sealing, sampling plan. Number 5, mixing and process controls. Number 4, filling and sealing flush volume. Number 3, mixing batch records. Number 2, filling and sealing IPCs. And number 1, filling and sealing batch records. So you see, filling and sealing is our most problem unit operation. And so if you're trying to start fixing things, I would recommend this is where you start. So now I'm going to talk about some reference documents. FDA guidances. You've heard about guidances all day long and yesterday. They are a good friend to you. Please use them. And I'm just going to talk about a few of the guidance that could be very helpful to you. The first one is good ANDA submission practices guidance for industry. That's a great place to start for your ANDAs. Process validation, general principles and practices. Sterile drug products produced by aseptic processing, current good manufacturing process. And you've heard about product-specific guidances for the last couple of days. And ICH Q8 through Q10, which is pharmaceutical development, quality risk management, and pharmaceutical quality management, respectively. All of those are your friends, and you need to get personal with those. 21 CFR 210-211, CGMP. And just ones that we look at a lot, 211-65, equipment construction. Because that's where we look for if your equipment is not compatible or things like that, because you don't want to use equipment that's going to change the quality of the product, right? Subpart F, production and process controls. So subpart F is 211-100 to 211-115. Written procedures, deviations, charging of components, calculation of yield, equipment identification, sampling and testing of IPCs and drug products, time limitations, and control of microbial contamination. The next one, the USP 87 and 88 for biological reactivity tests. That goes hand in hand with 177 for the indirect food additives for the polymeric components that you use. 1790, visual inspection of injections. 790, visible particulates. So the take-home message, the goal of all of this is for quicker approvals. And as you know now, the filling unit is the unit operation with the most deficiencies. And please refer to the FDA guidances, 21 CFR 210-11 and USP. And just want to make acknowledgements. David McChesney was one of our students who was an ORI student. And then Alexander Dura helped us with our data extraction. James Norman came up with the IR extraction. And then our management, Vidya Pai and Ying Zhang. Challenge question number 1. What is one of the FDA enhancements to help with quicker approvals? A, complete response letters. B, refusing to file applications. C, sending IRs and DRLs late in the assessment cycle. D, 4-port harmony for IRs. C and D. Number 2, which of the following is not true? The most common mixing operation deficiency was batch records. The filling sealing operation had the least number of deficiencies. The most common drug substance attribute deficiency was pH. Filtration deficiencies did not make the top 10. B, because we know filling had the most deficiencies. And we'll do questions at the end, which is now. Thank you so much.
You are right, Dr. Aldridge. You are absolutely right. Questions is now. So you can go ahead and take your seat, Dr. Aldridge. I'll invite the other speakers up to take a seat for our panel. At this time, with our final session, I'm sure you all know the drill by now. If you have in-person questions, please line up at the microphone so that we can address those at this time. Online, please submit your questions to the Q&A pod so that we're able to start going through those if you've not already done so. And we will get to as many as time allows. So with that being said, anybody in person? Maybe? Sure, of course. See a hand raised if you'll give us a moment here. Not sure your mic is on. Let me hand you this one.
Thank you all for sharing your wonderful expertise today. I think as a member of the audience and also as a healthcare consultant, a lot of us are really interested in what is going to be on the forefront for the FDA as there are more and more Americans that are buying into the supplement market, which I call the kitchen sink market. Yet within the pharmacology space, which I'm also have had much experience with and am interested to see innovate better, there are very few pharmacologic formulations that have conjugates or even vitamins that are included that are required for that medication to be more effective. So what can we expect from FDA in terms of guidances on kind of looking more at some of the supplements and reviewing them, even if you look at the top 100 most commonly sold over-the-counter ones, where those can be checked, whereas the sourcing or the extraction methods could be very toxic to a lot of people, in addition to if they're taken by folks that are also taking medication. So for example, red yeast rice is now being told that for a lot of people that's reducing cholesterol levels, but they may also be on a statin, which would be very, very toxic for them, for example. And on the other side, you know, while we do have some medications such as hydrochlorothiazide, triamterene that were co-formulated together to hopefully get something that's a little bit safer for some people, there's still such a large way to go in terms of bridging that gap and getting these geriatric patient populations away from taking 10 to 12 pills at breakfast and maybe 15 before bed or something. You know, in some cases it really is that high, just to kind of make some of these co-formulations a little bit more palatable and easy for them to maintain their regimens safely. Thank you.
Should be on for you.
Thank you. It's a great question. I think one thing that you've got here is the question is very specific towards compounding and supplements and things like that, which are regulated very differently than generics or brand name drugs. So unfortunately, it's slightly out of scope of what the scope of this sort of discussion is. So I don't think we're necessarily have the expertise to answer that. I think within the aspect of the generic industry, and especially with a generic drug, one thing that you get there is the formulations need to be quite often they're regulated, as you've heard of the last couple of days, very similar to what the brand name is, as well as it has a lot of information based upon that. So the generics have a lot more in that sort of space where you don't have necessarily that those differences and those considerations there. But I will say, like I said, I think one thing is it's a great question. I think it's unfortunately kind of outside of scope of what our experts will be able to talk about because we're especially within the generic sort of space. But you can submit those information, I think, probably to the docket or there's other sort of areas in which to engage with. And offline, too, if you want.
Thank you for that, Dr. Kozak. If there are any other in-person questions, you can go ahead and line up at the microphone. If not, we will go ahead and transition to the online questions at this time.
Thank you. First question is for Dr. Dan Damoudi, and it says, whether the AUC Infinity be removed as a target metric in accordance to M13A?
Thank you for that question. Yes, ICH M13A guidance provides recommendations of AUCT along with CMAX. However, at this point, we consider case-by-case AUC infinity calculations depending upon the drug product and the risk classification of that drug product. Thank you.
Thank you. Okay, next question for Dr. Liu. The types of surrogate matrix vary widely. Is it recommended that depleted biomatrix, such as strip plasma, is a better choice than artificial matrix, such as BSA buffer?
That's a good point. So again, the surrogate matrix, there's no better or worse one. It's more like a fit for purpose, like shown in my examples. Some of them are using the simple buffers, which can do the work. In other cases, a depleted matrix is a better choice. However, just I want to reiterate one thing is even though for a simple buffer, it's easier to prepare and usually it is cleaner, if you can say that, it's cleaner in terms of the interference of the endogenous drug. They usually behave slightly different from the authentic biological matrix, and additional work or efforts should be made to kind of fill the gap during validation, method development and method validation to make sure the matrix effect difference are not impacting the quantitation accuracy and so on.
Thank you.
Thank you. Another call for anybody in person if you have any questions. If not, we can go back to the online questions in the interim.
Okay, so this inquiry is addressed to Dr. Rhee. It's a few sentences, so bear with me. Can the sponsors submit information for an alternate drug product manufacturing site in response to a complete response letter issued during ANDA review when the originally proposed drug product manufacturing site has significant quality-related deficiencies, such as cGMP violations resulting in an OAI status identified after ANDA's submission? That's the question. And then, if so, what would be the applicable FDA goal date for review of the amendment? Thank you.
Thanks for the question. So if I understood the question correctly, so what's the timeline for the amendment to be reviewed?
That's the second part of the question. And the first part is, can the sponsor submit information for an alternate drug product manufacturing site?
Okay. Yeah, if for whatever reason the original facility is in bad shape, probably under OAI, to expedite the approval process, it is allowable to propose an alternate drug product site, which should be in good standing in terms of cGMP requirements. They need to demonstrate that they are capable of manufacturing at least exhibit batches for evaluation, because that's something that we are going to take a look at, of course. And then during this time, until the action is taken, the site should remain cGMP compliant. Otherwise, your application will be, again, CR'd, right? The answer to the second question, there's a GDF letter that detailing all this timeline for an amendment of your ANDA could be approved based on the need for a PAI or inspection that may be longer than if you don't have, then the case where you don't need a inspection. But I can't really remember, is it 8 months? 10 months? Yeah, 10 months if an inspection is needed, otherwise 8 months. Yeah.
Thank you very much.
One thing I want to add to that, one of the questions that came in was, do exhibit batches need to be manufactured under cGMP? Yes, they do.
Excellent. Thank you for that. It does look like we have a question in the room now, whenever you're ready.
Yeah, this is kind of a follow-up question, but slightly different. So if you are in the process of filing an application, your stability data is generated with the site, you have done your bioequivalence studies, and just before filing, your CMO terminates their agreement. And so obviously you have complete set of data, but the CMO is no longer willing to manufacture for commercial purposes, but they are at least willing to support the submission. So if we have to find an alternate CMO for commercial purposes, can we submit that application with at least one additional batch manufactured at a new or alternate CMO site?
So in terms of the number of batches that is required, I mean, that is probably you need to work with the agency. There's no required number of batch that you need to provide. I think it's understandable that, producing one batch is enough, but it may not. So it is recommended to communicate with FDA.
So what would be the best way to request that confirmation or guidance?
So one way to do it, maybe through a control correspondence, if your application has been CR'd, or maybe meeting request, right? That would be another venue. Yeah, control correspondence would be the best option.
Okay, thank you.
Excellent. Thank you for that response. And we'll also, again, invite anybody to the microphones in person. If not, we'll go ahead and go to an online question at this time.
Okay, online question for Dr. Aldrich. An important part of the fill-in and seal-in operation is visual inspection. What general USP chapter covers visual inspection of injectables?
If it's an injectable, start with 1790. You can also look at USP Chapter 1. Those are two great places to start, and they may refer you to others along the way, because 790 is for visible particulates. So 1790, 790, and 1 is a place to start.
Thank you very much. Yeah, thank you. Okay, any in-house?
None in-house.
Let's see if we could get another round in there. Dr. Dan Damudi, if the generic manufacturer incorporates pH modifiers comparable to those used in the RLD, can the product be considered low risk?
Okay, thank you for that question. Again, it's case by case. However, we encourage the applicants to provide the scientific rationale and the data behind not conducting that one of the study, whether it's a fasting or fed BE study, and no difference in the formulation, how the outcome will not get impacted. Thank you.
Thank you much.
Okay, and I think we have another question here in-house. The microphone should be working in the back there by you.
One of the most important parts of PO formulations is testing whether or not they can survive in various, as you said, fed or non-fed conditions. Have there been any other studies, even in the surrogate plasma or human plasma experiments, where they've looked at things like metabolic acidosis or potentially patients who might be at risk for DKA to test them under those conditions? So if under those conditions, a formula that is absorbed even with 80% bioavailability, what is the likelihood that there could be toxic metabolites that can then harm the human who is taking those medications? if those are not tested ahead of time, there might be unforeseen circumstances and toxicities experienced by those patients.
I can start. Yang, you can add, Nide. So we are talking about generic applications, generic products. So that means the NDA has shown about the safety or tolerability of this drug substance as such. So now when it comes to the BE studies, what we are evaluating is that formulation differences that exist, whether it has any impact on the safety and efficacy, right? So in that aspect, the BE studies are meant to compare the test formulation outcome with the RLD, so that outside it's a switchable can happen. But if you are talking about metabolites and toxicity, that era was already done, have been in the NDA phase itself. So we have some predictability on how API behaves.
Right. I mean, I looked at a bunch of INDs and ANDAs, and I worked at an IRB, and we had successfully passed our FDA audit at the time. So I had pretty much read all of those protocols during my time there. Very fascinating. And I don't recall them specifically ever requesting them to check for patients who are in metabolic acidosis. And that can be a whole slew of different patients, ranging from something like DKA to something like other medications that they're on to even sepsis. So if we're not requesting that they check under acidotic plasma conditions for them to check what those medications are capable of doing as a metabolite, if we don't have the RLD data on that because they weren't done then perhaps it might be worthwhile asking them to do them now.
Well, again, tying back to the bioequivalence definition, it's always the switchability, comparability between the test and RLD formulations. If that question is meant to for the NDAs, probably we need to ask the NDAs officers to get more information on that.
Okay, thank you for considering it.
Thank you for that. And if there's no other questions in person, we can go ahead and switch to an online question at this time.
Okay, this question will be directed to Dr. Liu. In method validation, is it recommended to prepare quality control at all levels, low, mid, high, etc., in both surrogate and authentic matrix, respectively?
That's a great point. So usually in method validation, the QC in both matrix types are recommended to prepare at all concentration levels for certain validation parameters, such as accuracy and precision, etc. That is because there will be a direct comparison, not only for the accuracy of the two types of QC performance, but also try to identify any potential issue down the road, just like I mentioned in one of my example. Even though both sets of QC met the accuracy and precision criteria, they are past QCs, but if they perform slightly differently, or in some cases significantly differently, in other terms like internal standard response or matrix effect, etc., it's easier to fix that problem ahead of the time. However, there are certain validation parameters that is not recommended. It's okay to have them, but not required, such as the long-term stability, etc. Just like the regular method, usually a bracketing principle is recommended to have the low and high QC. LLQ-QC is a different story because it is going to be very, very challenging to have a low enough concentration, endogenous concentration, authentic matrix to actually have the LLQC. So the general thinking at this moment is the LLQC is only needed to be prepared in surrogate matrix, not the authentic.
Thank you for that.
Yeah, and I think we have time for at least one more question.
Maybe two. Okay, for Dr. Rhee. Given that incorrectly identifying endpoint of drying is a common deficiency and endpoint determination is highlighted as a key regulatory expectation, what are the FDA's recommendations for establishing scientifically sound and reproducible methods for determining primary and secondary drying endpoints?
So that's a really long question.
One more question,
Could you repeat that question
Sure.
again?
Given that incorrectly identifying endpoint of drying is a common deficiency, and endpoint determination is highlighted as a key regulatory expectation, what are the FDA's recommendations for establishing scientifically sound and reproducible methods for determining primary and secondary drying endpoints?
Okay. So we as an agency does not mandate specific methods to do your development study, as long as you provide us scientifically justified method development, meaning process development, and validate it. And that is something that we're going to look into, whether what you did was adequate enough. So there's no, again, a specific way to do it. There are multiple ways to achieve just one goal. Just based on literature, there are many available PAT tools are available. You can pick one or two based on your needs, specific product or specific process, right? And then give us a full set of data, not just saying that we did this for many years and, here it is. I mean, it's not gonna fly, so, yeah.
And I want to add that Commander Rhee and several other colleagues wrote a paper on regulatory considerations for lyophilized products. Look it up, take a look.
Thank you both. This could be our last one. Dr. Aldrich, what is the suggested guidance for commercial manufacturing process information in ANDA submissions?
You mean the good submissions? Is that the one you're talking about?
Good submissions?
In ANDA exact title.
It doesn't say much more than that.
Good A&D submission practices, guidance for industry.
Okay. And that is the name of the guidance.
Awesome.
I can stop there.
Okay.
Thank you.
I think that is all the questions that we have time for today. So with that, you guys will please help me thank again our speakers and panelists today. Okay. And now for our closing. First, we're going to have a few closing remarks from Captain Kendra Stewart, who's with us here today, if you'd like to come up to the podium at this time. Captain Kendra Stewart is the Director of the Office of Regulatory Operations for the Office of Generic Drugs, whereby she oversees the scientific and regulatory operations. Captain Stewart received her Doctor of Pharmacy degree from the School of Pharmacy at Florida A&M University and completed her clinical pharmacy residency at the James A. Haley Veteran Affairs VA Hospital in Tampa, Florida. She's a lifelong public servant and has devoted her career of over 23 years to various scientific and managerial roles in the federal government, specializing in the areas of health informatics, drug policy, and regulatory operation. Please join me in welcoming Captain Stewart.
Good afternoon. Well, I will not hold you long. Again, my name is Captain Kendra Stewart. I am the Director of the Office of Regulatory Operations for the Office of Generic Drugs. And I just want to take a few moments to thank you all for being here and to close out this year's Generic Drug Forum. Before we close out, again, I want to, on behalf of the Office of Generic Drugs, as well as the Office of Pharmaceutical Quality, sincerely thank each and every one of you for joining us. We truly appreciate the time, engagement, thoughtful questions, and discussions you brought to the sessions over the past 2 days, and we hope the sessions have been both informative as well as valuable. All right, so just as a quick recap, the primary objective of this event was to gain practical regulatory knowledge and to enhance your ANDA applications, streamline the assessment process, and ultimately reduce review cycles. So with that, each of you play a vital role in our shared public health mission to ensure that patients have timely access to safe, high quality and effective generic medicines. So your participation in this year's Generic Drug Forum really reflects your continued commitment to that mission, particularly through the submission of high quality applications that help bring these important therapies to patients more efficiently. As we look ahead, we remain focused on advancing that mission. It is not only a goal, but a strategic priority for OGD in particular to continue modernizing our processes, enhancing efficiency, and fostering innovation and excellence in generic drug development and approval. As a quick reminder, our OGD deputy, Darby Kozak, referenced in his opening remarks this helpful webinar, this helpful web page that includes webinars and other resources for generic drug manufacturers. And so again, I just want to highlight this important resource page for applicants developing generic drugs as part of OGD's enhanced communication with generic applicants to reduce common deficiencies. This resource has an on-demand training, it has presentations and more to help improve the quality of your ANDA submissions and potentially, again, decrease the number of assessment cycles. This is a nice QR code, and it will be available at the conclusion of the presentation, where all of the presentations will be housed on the event page. So you'll be able to reference that QR code to be able to get to the Resource for Industry page. So in general, as we conclude this Generic Drugs Forum, I do want to emphasize the significance of our collective efforts in improving access to generic drug products. Many of us here have likely heard the commonly cited statistics that generic drugs make up more than 90% of prescriptions filled in the United States, or that generic drugs save Americans in the U.S. healthcare system more than $1 billion a day. So with these compelling metrics in mind, consider the substantial impact of our collaboration. By working together to submit and assess well-prepared generic drug applications, we can continue expanding access to safe, effective, and high-quality generic medications. So as we move forward, let's continue to build on that momentum. Together, we can further improve the generic drug development process, addressing emerging challenges and ensuring that American patients have access to the medicines they need. So finally, thank you all for your participation. We hope you have found the Generic Drug Forum valuable, and we look forward to your attendance at next year's event. Thank you. Safe travels.
All right, and with that, this concludes our Generic Drugs Forum 2026. Again, thank you to everybody who attended, who participated online, who joined with us, and we truly appreciate everything you guys do to help us keep going. Again, a few reminders for today. Recordings will be posted to our event webpage once available. As mentioned before, we will put up the YouTube live stream link as soon as possible, and then the official recordings will come once they're available on there. The certificate of attendance is also available on that main one-stop shop page, so you can go on there now and keep your eyes on your inbox for the official thank you email that also has that evaluation link. I just know some people want to get it done early, so you have access to that already. And then from there, that certificate is available for only 2 weeks. Please leave your feedback for us. We always love to learn how to grow and do better. And thank you. Have a great rest of your night and safe travels if you joined us today.